My name is David Leopold, and it's really an honor on behalf of the organizers to welcome everybody here to the two-day symposium entitled Developmental Origins of Brain Circuit Architecture and Psychiatric Disorders. So we're very excited and look forward to a great couple of days. I'd like to mention that this meeting is sponsored by the National Institute of Mental Health, along with funding from the Foundation for Advanced Education in the Sciences, or FAES. I'm going to say a few brief words about the program, but first allow me to invite Dr. Susan Amara to come to the podium. Dr. Amara is the scientific director of the NIMH, and she will say a few words to open the meeting.
Hi, Larry. So I'm really excited to welcome everyone here to this. It's great to see so many familiar faces, and I have to say, it's a wonderful, I think, wonderfully organized program. I feel really excited. I'm looking forward to all the talks. One of the things that I think is important to us at NIMH is the idea of thinking about the basic science of development in the context of psychiatric disease. And really, since I've come here, I didn't really come from a mental health background, but it's become clearer and clearer to me, and I think to everyone, how important and how crucial it is to understand basic and fundamental aspects of brain development in order to really appreciate and learn to begin to address neuropsychiatric disease. And so this symposium was organized by a great group of organizers here, and with the idea of really beginning a dialogue with all of you about building programs and development here, and really thinking about how to use some of the amazing tools and findings that you all will talk about over the next couple of days to really establish and build things here as well. So we hope to seek your input, and we hope to hear great science, and I know we'll hear great science. So with that, I think I'll turn it back to David.
Thank you, Susan. So this meeting was born of a shared and I think growing sentiment that the understanding of development of the brain is essential for comprehending its basic organization and function, as well as the nature of dysfunction in many diseases. In the past 2 decades, research in neurodevelopment has shot forward through a series of transformative discoveries of how brains build themselves. Many of the pioneers responsible for those discoveries are here, and we can look forward to hearing the most recent research in this burgeoning field. There's also been a gradually growing recognition that we need to learn more about the development of the primate brain, and in particular the human brain, where the interaction between genetic regulation, circuit development, and early life experience during a protracted childhood are at the crux of many neuropsychiatric disorders. The program is designed to give a range of perspectives on these topics from leaders in the field with ample time for discussion. So each day is consisting of a keynote address, 2 sessions consisting of 4 talks each. The schedule is shown here. As part of each session, there will also be 3 5-minute data blitz talks from trainees from the labs of the main speakers. The moderators are tasked with keeping the speakers on time through the green, yellow, red system you see here. Red means time is up and it's time for questions. You'll have 5 minutes before the red goes on to tell you that you're getting close to the end of your talk. This is not pertaining to the Data Blitz speakers. For each Data Blitz speaker, we'll probably have time for just 1 or 2 short questions, and for the rest of the speakers, there's 10 minutes of question time. And we really encourage a discussion during this period. In addition, at the end of each day, there's a 30-minute period reserved for discussion. We're going to bring all of the speakers up to the front and in the hot seat and ask them to be interrogated and to interrogate one another, and hopefully we'll have some fun during that period. And throughout the question sessions, we'll be running the microphones around. Right now we have 2. We're trying to get a 3rd so that hopefully you won't have to get up out of your chair to ask a question, but if that doesn't work, you can always line up in the aisle. And all of the organizers and people that can help have these sort of military stars on their badges. So then let me begin by introducing Betsy Murray, who's the chief of the Laboratory of Neuropsychology here at NIMH. And she will introduce the opening keynote speaker.
Thank you, David. It's really a distinct honor, a high honor, to introduce Larry Swanson for this keynote lecture. Larry was a graduate student and a postdoctoral fellow at Washington University at a time when there was really a revolution in anatomical techniques. And early in his career, he was able to work with at least 3 people, probably more, who pioneered some of these new, or then new, anatomical techniques, including Max Cowan, who's famous for his anterograde tracing techniques, Boyd Hartman, a pioneer in immunohistochemistry applied to brain connections, and Hans Kuipers, who was a major practitioner of retrograde tracing techniques. And these all happened in the pretty early days of those techniques and of Larry's history in neuroanatomical circles. Before I completely forget to say something about Larry's academic history, I'll just say something now and then go on to other things. But after earning his PhD and doing a postdoctoral fellowship at Washington University, Larry became an assistant professor in the Department of Neurobiology, I should know this, anatomy and neurobiology at Washington University. He stayed there for several years, then went on to the Salk Institute, where he stayed for about 10 years, and then went on to where he is now, University of Southern California, where he is now a university professor and an Appleman Professor of Biological Sciences and Neurology and Psychiatry. It was during his postdoctoral years that Larry began to study the organization of brain circuits that controlled motivated behaviors. He really went on to dissect anatomical connections, mainly of hypothalamus, relations with amygdala, hippocampus, that underlie our motivated behaviors, including ingestive behavior, reproductive behavior, defensive behaviors, and so on. And one major review coming from this work, I have props today, was this real tome, this review, Cerebral Hemisphere Regulation of Motivated Behavior, which is still a must-read for anyone interested in sort of functional neuroanatomy. It's published in 2000. If anybody wants to borrow my copy, you're welcome to. And anyway, it outlines the basic principles of these networks underlying, eating, these basic processes of eating, drinking, defensive behaviors, reproductive behaviors, foraging behavior, and so on. Another personal favorite of mine is the article by Swanson and Petrovich, a TINS article entitled, "What is the Amygdala?" I have that too. Yes. Also available for borrowing. And in it, the authors point out that the amygdala is really a collection of nuclei that can be determined to have cortical parts, claustral parts, striatal parts, and in their own words, the amygdala is neither a structural nor a functional unit. In other words, there is no such thing as the amygdala. Larry's been part of virtually every important advance in neuroanatomical work over the past 4 decades, and in addition to studying the organization of networks that control motivated behavior, using these traditional tract tracing techniques, he's employed immunohistochemical methods, which for the first time really helped us understand sort of the chemistry of cell groups, and really Larry has written a lot about this. The bigger picture is that these cell groups could be identified with neurotransmitters, neuromodulators, peptides, which when you put it together with the known actual anatomical connections, just expands our understanding of what these cell groups are contributing to behavior. Larry's group has also characterized functionally relevant changes in gene expression and gene expression patterns in the brain. I think people understand now that gene expression, especially in the embryos, is much different than it is in adults. So if you look at gene expression in developing animals, in embryonic animals, especially brain gene expression, it turns out that there are patterns, very similar patterns across many vertebrates. And so these gene expression patterns are almost like biomarkers to help us understand homologs across many species. And for this, he developed computer graphics and database approaches to display and model results. And more recently, Larry's turned to connectomics as a tool to analyze and model nervous system circuitry. So Larry's a prolific, and I hope he'll talk about that today, but we'll see. Larry's a prolific writer. In addition to authoring over 200 peer-reviewed articles, he's the author of 2 rat brain atlases, one for adults, one for developing rat brain. He also has associated graphic files atlases, photographic atlas of the rat brain. He's the author of a book, Brain Architecture, Understanding the Basic Plan, and also 2 translations of books by Santiago Ramon y Cajal. One of those is New Ideas on the Structure of the Nervous System in Man and Vertebrates, and another interesting title, again, a translation from Cajal, is Advice to a Young Investigator. And our, of course, many edited volumes, which you can find on his website. So I won't dwell on honors and awards, but just to say Larry's a former president of the Society for Neuroscience and a member of the National Academy of Sciences. A hallmark of Larry's work is his relentless focus on just the fundamental principles of brain architecture, the architecture of the mammalian brain. Accordingly, I can think of no one better suited to give our day one keynote lecture for a symposium on the developmental origins of brain circuit architecture and psychiatric disorders. So please join me in welcoming Larry.
Thanks, Betsy, for that incredibly nice introduction, and thank you to the organizers for, it's a real honor to be part of this stellar group of people. I'm really looking forward to this 2 days, every single talk. What I want to do this morning actually is give you a progress report, basically an early progress report on a line of research that we started about 4 years ago, and tell you a little bit about how we got into this line of research, and if there's time, I'll talk a little bit about where I think it might be going in the next couple of years. So I think the first experiment I saw when I went to grad school, I was in Eli Robbins' psychiatry department at Washington University, was a freely moving rat. This is not optogenetics. This is back in 1968, polyethylene tubing. But it's a freely moving animal where you can inject nanoliter amounts of neurotransmitter substances into different parts of the brain. And if you're in the correct part of the hypothalamus and you put acetylcholine in, the rat will stop what it's doing and start drinking. And if you put norepinephrine in the same site, it will stop what it's doing and start eating. And this was actually a phenomenon that was discovered by a guy named Pete Grossman, his PhD thesis at the University of Chicago in 1960, published in Science. And it wasn't so much this phenomenon, which is really interesting, that he discovered, but he hypothesized in that Science paper that there are neurotransmitter-coded circuits for different classes of motivated behavior, like hunger and thirst, eating and drinking behaviors. And so actually in my PhD thesis, I found a hot spot for this in the periventricular part of the hypothalamus. And then as a postdoc, started wondering what are those neurotransmitter-coded circuits for different classes of motivated behavior. And that's something I'm still working on. This is pretty much the topic of my talk today. But, you know, here, Betsy mentioned that I got into this just when this revolution in neuroanatomy was, track tracing was taking place. But just to put this in a little bit of perspective, here's a table of known connections of the hypothalamus through history. And by this I just mean region to region, like retina to suprachiasmatic nucleus, periventricular nucleus, macro connections from to gray matter regions in the brain. And around 1940, they had normal silver and myelin degeneration methods, and the reviews at the time listed about 55 connections of hypothalamic cell groups with other cell groups in the central nervous system. By today's criteria, 82% of those were false positive results. Nauta came along in the 50s and invented a nice method for looking at degenerating axons. When he wrote a review of this in 1969, just when I was going into grad school, he listed about 75 connections of hypothalamic cell groups. About 50% are false positives. And if you look down below, you can see the reason for that is the lesions were either too big or interrupted fibers of passage and gave false positive results. So then there was this revolution in track tracing where axonal transport of molecules up and down anterograde and retrograde transport methods were introduced in the late 60s, early 70s. And by 1987, when I reviewed that literature, it had gone up to about 450 connections of hypothalamic cell groups. When we wrote this review article in 2003, it was up to about 3,000 connections of hypothalamic cell groups with the rest of the CNS, and today it's around 10,000. So, when I was in grad school, people thought they could figure out how 50 connections work, and you sort of understand it. But it's gone way past anything that any human being can understand, this number of connections. And so it turns out these connections just to the hypothalamus and these motivation emotion systems involve the entire central nervous system. I'm not going to go through all the reasons for that, but I mean there's initiation of behaviors, there's exploratory behavior, consumatory behaviors, reward, punishment, there's satiety, there's remembering where the goal objects are, so on and so forth. So really to actually explain fully any type of behavior really involves understanding the entire organization of the nervous system. And so the question comes up, here's the nervous system. This is what we're actually trying to understand. A great drawing from Vesalius in 1543. I think it's still probably the best drawing of the nervous system. But, to figure out any system, not just the nervous system, but any system in engineering terms, it's a very simple, straightforward process. You need a parts list. You need to know how each part works, and then you have to know how are those parts interconnected to form a functional system. And it turns out, actually, that we don't have the basic design features for the central nervous system like we have for every other system of the body. It's very easy to teach how the cardiovascular system works or the digestive system works, but the fundamental principles of the nervous system are still actually very unclear. And I think this is a big challenge for 21st century neuroscience to figure this out. And of course, my hero, Cajal, probably said it best. He said, to extend our understanding of neural function to the most complex human physiological and psychological activities, we must first generate a clear and accurate view of the structure of the relevant centers so that the basic plan, the overview, can be grasped in the blink of an eye. So in 2003, Mihai Bota, Hongwei Dong, and I wrote a perspective on the basic idea was we should be using in systems neuroscience all of the tools that are being developed for molecular biology. In other words, bioinformatics. Today, all of those incredible tools we use for transcriptomics, for example, and interpreting thousands of genes, we should be adapting those same basic computer science online programs to understand neural systems and brain networks. And we laid out in this paper sort of what some of the basic roadblocks to that might be. But in essence, coming up with a conceptual structural model for the nervous system. Analogous, really, let's say, to the double helix that Crick and Watson came up with that revolutionized molecular biology. So we started filling in a table, and the key thing here was in 2005, Olaf Sporns coined the term connectome, which is really just a lookup table of connections. And so we started forming a connectome for the rat central nervous system, and there are about 418 gray matter regions in the rat central nervous system. And so you have that list of 418 gray matter regions here, and the same list here. And so really you just look up what the connections of one region are with the other regions. And we started filling this in and with basically data from our own lab. And then decided about 4 years ago to actually do this systematically and try to actually figure this out systematically. And you might think that tables are trivial, but think about the periodic table. And this is Mendeleev's original article in 1866, where he actually, based on a lot of thinking, basically atomic weights, ordered the elements in these rows and columns. And this led to Bohr's, eventually to Bohr's understanding of the atom, to electron configurations, and so on and so forth. Extremely powerful because, first of all, even he, at this period, said that this was predictive. He's got question marks in there on things that should be there that they didn't know yet. It's a testable model, and it's an incredible hypothesis generator, a table of information, when it's arranged in an interesting way. So, every chemistry classroom in the world has got a periodic table, and now it's up to 118 elements. And the other thing you can see is, relationships, when the table is arranged correctly, relationships between certain groups of atoms, okay? Modules of atoms, if you want to think about it that way, communities. So it's a very powerful approach, actually. And what about the elements of the nervous system? And, well, here's one way we actually think about it in a nested way, just to get boundary conditions on how difficult this problem is. At the macro level, this is gray matter regions. There are 418 gray matter regions roughly in the rat central nervous system. But let's just round these things off and say there are about 500. But of course, that's 500 on each side. So there are about 1,000 gray matter regions in the rat or mouse central nervous system. And order of magnitude about the same number in humans. So each gray matter region is composed of a set of neuron types. That's actually what defines a gray matter region, a set of neuron types. And nobody knows how many there are, but if there are an average of 10 per gray matter region, there are about 5,000 neuron types on each side of the brain in mammals, about 10,000 neuron types, sort of at a meso level. And then, of course, each neuron type is a population of individual neurons, and then each individual neuron has a set of synapses. So you can think about this actually in a nested way of thinking about how to organize information about the nervous system. And I just put up, for comparison, rodents and human numbers there. Okay, so this complexity, the revelation to me about 4 years ago was not a revelation to a lot of people, but formal network analysis based on graph theory. And the easiest way, in neuroscience, it really hasn't been applied very much yet. It has been, especially in human imaging, but not so much in basic neurobiology. But looking at airline flight maps globally on a flat map of the Earth, the basic terminology is really simple. Nodes and directed connection. Nodes are the airport's connections of the flights from an airport to another airport. The weight of that is really the number of passengers, let's say. Hubs are nodes that are more highly connected, and Rich Club are very highly connected hubs that are connected with each other more than would be random, and then subsystems within this. So this is just sort of a basic vocabulary that we're going to be using in connectomics to talk about the organization of the nervous system. And so here's our strategy for actually solving the global nervous system wiring diagram, to try and figure out what's the basic plan and a conceptual model for systems neuroscience. So we decided to start with the cerebral cortex, because it's up at the front end of the nervous system, and everybody's interested in the cortex, cognition, voluntary behavior. It's a sheet that's topologically easy to understand. It's divided up into states, basically. And the rest of the central nervous system is topographically connected with the cortex. And we decided to build this connectome for the rat And the reason for that is that by far the most data on connections in mammals is in the rat, by far the most. And we could then use that as a benchmark for other species, for data to come, and at the same time develop the best analysis tools and strategies for trying to understand the organization of this. And then from cortex, just work our way systematically down the central nervous system, basal ganglia, thalamus, hypothalamus, midbrain, down to the spinal cord, and then out to the ganglia and the rest of the body. Okay, so here's what we did, actually. We started with the intrinsic circuitry of the cerebral cortex, the association and commissural connections, which really is the substrate for cognition. And we limited our analysis of the literature to experimental axonal transport methods. We analyzed the entire cerebral cortex, which means the neocortex, the hippocampus, olfactory cortex, subplate parts of the cortex, claustrum, layer 6B, et cetera. And in the rat, there are 77 gray matter regions in the cortex on each side. So that's 154 gray matter regions. We gave a qualitative weight estimate on a 7-point scale to each connection. We converted all of the data in the literature to one standard atlas, the 4th edition of my atlas, because when you have a list, a connectome, you actually have to have an internally consistent list of parts. And we entered this data into a custom Excel worksheet that Joel Hahn developed. And here it is. It's got a minimum of 18 columns of metadata for each connection report. Each connection report, A to B, is a row. Excel goes up to 1 million rows. And as you'll see in a couple minutes, that's not going to be enough. But it's good enough to get started. When there's more than one connection report for a given connection, we rank ordered them based on method validity, injection site location, and the weight. And I collated all of this myself. And really, we're trying to create a gold standard connection database that is online. So everything we've published you can find at this neuroneproject.org website. And we've done the network analysis in collaboration with Olaf Sporns. So just for historical purposes, the first cortical connectome was actually done by a guy named Wendell Krieg, who founded the Cajal Club in 1946. And here's the lateral and medial view of the rat cortex. He had 33 regions and 50 connections between 33 regions on one side of the cortex. Well, you can probably imagine. Actually, I didn't know what to expect when we started this, but here's what we found. We found no statistically significant right, left, or male-female differences in the rat cortical connections. So this applies to actually the adult rat. There are 23,500 possible connections between those 154 regions. We got a total data set of over 32,000 connection reports from 185 articles using 17 different methods. And remarkably, we found data for 94% of all of those possible 23,000 connections in the literature. Only really a 6% empty, no data for the matrix. And we found basically 4,300 connections, macro connections between the 77 cortical regions on one side of the brain, and about 1,000 commissural connections. So the rat cortical connectome is incredibly dense, actually. There are an average of 35 outputs from each cortical region to other cortical regions. And about 37% of all possible association connections are formed. There's data for at least 37% in the literature, and about 9% of all commissural connections. So this is a connectome based on really just topography. It's just the list here of 77 regions is the list in my atlas. It's more or less topographic. And what you can see, sorry. These are the association connections from one side and the commissural connections from this side to this side. And since there are no right-left differences, here are the association connections on the other side and the commissural connections. Well, okay, this is the data. What does it mean? And this is where network analysis comes in and starting to look for communities or modules or subsystems. What is most highly connected with what in this data? And this we published last year, modularity maximization algorithm for weighted networks. The idea is actually that using this approach, there's a spatial index that you can vary through basically between in a reasonable range from 0.5 to 1.5. And when you do that, you find that the most stable solution is a 6-module solution here. And it turns out there are 3 identical modules in each hemisphere, the right and left hemisphere. So of these 5,000 connections, they actually form 3 identical subsystems on each side of the cortex. So at a high level of connectivity, there's a very simple arrangement, actually. 3 subsystems. Now, of course, these are the modules, and there are, of course, less dense connections between modules, but these 3 modules. So the question, of course, is here's the list of 77 regions. What does this mean, really? Well, so I made a flat map of the rat cortex for my atlas, and so we can map these modules onto a flat map. And what you can see here down at the bottom is that there's a very simple, it's not a checkerboard arrangement at all. There are like 3 continents. There's a ring and a shell arrangement to these modules, which are, the blue one in the center would be the core. And what you can see there, that is basically visual, auditory, somatic sensory, and association cortex associated with that, more or less dealing with the external world. The yellow one, which is actually ventral, you can see if you start over here, well, you probably can't see it, but this is medial prefrontal cortex, olfactory cortex, hippocampal formation, and gustatory and visceral cortex over here. More or less dealing with the internal environment, plus the hippocampus. And the green, this dorsal part, you can see in a cross-section up here, basically the cingulate gyrus. This really is orbital, anterior cingulate, retrosplenial, and subicular cortex, which to a first order of approximation is similar to the default mode network people have found in human fMRI and actually in mice and rats. Very hard to define the function of actually, but probably very high level prioritization type things. So these 3 modules have some very, very basic functional significance. And, you know, I just threw this early embryo in. This would be predicting where those are in an early rodent embryo. So to summarize this intracortical connection data, this is an alternative way of looking at cortex than sort of lobes or streams of information. It's really based on overall connectivity. There are 6 topographically non-overlapping modules, 3 on each side. Each region and each module has a unique pattern of connections, and these modules or subsystems have broad functional implications. Okay, well, the next step really is next bring in the cerebral nuclei or basal ganglia. Okay, so actually to look at the connections not just of cortex, and we've published the cerebral nuclei or basal ganglia, but also the connections between those. And this is the end brain or cerebral hemispheres or telencephalon. And don't forget that's a bilateral organization. And so things start getting complicated when you look at this, because you're looking at the connections between cortex and between the cerebral nuclei, but you're also looking at the connections between cortex and cerebral nuclei. So as a matter of fact, just to do this end-brain connectome, there are 16 subconnectomes involved. 244 squared possible connections there. So we published that this year. And here are the basic results for the cerebral hemispheres. No significant right, left, female, male differences. There are about 60,000 possible connections. The total data set, there are about 87,000 connection reports in the literature, 253 articles, about 130 labs are involved. This is getting to be big data, using 19 methods. And about 26% of the connection reports came from our lab. We've been doing this for 40 years, basically. So for the end brain, we actually had 96% coverage of the data matrix. It's only 4% of possible connections there's no data in the literature for. Stunning. The rat connectome is in the literature. We found a little over 8,000 association connections on one side and about 1,700 commissural connections. So again, it's a highly connected network with a mean of 41 output connections per region. About 29% of all possible ipsilateral connections are made, about 6% of possible commissural. So overall, when you look at the whole thing, about 18% of all possible connections are formed. Okay, so a new method came along just this year. It was published by Jube, Sporns, and Fortunato in February. It's called a multi-resolution consensus clustering. And when we applied this to that data set, 4 top-level modules came out. And this is the bilateral solution. So out of all of those 1,000 connections, thousands of connections, there are 4 basic modules. And it turns out 2 of them are identical on the right and left side. And these are actually bilateral. So again, what does this mean? This multi-resolution consensus clustering, instead of looking at trying to find the best spatial resolution, it actually looks at all spatial resolutions and tries to find a solution that actually matches the best of all of them. And when you do that, you come up with a hierarchy, actually, of solutions, a hierarchy of subsystems. And this is really important because you can dig down into subsystems. So at the top level here, there are 4 subsystems or modules. And at the deepest level here, there are 62 smaller subsystems. And the basic idea is that at the top level, you have these 4. You then apply the clustering algorithm to each one of these and find the subsystem within and keep digging down. So here it is on a flat map, and it's not actually the same. And this is a very important principle. The overall organization of communities or subsystems depends on how much of the total circuit you're actually looking at. So if you just start with cortex, you're going to get one solution. If you add the basal ganglia in, that slightly changes. And we're not really going to know the answer until we have the whole nervous system, actually, and see what the subsystem organization is. But at the same time, it's important just to know, for example, what is the organization within the cerebral cortex or within the thalamus, whatever. So you can see here, module 2 is identical on the right and left sides. Module 3, which looks like the default mode network, is the same on the right and left sides. And then you have this bigger module over here, M3 and M4, which are identical on the 2 sides. So what are these? Well, take a look at module 2, which is up in here. And if you can read actually what module 2 is visual and auditory cortices. And so here's module 2 here. And if you look at this, you can see these 3 branches here. And when you cut and look over here, this is what you see. This is this branch. Down here, these are the 10 visual cortical areas that are found, that have been identified in the rat on the right and left side. Over here are the 4 auditory areas on the right and left side. And then you also have these 2 here, which are inferior temporal, posterior parietal, and layer 6B actually cluster over in here. So the idea here actually is that if you're interested in bottom up, you can take any one of these regions here and see what is most highly connected with that particular region. And in this particular case, there are 3 here. So this is a hypothesis-generating engine that if you're interested in layer 6B, the 2 things that are most highly connected with it in the rat cerebral cortex are ectorhinal and posterior parietal. So it might make sense to actually start with looking at the most highly connected parts of the circuit and then working your way out through the tree. So I'm not going to go through these other systems, but system 1 is the sort of the default mode network. And then there are 3 subsystems in number 3. One of them is associated with the main olfactory cortex and nuclei. Another one is related to the accessory olfactory system. And this has a lot to do actually with social behaviors and affect. This one, the 2 most obvious components are the nucleus accumbens and the ventral pallidum, and the central nucleus, the amygdala, possibly most closely related with ingestive behaviors. And then a really big system over here with somatic sensory, gustatory, visceral, motor, and the familiar caudate and globus pallidus nuclei. Okay, what about, I'm just going to briefly mention here hubs and Rich Club, but what's really interesting in the cortex is that the Rich Club and hubs, these like JFK and Heathrow and LAX, form a C-shaped continuous region in the cerebral cortex. And there's one Rich Club back here in the inferior and medial temporal lobes, and it's actually centered in the medial entorhinal, lateral entorhinal area and perirhinal area. If anybody's interested, I can come back to this in the discussion because this is where Alzheimer's disease starts, and we're developing a network model of the spread of Alzheimer's pathology based on this map. The other Rich Club is up actually in premotor and prefrontal cortex. And then in between here is actually some olfactory cortex. And there's only one part of the basal ganglia that's a Rich Club, and that's the ventral pallidum, substantia nominata. Okay, so what's next? Well, what's next is the hypothalamus and thalamus. And Joel Hahn is going to speak for 5 minutes on the hypothalamus at the end of the session. That paper has been submitted. We're writing up the thalamus right now. So this actually is going to be the forebrain connectome. And there are 64 possible connectomes within the forebrain because it's an 8x8 matrix. So there are about 217,000 possible connections in the forebrain. And I think we'll have that done in about a year and a half. The rest of it, well, the rest of it, actually, you actually have to include the body, the muscles. I mean, what is the nervous system doing? You've got to put in the muscles, the glands, the dermatomes, et cetera, et cetera. So as a matter of fact, you have actually the parts of the central nervous system and the parts of the body that it's actually innervating or getting information from. So we call that whole thing, we call it the neuron. And that's about a 2K by 2K matrix at the macro scale. In other words, about a 4 million possible connections, just at the macro level. I don't know when we're going to finish that, but I actually think maybe in possibly 5 to 7 years. So here's overall implications. The rat neurome has about 4 million possible macro connections. It's a dense network. I'm predicting that it's going to probably have somewhere between 10 and 20% of all possible connections. In other words, 400 to 800,000 at the macro level. I think the first draft of the mammalian neurome is in the literature. The collated connections are online. And I think, since I'm here, I put this in, but I think since 1974, NIH has spent about $500 million on the rat connectome. And it is sitting there in the literature waiting for somebody to dig it out. Okay. Despite this complexity, I think there may, in the end, be something on the order of 3 to 5 major subsystems that comprise the central nervous system. A little bit like the 10 systems that comprise the human body: respiratory, digestive, skeletal, nervous, so on. That's the way we're thinking this may actually end up. So a caution: network features. Network topology is not absolute, but depends instead on the extent and localization of network connections. Until we have the whole thing, we can't be sure. The bottom line, though, is the data and methods are available now to create a subsystem hierarchy and top-level conceptual model of the mammalian nervous system structure, function, organization. And I think this macro-level map is a framework for the tidal wave of information that's coming in at higher and higher levels of granularity. So thinking about the future, I can try and justify this in the discussion, but I think that network subsystems predict differential gene expression patterns and differential susceptibility to disease and disorders, like it does at all other levels, at cells, tissues, and organs. There's the genome and neurome analogy. There was so much skepticism about the genome project when it started, and all of the same objections have come up on connectome. Josh Sains and Jeff Lichtman wrote a fabulous article in 2008 called Ohm Sweet Ohm, where they actually compared the objections to the genome and the neurome, and really we've got genome-wide association studies now that are providing insights into psychiatric mental disorders. We are going to have connectome-wide association studies someday, and I think the combination actually of genome-wide association and connectome-wide association insights is really going to be powerful in trying to understand how cognition works. Comparative studies, this very quickly, all the same things you do with genomics, but I just took a flat map of the human brain and said, if what we're seeing in rat is true in human, here's what it would look like, okay? And the same thing on the lower right. Is there a pattern of a pre-pattern in the early neural plate, or neural tube, let's say, that is part of generating these 3 islands of highly interconnected parts of the cortex, for example? This core shell arrangement. Well, and of course the gigantic question is, how does this system actually work? And so, okay, here are the association connections on one side of the rat cerebral cortex, the macro connections. This is from version 1.0. But how are we going to figure out how this system works? This is a huge challenge for technology. And see that little white square up there? That's actually the primary visual cortex, which is by no stretch unimodal. It actually has inputs from 39 other cortical regions from all of the subsystems, and it has outputs to 30 other cortical regions. We have to rethink how we're thinking about parts of cortex and how we actually study these realistically. So here's a quote from an article last year. Reductionistic approaches tend to view individual genes or microscopic features as explaining particular behaviors, whereas in reality, networks of molecules, cells, and larger structures are involved in all function. And I think this is part of the thinking in the future of systems neuroscience. And here's my final slide, just sort of summarize this. You've got networks at all levels. I mean, up at the top is the part of the network underlying LTP, right? It's an incredibly complex molecular genetic network. Down at the bottom are social networks of animals, behavior and cognition. And circuits and wiring diagrams are somewhere in the middle between the molecular and behavioral. And they really, I think, a roadmap of the nervous system is going to provide basically a GPS system for getting around the nervous system, which I think we desperately need, a way for people online to figure out how to navigate all of these circuits. This is going to give us boundary conditions, just like having the sequence of the human genome, boundary conditions for how complicated this problem actually is, 17,000 genes, how many parts, how many connections. And this network model really is, in the end, it's a hypothesis-generating engine to actually design experimental tests of what we know, what we think we know in the literature. And then through a cycle of modeling and experimentation, cycling and modeling, I think we'll get a better and better roadmap, a better and better GPS system for the nervous system, and hopefully begin to understand how to get at these horrible problems, diseases related to mental health. So thank you very much for listening, and thank you, my two collaborators on the project, Joel Hahn and Olaf Sporns, and the Kavli Foundation, which is paying part of Joel's salary. Thank you.
Questions?
Eric?
Yes, so I wonder how much have you taken into consideration the degree of connectivity, and if any information exists on the degree of, I know this is even hard, synaptic strength, because you may have very sparse connection of 1 to 2 axons per neuron versus hundreds.
This, unfortunately, or fortunately, this is a data-driven exercise, and there is virtually no data available on that in the mammalian cortex. And the data that's available is very sparse and nowhere near enough to create a model. So we're actually going at the simplest, crudest possible level of analysis just to get started. It's sort of like looking at the freeway system to begin with, something really big. So it's very crude, very qualitative, but it's really the only level at this point where we can actually have enough data to analyze.
It has some differences in arrow thickness.
So listen, what neuron, and this is in the literature, but anatomists have in the past described a connection as weak, very weak, moderate, strong, very strong. It's a 7-point scale, and we pretty much just take it from the papers and the diagrams. As quantitative data comes along, it can just go right into the spreadsheet. But at this point, there is essentially no quantitative and almost no physiological data. Hopefully, let's say in the mouse visual cortex, we may start getting this kind of information. But to get it for the whole nervous system, I don't know when that's going to happen. So we're just starting really, really crude.
Thanks for an amazing talk. You were saying that the connectomes were about 95% full, basically, in terms of the cells. What did you see when you looked at asymmetry in terms of the strength of connection between nodes? What sort of patterns emerged then? So out being stronger than kind of in?
Yes. And so it's very easy. Just we have a list of, and on one side is the number of inputs and on the other outputs. And so there's a huge range. The most highly connected part of the cerebral cortex in rats is the lateral entorhinal area. And the least connected is more or less the dentate gyrus. Very interesting. And so you can actually look up what that input-output ratio is for any of the connections. And by the way, all the data is online, and so the point is we're sort of almost doing a superficial analysis, network analysis. People can take this data and do whatever they want with it, basically.
So given all the tools available in mouse genetics, what is your feeling about the comparison between the rat and mouse maps?
Well, my feeling is based on data, and there's not a whole lot of data on mouse connectivity. There's very little. I've gone through all the literature. And I think nowadays, if I were to guess, I would say qualitatively they're the same. I think you can, if you wanted to make guesses on what's going on in the mouse from the rat, you'd probably be right qualitatively. But there's very little systematic neuroanatomy going on in mice at the moment.
Hi. So there is a recent study from Kennedy Lab that looks at connectivity in the mouse quantitatively, and they also find that the connectome in the mouse is very dense. And they also found that primary visual areas receive a lot of input from multimodal cortex. So I was wondering if you could have, what do you think about, so in the monkey, the connectivity, the density of the connectivity density it's lower than in a mouse. And primary areas seem to receive less inputs from these multimodal areas. So I just wanted to know what your opinion about this unimodal versus multimodal and how they apply across species.
Yeah, well, there is no such thing as unimodal anywhere, actually, except in the cranial nerves, I think. But Betsy would be the one to talk about primate neuroanatomy. But I worry about the sensitivity of these tracing methods in huge brains, you know. And I could well be wrong, but I think we have to be pretty careful about comparing numbers and density of connections in primates and mice and rats, actually.
So it's very interesting to take your data, to think about clinical conditions like seizures and how you can generate seizures from, let's say, a very discrete region of piriform cortex. And if I were to use your connectivity map, would I be able to trace that all the way out to motor outputs?
Yeah, no, can I get back to my slides here? Maybe just one, I want to show one thing. Can I get back to my presentation?
Yeah.
Okay. Oh, right. Here. So, you know, I showed this map of Rich Club and Hubs, and Scott Small and I were just starting to think about this in the last couple months. And I don't have time to explain this in detail, but we can talk about it later. But Alzheimer's tau pathology starts in the dendrites of layer 2 and 3 cells in the lateral entorhinal area and perirhinal cortex, which is that red area right there, which receive the most inputs from different parts of the cortex. In other words, the most diverse glutamatergic inputs from the cortex, which may end up stimulating this endocytosis, exocytosis cycle and releasing tau from the dendrites of layer 2 and 3 cells. The first place it's going to spread is to the adjacent ectorhinal and medial entorhinal cortex. And when you follow Brack's sequence of Alzheimer's spread, that's the next place it spreads. And so then you get from this region here, following this second wave, then a tidal wave going to the rest of the cortex, basically, and following these pathways. And so but the interesting thing is that it's a retrograde spread, not an anterograde, because the dentate gyrus is not affected early on in Alzheimer's, and yet it has by far the biggest output of the entorhinal area. So the places that really are affected most get it retrogradely. So there's an anterograde input to this Rich Club and then a retrograde spread. Now this is a pure hypothesis at this point, but you have a different organization actually up in the premotor prefrontal cortex, which is where you're going to get some other cognitive problems, but the spread pattern is probably going to be different because the network organization is different. So I think we're going to begin to start thinking about these diseases involving cortex and basal ganglia actually in terms of this overall connectivity.
We have a last question, and then you can hold the question for the general discussion at the end of the day.
Very enjoyable talk, Larry. I received very different distributions of receptors in different rat strains. Do you think that the connectivity is the same between rat strains, or do you think it's dissociated from gene expression of receptors and other things?
Well, now you're getting into the function of these, maps. And I think it's just like Google Maps in a way. First, you need a road map, and then you can start adding cars, you can start adding restaurants, you can start adding metadata to the maps. So in terms of how this network is actually working and how it's differentially working in different animals, different strains, different species, it's all data dependent. You just have to have the data, and then you can model it and start comparing.
Okay, let's thank Larry again.
I know.
Yeah. Okay, good morning, everyone. My name is Suhyun Lee from NIMH, and it is my great pleasure to opening the fourth session of this very exciting symposium. We have the fourth session title is Viewing Brain Architecture Through the Lens of Evolution and Development. We have four main speakers, Paula Alorta from Harvard, Lee Krubitzer from UC Davis, Erich Jarvis from Rockefeller, Susanna Herculano Hausel from Vanderbilt. In addition to these four major speakers, we have three trainee short talks by Joel Han, Lindsay Cantine, and Lin He Zhu. We will have a coffee break after first two talk, and as I said, because of a time constraint, if you didn't have a chance to ask a question, please hold your question until the end of day. We will have a general discussion. So please join me welcoming our first speaker, Paula Alorta.
Well, first of all, thank you so much for having me here. This is a fantastic conference. I'm really excited to share with you some of the recent work that we have been doing. We are interested in understanding how, during development of the brain, this great diversity of cell types and eventually connections are generated, how cells are specified and interact with each other to form the incredible diversity and of functions, the incredible diversity of cells that we find in the central nervous system. And most of the work that we do is done really in mice as the model system, looking at development of the cerebral cortex in particular in utero. However, as you can see from my talk today, I also would like to tell you a little bit today about some newer work that we have been doing, really asking the question of to what extent this beautifully choreographed process of development of the brain could actually happen or be instructed outside of the embryo, which is, of course, is the perfect system in which development occurs, within 3D brain organoids grown from pluripotent stem cells. And I'm excited about that type of work because perhaps, and just perhaps, there is an opportunity to begin to understand some fundamental principles of how the human brain develops. And so I'll tell you a little bit about that today. So the region of the brain we're interested in, as I mentioned, is the cerebral cortex. We all know that the cerebral cortex has an incredible diversity of cell types. There are many, many different classes of neurons that we can classify in different ways. Of course, the projection neurons, which are the excitatory neurons that connect the cortex to all of its different targets through long-distance projections, and these are organized, of course, in the layers. And as well as inhibitory interneurons that connect with the excitatory neurons and with themselves to modulate the cortical microcircuit. And this diversity of cell types interacts with a diversity of glia cells. I'm just indicating here the major classes because we're only beginning as a field to understand what diversity means among these different cell types. But it's clear that there is a great interactions among all these cells during development. Now, we have known about this diversity of cell types, their anatomy, for a long time, but understanding mechanistically how this diversity of cells is generated in the embryo, how progenitors, during this relatively short period of time in development in mice, generate this diversity of cells, that become still an open question. And so what I wanted to do today is to give you just a little bit of an overview, because I have a very limited amount of time, about what we have been doing, how we have been approaching this question of how the diversity of neurons of the cerebral cortex is generated in the embryo. And now we transition from looking at mechanisms that rely on single genes and single molecules to take a more holistic view of this process. And then spend most of the time telling you about the idea of whether some aspects of cortical development could actually be modeled outside of the embryo and how things really work there. So we spent the last perhaps 15 years or so really asking the questions of how the many different classes of pyramidal neurons of the cortex are generated. And we know that in the mouse, all of these neurons are born during embryonic development from a pool of progenitor cells located in the dorsal wall of the developing telencephalon. And by the end of development and then into adulthood, these neurons become organized in 6 layers. And of course, there is a lot of complexity within these layers, whereby there is not a single neuron type in a layer, but there are many different classes of neurons within each layer. And so understanding how each one of these neurons, and I'm only indicating very few here, is generated requires doing something that applies across the entirety of the nervous system, requires the ability of purifying these individual neuron types, not only at the end, but through development from this complex cortex. Try to figure out a way to purify them and then profile them at a molecular level, because there is so much diversity of cells within this tissue. So without showing you any of the primary data, this is all published, we spent many, many years developing methods to sort the cells out of the cerebral cortex using a variety of different approaches, from retrograde labeling of the cells to antibody labeling of the cells to more recently, of course, high-throughput single-cell sequencing, purified the cells, and then identified codes of genes and transcription factors that not only label specific classes of cortical neurons, but actually work at a functional level to control specific aspects of development of these cell types. And this is just one exemplary sort of heat map from some of the work that we've been doing in the past, where we've taken in this specific experiment, 3 classes of neurons: the callosal projection neurons of layer 2-3, the subcerebral projection neurons of layer 5B, and the corticothalamic neurons in layer 6. So these are broad classes per se, but these are the 3. And we've taken them at 4 different stages of their development, shown here by the triangle. And you can see that in each case, we can find scores of genes that are very, very specifically expressed in one class and not in another. And I can show you another heat map where I can show you that these genes also vary dramatically during development. So the signature of genes are dynamic as the neurons basically develop. These not only include protein-coding genes that perhaps are easier to study, but they include a lot of modulatory RNAs, and in fact, every possible imaginable RNA dynamics here is present and controlled differently in the different neuron types. It's extremely complex how molecularly these cells are being made. So if you're interested in this type of data, we have a data set online where you can search by, we've tried to make it as simple as possible in terms of its interface, so you can search for your neuron of interest, your molecular pathway of interest, and you'll pull out all of these genes that we have identified over time. So it's clear that transcription, though, which is pretty much what we were able to do for many, many years, starting with very small population of neurons in the nervous system, transcription only gives you a partial molecular picture of these cells. And in fact, what one would really need to do in order to understand molecular principles of how these cells are generated is to begin to combine information across multiple molecular modalities. So transcription is one, and you can go to very high resolution in transcription at the level of single cells and single molecules, but epigenetics is another. And there are many forms, of course, of epigenetics. But here I wanted to show you a few slides from a recent studies where we have basically taken the information we had on genes that label specific classes of neurons to generate, in collaboration with Josh Wang at Cold Spring Harbor, specific Cre lines that label very specifically, these are Cre ERT2 lines, only some classes of neurons, and use these this genetic approach to purify specific classes at different stages of development as well as postnatally. And let me give you some resolution here. In this specific experiment, we've used these two lines here, the CAX2 CREAT2 and TLE4 CREAT2, to label primarily the commissural neurons of the upper layers and the corticothalamic neurons of layer 4. You can never reach with a single CRE line very high level of resolution, but these broad classes were purified. They're ERT2, so you can use tamoxifen to fate map the cells, and then we sorted them by FACS sorting and all of these different ages that are indicated here. What we did here, though, that in addition to gene expression analysis, we also provide the chromatin using a TAC-seq, as well as did a whole genome DNA methylation analysis. I don't have the data here because it's very new, but we have done all of these also at a single cell level. So each one of these modalities now we have to provide not only the major population, but also the diversity within each of these major populations. And what we see when we do this kind of studies is that, of course, there are genes and epigenetic changes that fall in different categories. But what is very, very clear is that the majority of the changes across all of these three modalities, RNA, ATAC-seq, and DNA methylation, are related to time. So there are huge molecular changes that occur in these neurons as the neurons develop, and less changes between two different classes of neurons. So neurons have a lot in common, at least these classes of neurons, in terms of the mechanisms that they use basically to develop. And so you see here in the yellow and the green, these are shared developmental changes. So changes that change just by time and not by neuron type. While down here in red for the callosal neurons and in blue for the corticothalamic neurons, you're going to have changes that are different in the two neuronal classes. And when we look a little bit more closely about the percentile presentation of these general changes shown here, what you see is that despite the fact that changes over time and over development are the major axes of variation across these three different modalities, if you actually look down here, you'll see that this is very true for transcription. But if we look at the epigenetic modality, there are more changes that are subtype specific, neuron type specific. A way to read this, there is a lot of additional data I'm not showing you here, but the message here is that perhaps epigenetic changes will help us more in understanding differences between neuron types rather than transcriptional changes, which is what we've been able to do thus far. And I think this is very important because we need to focus on these additional molecular modality to really understand these very complex events. Now, one interesting observation that emerged from this recent work that we have done is that because we could profile these two different classes of neurons, both during very early stages of fate specification and establishment of their identity, up to about P7, to stages P7, P21, P48, when they have already positioned into layers and they perform aspects of connectivity and critical periods of plasticity, for example, we were able to ask whether different distinct regulatory mechanisms are at play in these two fundamental ages, basically, of the neurons, where the neurons do different things. And this is exactly what we see. So I'm showing you an example here for ATAC-seq. These, again, are open chromatin regions used by cells. And so if you look at the two neuron classes and you look at the open chromatin region and you consider just the changes in these regions that occur with time. So they're in common in the two neuron types, but they change with time. What you see is that the open chromatin regions that are open early as the neuron are establishing their cell type specific identity are more represented in proximal regions, so in regions that are very close to the start of a gene, so in the promoter regions. Well, if you take the open chromatin regions that are open late, P21, P48, critical periods of plasticity, those tend to be more distal regions and associated more with enhancers. Okay, some more with promoters, some more with enhancer. Now, if you look at the presence of CPG islands, which are also an indication for promoters in the two open regions, you'll see that they're overly represented in the early open, so early development, and not that much represented. So again, this shows that one maps preferentially to promoter and the other one to distal regions. If you look at sequence conservation, so the sequences that are open, and you compare them in different species, this is just a comparison among placental mammals, there is a higher degree of conservation among placental mammals for the sequences that are open early versus the ones that are open later. So there is perhaps more evolutionary pressure on the early one than the later one. And if you look at the transcription factors that bind these sequences, you'll see that these transcription factors are more ubiquitously expressed and used for the ones that are used early and less for the ones that are used later. Let me show you one more slide of this, and then I'll make the point I'm trying to make here. So if you look at the ENCODE project, which is a project that has looked at regions basically of the DNA that are open, therefore accessible to transcription and regulation. And if you look at these regions across a variety of different tissues, this is not just nervous system, this is every tissue and cell line that they basically profiled across the body. And now you take the sequences that were open in our neuron early when they were phase specified versus late when they were going through critical period of plasticity. You will see that these early regions are open, shown in yellow here, across pretty much every tissue that you can think of. Okay, the majority of the tissue use this region to do something that has to do with their biology. But if you now look at the sequences that the brain or these neurons use late through critical periods of plasticity, are pretty much closed in every other tissue. And in fact, they're only open in the adult brain, not even in the fetal brain. What does this mean? We have a lot of work that we have to do, but I like to think that establishment of the building blocks of the cerebral cortex, so the establishment of cell identity of those neurons, is a process that evolution does not want to mess with. So you want to have very concrete, rooted, evolutionally constrained mechanisms that have been validated across many different organs and systems to control these events. And perhaps multiple species do it in a similar sort of way. But now if you look at what the brain and these neurons have to do later on, when the critical periods of plasticity are in place, where they have to strengthen or weaken their synapses and make specific connectivity, perhaps that's the time when you want to have mechanisms that are more unique to neurons and less constrained. Perhaps that's where evolution is going. And I'm very happy to discuss these principles, but I think it's an interesting concept that requires, of course, further investigation. So what have we learned from these early studies that we have done in vivo? And I'm trying to summarize here work that I haven't showed you, but basically we learned a few things. The first is that there is not a single gene that labels a single neuron in the cerebral cortex. There are always signatures of genes that you need to use to distinguish the many different classes, because the diversity is extremely high. These gene signatures are temporally dynamic. So during development, they change like crazy. And so a gene that or a set of genes that may label a certain neuron type later on in the adult may not label it during development, and vice versa. And this is extremely important, I think. Epigenetic mechanisms are extremely important, and perhaps they tell us more about neuron type-specific features than transcriptional mechanisms, and new technologies now allow us to study these things. We couldn't do it in the past, but we can do it today. And that divergent regulatory strategies may be in place during periods of establishment of cell identity versus periods of connectivity and synaptic plasticity within the brain. We have learned another fundamental lesson here, and it's the fact that over many labs and many large efforts, we are cataloging the cell types, molecularly, the cell types of the nervous system. And we're building these gigantic data sets now with single cell resolution of all of the genes that are expressed at a point or another in a given cell type or another. Except I think that to this point, that has not moved us closer to understanding actually how these genes then build the cells that we're really studying. And I don't think that this is a game of single neurons and single genes. This is more of a systems level game now, where you have to take all this genetic information, molecular information that we have, and somehow turning into biological understanding. So I think that there is technology now that allows us to do this. And so this is a project, just to illustrate to you, run by Ashwin Shetty in my lab, who developed an in utero CRISPR library screen. And by that I mean a very large library of guide RNAs, about 8,000 of them. They cover a large spectrum of genes, so you can can make it larger or smaller depending on what genes you want to test, but you can test a large number of genes. And this library is electroporated in utero in mice, in progenitors of the cortex as they make their neurons. And then we can use high-throughput single-cell sequencing to purify the cells that were targeted by these libraries, where you generate basically knockouts, and ask what changes are occurring in response to a certain combination of genes. So the hope of that, it's not by no means trivial because the number of genes is very high and the complexity of the cell types we look at is high. But I think that this is an approach that may lead us close to turning this massive amount of molecular information in the atlases into understanding of the biology of the cortex. Okay, so we are continuing to understand better and better how the cerebral cortex is generated within what I call the perfect system, which is the embryo in utero. Every time the cerebral cortex is made in an embryo, it's always made the same way. That's very routine. That allows you to use the mouse as an experimental system and to understand a lot of things. However, we are moving towards really an era where perhaps we have an opportunity and maybe an obligation to begin to understand the development of the human brain. And this is not just for our own interest in understanding the biology of the human brain, but also because of human brain diseases like psychiatric diseases where you really need a human model. And so some time ago, and I should go back a second. Some time ago, we began work that basically centered around this fundamental question. What aspects of cortical development, if any, could one reproduce or model outside of the perfect system, that embryo? And based on what we could do, could we use this system to begin to understand human disease? Now, there are many reasons why one would like to use organoids to begin to understand human biology. Well, I don't have to tell this audience that the mouse brain is different from that of humans, and their development, of course, is also extremely different. And of course, we have very limited access to this process of development, as it should be, and we never have access to that process, really. And finally, we now have, as of the last few years, a pretty complete picture of the genetics of prominent neurological human diseases, and I'm in particular referring to the psychiatric diseases, where it's very clear that the genetics of these diseases is not simple genetics that relies on individual genes, but is a complex genetics that relies on small variants of small effects scattered across the human genome. And so sort of one is stuck with how you're going to model that kind of genetics within any experimental system. So human models, I think, are really needed. Okay. Now, the question then became, how do you make any model of the human cerebral cortex in the dish from stem cells? And this is really where we got inspired by the beautiful work of Yoshiki Sasai, who in 2011 did, in my opinion, a fundamental experiment. He showed the world, through these beautiful images, I'm going to start this movie, that you can take an aggregate of embryonic stem cells, put them in a culture dish, and over time, these cells will undergo self-morphogenesis, self-patterning, and change to generate an optic cup-like structure, where you can see a neural retina labeled here in green, and it's hard to see behind it, but there is a layer of retina pigmented epithelium. So the power of organs during development to self-organize and self-assemble has been studied, of course, in a variety of different developmental contexts. But for me, at least, this was the experiment that showed us how a very complex region of the central nervous system could, in a way, self-emerge from embryonic stem cells. So there is a power within the early developing nervous system to self-organize its beginning in some sort of way. If you look within this retina, it's very primordial, it's very young, it's very fetal, but you can see that the cells that are generated even layer, in the case of the retina, somewhat within this system. So there is a lot that can be self-organized. So the question then became, if the retina can do that, can one self-organize the beginning of a neural tube and go from there to see what one can generate? And so here, pioneering work again by Yoshiki Sasai and by Jürgen Oblich had shown that you can take some monolayers of pluripotent stem cells, turn them into embryo bodies, and there are a variety of different protocols here, I'm generalizing, that you can use. and then turn this embryo body into neuroectodermal bodies, so the beginning of the central nervous system, and then grow them in different ways in culture for a short period of time at that time. And so to generate structure that contained some of the cells that you normally find in the very, very early human nervous system. So this is when we started this type of work, and by we I say it really was the work of a really talented postdoc in my lab, Giorgia Quadrato. She now has her own lab at USC. And the first question that we ask is, can we see how far we can go in controlling or allowing the development of these organoids in culture? It takes a very, very long time to build the nervous system. And in fact, in humans, it takes the majority of gestations, of course. And that's just to generate the brain of a baby. And then we all know that it takes 20 years of experience-dependent maturation and connectivity to make the brain that we have. By no means we're targeting that process. We're trying to generate some early stages of development and enough cellular and connectivity complexity to be able to use it as a model system. So to do that, Georgia asked the question of whether it's possible to extend the growth and development of these organoids in culture substantially, like for months and months and months, to try to get later stages of development. And so she indeed succeeded at doing that. And in the first iteration of this work, she could grow this organelle for about 9 months in bioreactors. These are flasks where the media spins to allow oxygenation of the tissue. And by 9 months, she could grow them in a healthy way, so there wouldn't be much cell death in the center of the organoids and so on and so forth. And so these basically gave us, it's hard to compare, of course, to the endogenous tissue, but a much longer timeline of development, if development was occurring at all within this tissue. And so with this milestone in place, she really asked, what is within a human brain organoid? It's this very simple-looking piece of tissue. They get to about 4 to 5 millimeters in diameter, and they don't grow past that. And so we first did a lot of immunohistochemistry, but it became very clear very early that if these were anyway close to the complexity of cell types that you find in the early human nervous system, by using immunohistochemistry here, we were not going to go very far, because the diversity of cells was too high. And I just told you that in order to classify really what cell types really are in a system that we don't understand at all and is completely new, we need to use many, many genes and not just single markers. However, even if you stay by these 3 markers, PAC6 intended to be a marker of ventral progenitors, CTIP2 and SAB2 intended to be markers of different classes of pyramidal neurons. What you see here, that at the very beginning of this completely disorganized organoid, you get some sort of an attempt to layer things. So you have a germinal zone here, maybe a ventricular zone-like structure, a subventricular zone-like structure, and then you get a cortical plate that, of course, looks very different from the cortical plate that you normally see in the embryo, but it contains a lot of different neuron types that you see in the embryo. And I should move a little faster here. So we realized that we had to understand what's inside these organoids. We had to profile them at a single cell level. And so we did a lot of single cell profiling, about 67,000 cells at that time, across 20 individual organoids. And here are all of the single cells that are clustered based on how similar the cells are to each other. And you can see that we find many different classes of cells, including cells of the forebrain. And when we went inside the forebrain, we found that some of the major classes of neurons that we find in vivo were present within these organoids. Now, I always stop here and think about how this was a skin cell that became a stem cell that was put inside a bioreactor, led to self-organize for 8 months or so. This was 6 months, actually. And yet, among all of the different cell types that you can make, was able to make a lineage bifurcation decision that gives you callosal neurons and corticofugal neurons. Granted, very different types of neurons, but in the big scheme of things, I think that this is pretty remarkable, and it tells you something about what is written in the genome of the cells to allow this to actually happen. In fact, some of the main cell classes that are present within the fetal human brain were generated within these organoids. We work with Jeff Lichtman extensively to build 3D reconstructions of these organoids. This is an organoid that was rebuilt by EM at 8 months. And what we found is that there are clear features like synapses shown here that are very similar to the ones that you found during late stages of human brain development. And let me me see if I can start this movie. And we did a lot of tracing with Jeff about the features within these organoids. And what was really awesome for us to see is that they could achieve enough maturation to actually begin to develop dendritic spine-like structure, so you can see here, on which synapses could be formed. And this was really incredible for us because this was the first time that we could see actually dendritic spines in a human neuron derived from a human embryonic stem cells, just because it takes so long for these cells to develop. However, there was a big elephant in the room. And I should tell you, and I'm not showing you the data, the neurons become active, they form networks, the synapses are active, and so forth, and so on and so forth. However, the big elephant in the room, and this is what distinguishes organoids fundamentally from brain, is that every single organoid that we looked at was different from the next. Okay, so there is self-organization, but not a single one looked like the next, which is very different from when we study the embryonic brain in utero. And here, for example, I'm showing you 19 organoids. The colors indicate the different cell types that are present within each. And you'll see blue is forebrain. Only 3 and perhaps 4 organoids even form cells of the forebrain. Okay, and here are they're very, very different. So very recently, Silvia Velasco in my lab decided to face this problem. If we don't solve this problem, we can't use these organoids to study anything because they cannot be used as experimental system. And so she set up to do work to develop organoids that would have some features, some fundamental features. One, they had to be able to develop long-term, or we'll only study very early aspects of progenitor biology. They have to generate the cellular diversity. We were focusing on cerebral cortex. They have to do it no matter what stem cell line you start with, and they have to be reproducible organoid to organoid in its cellular diversity. So here are the many, many, many different protocols that she set up and studied and extended to grow for at least 6 months or longer. So she succeeded at that. Each one of these can be grown from multiple stem cell lines. That's not really the problem or the big deal. The big deal is this: can we get them reproducible? So she concentrated on this specific model here, which is a model where you combine self-organization as well as patterning to try to constrain what type of cells you get. And here is what she got. So this is one of these these are 3 of these organoids grown for 3 months from this specific line, the PGP1 line. These are about 17,000 single cells that we sequence, all organized in their TSNE plot. And you'll see that after a lot of classification, we can see that some of the major cell classes that you find in vivo are present here. The beauty of this is that when we looked at individual organoids, they were basically indistinguishable from each other for their cellular composition, which is something that really opened our heart because now we can begin to talk about an experimental system. So here are all the cells together. Here is organoid number 1, number 2, number 3. I challenge you to find the differences between these 3. Can it happen no matter what stem cell line you use to start? The answer is yes. That was an IPS, human male IPS line. This is a female ES line, 32,000 cells. Here again are all of the cell types that you find, which are the major cell types of the cerebral cortex. And one more time, if you look at organoid 4, 5, and 6 at 3 months, they're basically identical. Can these cell types be maintained over time? And can we add additional cell types that are clearly missing from this list here, most notably glia? And so we extended the growth of these cells for 6 months. So now you have the PGP1 line, 6 months, and you'll see that in addition to the cells we had before, now you begin to see astroglia. You also begin to see 2 classes of neurons that we did not expect at all, coming from the fact that these are organoids patterned towards the dorsal telencephalon, so they should mostly form cells that come from the dorsal germinal zone, while now you have interneurons and interneurons precursor that come from the ventral germinal zone. I would be very happy to discuss why I think that is. And I'm basically 2 more slides. And again, look at the 6 months. Despite the long time in culture, now you have the organoids that have exactly the same cellular composition across these all different cell types of the cerebral cortex. You can cluster all of these cells together from the 2 lines and ask how similar a certain neuron made in one line and in one experiment and in one bioreactor will be to the next line, different bioreactors, different experiment. And one more time, you find that they're basically identical. So you can begin to compare across experiments and across cell lines. These organoids grow for extended periods of time, and a major elephant in this room is also that they tend to die in the center as you grow them for a long time. But here, Silvia was able to develop a protocol that took care of that. So this is an organoid cut through, and this is a stain for caspase-3. The outside of the organoid has some cells that die, much like you'll find in a developing embryo. There is some cell death there. But you can see that in the center of the organoid, there is basically no cell death. And if you look at the expression profile of hypoxia gene and apoptosis signatures from all of the single cells, you'll see that there is basically almost no cell death that one can detect here. So where are we going with all of this and why this is important? I think that we have made major progress in understanding how cellular diversity is generated in vivo. And it's clear that from this latest data I showed you that perhaps these aspects of establishment of cell identity can actually be reproduced in vitro, even in a complex systems like a human brain organoid. However, as you have just heard, of course, the computational power of the cerebral cortex does not only come from the cell types that compose it, but you need to actually build the right type of anatomy where cells are organized into layers, where connectivity is precisely orchestrated to allow us to study something functional. And so this is where the field is going, in building that anatomy within those organoids. Should we be able to do this, then we'll be able to really ask what this complex human genetics associated with neuropsychiatric disease really does to the development of the brain, to perhaps understand what goes on in the brain of these patients. So I'll end here by, I think I thank the people that did the work as I was going through, but in particular, Giorgia Quadrato here for starting the work on organoid, and Silvia Velasco, who is now developing these newer protocols. Thank you so much.
Fantastic talk, Paola. But looking at your list of the cell types in the organoid, I didn't see Caja-Rechus cells. Do you have Caja-Rechus cells in these organoids? Okay.
So we have...
And you have subplate, which is also surprising.
Yes, yes. So we do have subplate. The way we have classified them and the way I show them there, it's an extremely conservative way of classifying the cells. So we classify them based on programs of gene expression that the cells have. And granted that we know very little about what are the program of gene expression of the human cell types very early on during development, we're mostly building it on what we know about the mouse. And so I'm only showing you the classification of the cells that I know for sure I'm classifying correctly. This said, corticothalamic neurons at this early stage of development express a lot of genes in common to subplate, although some are different. And so we do find the subplate as well. I just haven't put it there because I don't know if I have enough genes to really call it subplate. The Caja-Rechus cells. So we do have some cells, small clusters, that express the markers of Caja-Rechus cells. Again, they express some of these markers. I don't know for sure if they are Caja-Rechus cells, and that's why I haven't put them there. So it's the conservative view, but both of those classes I think we actually have. Yes.
Oh. Yeah, very nice talk. So I was wondering if you had a chance to just do a comparative study with the organoid RNA-seq data to any fetal brain data set. Yeah. I mean, is there any...
Yes, so I haven't showed the data, but we have done two things. We have taken the single-cell data from our organoid and then compared them to the beautiful single-cell work that Arnold Krigstin did to try to establish correlation at the gene expression level for identity of cells. And that works actually quite well. So this is how we could identify the different types of, for example, radial glia. And in general, because the human data set is not that complete later on, of course, major subdivisions of neuronal subtypes.
So your classifications are based on fetal cell type data rather than adult?
It's built on a lot, yes, it builds on a lot of different data sets. First, all of the data sets that we have through development of the mouse neuronal subtypes. So we have used that because we're very familiar with that. All of the human single-cell data is available, not only the fetal, also the adult. But again, fetal cells have different gene expression profile than adults, and so you have to take it at the intersection of all of this.
Yeah, in your ATAC-seq or epigenetic results, for the adult brain, right, you kind of said, I thought you said that the more distal regions, the regulatory regions, are sort of differing compared to the other tissues, but in your plot, it looked like it was both the proximal promoter regions and the more distal to the gene, that the brain was just different from all the other tissues.
Yes, it is.
Is that right?
So, okay, so two things. In both the early neurons that we purified and the late ones, of course, you have a representation of open proximal and distal, but the proportion of the two, the relative proportion of the two is different in the two ages. So early on, you have much more proximal than later on, okay? And proximal really, it's like these sort of more basic forms of gene regulation that rely on promoters and the classic mechanisms that we have all studied in graduate school, while the enhancers are much more complex and allow you to regulate things in a much more sophisticated way. So I find it interesting that later on, when you do perhaps critical periods of plasticity, you use more enhancers. And then the second question that you say, when you look at ENCODE, right? And so the late open seems to be very unique as a whole, more unique to the adult brain than the other tissue. What that means, I don't know, but I think it tells you something about the uniqueness of what the brain has to do and perhaps the evolutionary pressures on that has to do with plasticity, I think. Oh, okay.
So in mapping adult human cell types back to mouse, where we have a lot of data, basically, would you expect that we would find more similarities in transcription factors that regulate the early stages of development rather than the membrane proteins that are expressed later
That would be my prediction. So we have only started a little bit that type of work by purifying out of the human cortex using an antibody method. So we develop a technology that allows you to dissociate the cells, and then you fix them, you make little holes in the membrane, and you have antibodies going in, and you can sort, based on antibody combinations, specific classes of cells from any tissue. And so we have applied that to the human developing brain, and so we have pictures, molecular pictures of purified neuronal classes of the human cerebral cortex. And now we've compared those to the same classes in the mouse. The thing is that we have completed the transcriptional analysis because this work started a long time ago, and only now we're doing the epigenetic comparison. But at least at the transcriptional level, it seems that some of the fundamental, core genes that define the identity are the same, and the ones that change are other genes that we don't fully understand what they do. But that would be my prediction.
We'll have a last question and then move on.
Hi, Paula. That was a really exciting talk. Hi. So I'll just what's going on with those interneuron precursors and the mature interneurons?
Yes.
What do you think is going on there?
Do we really open that kind of worms? I actually think that is very likely simply due to the fact that you have these early, let's call them embryo body type of things, that you lightly pattern, because this is still a system where I would like it to have some self-organizing capacity. You lightly pattern by inhibiting Wnt and TGF-beta to go dorsal. But of course, not every single cell in that aggregate will receive the patterning. So it's possible that there is a very small percent of cells there that are not patterned. And then if you let these organs, you don't see at the very beginning, but if you let them go for 6 months, eventually this will emerge. Now, what is interesting though, and so that would be the most simple explanation of that, and I think that's what's going on. It's interesting though, one, that we consistently see this. Two, that we primarily see interneuron progenitors and sort of immature interneurons. We cannot say that they're cortical, but so there are a lot of other cells that should emerge. If this was a bit of loss of imperfect patterning, so I don't know, but they are there. It might be an advantage from a modeling viewpoint, because now you have the excitatory and the inhibitory if they continue to develop. But that's what we see.
Let's thank you, Paula.
Thank you so much.
Our next speaker is Leah Kribitzer.
Yeah, thank you so much for inviting me. I thank the organizers, although I'm really not crazy at all about having to give a talk after Larry and Paula. So this is going to be like a serious Fred and Barney talk compared to that kind of technology. However, I'll do my best. So I think Larry raised a really good point when we're trying to understand brain evolution and brain development, that oftentimes we just don't think about the body. And the two are enslaved by each other. The brain and the body are enslaved by each other. They are intertwined, and one can't work without the other. So the portion of the brain I want to talk about today is the neocortex, and we've already talked about this a little bit. One of the reasons we're interested in the neocortex is it's a part of the brain that's involved in higher cognitive functions, perception. In humans, it's involved in language. Also, if you do comparative studies, it's a part of the brain that's changed most dramatically over the course of evolution compared to the spinal cord or the brainstem, and I would even argue the dorsal thalamus. The question in our laboratory is a very broad question. What we're interested in is how you go from a very small mammalian brain that existed some 200 million years ago with very few cortical fields to something that's very, very complex. So how does evolution build a complex brain? And there's a problem with this approach, and that is that we can't study evolution directly. So there are two ways we can circumvent this problem of not being able to study evolution directly. The first is we can do a comparative analysis. We can look at what has evolution produced, and evolution has produced bodies and brains. And we don't want to just focus on a few species. If we want to know, we're not interested in the brain. We're interested in this brain thing, or brains, overall. So you want to look at a lot of different species or a lot of different products of evolution. So you use a comparative approach, and so you can study a lot of different mammalian brains. And we do electrophysiology, neuroanatomical tracing. We're starting to do some molecular work and starting to do really, really hard stuff, which is behavioral work. And by doing this, we can understand what has evolution produced. It's produced homologous networks. It's produced homologous areas. It's also produced specializations. The problem with the comparative analysis is that it doesn't tell you how phenotypic transformations occur. So in order to understand how phenotypic transformations occur, you have to study development. Because although Zoltan said he said this first, the evolution of the neocortex is actually the evolution of the developmental mechanisms that give rise to aspects of the phenotype. But I said that a while ago. But then he said Ramon Cajal said that. Okay. So what I'm going to focus on today predominantly is the how part, but I do bring in some comparative work. And so our comparative analysis, so over 30 or so years of doing comparative analysis, we can generate these sort of cladograms, and these are meant to, the different colors indicate different cortical fields, so visuals blue, somatosensories red, auditories in yellow. And what you'll see is that over the course of evolution, there's a common plan of organization that has been retained from the common ancestor. And this includes this constellation of cortical fields, some of which I just pointed out. What's really interesting is that you can't get rid of aspects of this plan. So even in something like a blind mole rat where the skin has grown over the eyes and the eyes have gotten very microphthalmic, you can still identify a geniculocortical pathway. You can still identify a V1. V1's been co-opted by the auditory system. You have changes in subcortical connections, but there are aspects of the network that you just simply can't get rid of. You shrink it down, and you can co-opt it at different levels of the nervous system, but it still remains. However, there are really large changes that we can describe as well. So this is a macaque monkey, a flattened view of the macaque monkey. Left is to the... rostrals to the left and medials to the top, and this is a mouse brain. And what I'm showing you is similar aspects of organization, so a V1 and S1, posterior parietal cortex, auditory cortex, but there are a lot of different changes. There are changes in the size of the cortical sheet, there are changes in size of the relative size of cortical fields, there are changes in the number of cortical fields, or the addition of cortical fields. There are changes in cortical field connectivity, and I'm just showing basic networks here, Larry. But I want to point this out for these mouse people, that this is not to scale. This, if I put a mouse brain in a macaque monkey brain cortex, that's about the space that it would occur, or the space that it would occupy. Okay, so the questions are, to what extent are these differences in the cortex associated with genes intrinsic to the developing neocortex? And I think a lot of people are going to talk about that today. Genes associated with the development of the body plan, which people don't think about too much. Epigenetic influences on the body and the brain. Sensory stimuli present during development. and the use of specialized morphology or sensory receptor arrays. And what I hope I can convince you of by the end of this talk is that it's actually some combination of these factors. And for different species, it's different combinations. And for different individuals within a population, it's probably some different factors as well. Okay, so this is just an overview of my talk. I'm going to give you some examples from the natural world. Experimental manipulations in peripheral morphology, a relatively recent study we started called Laboratory Rats Gone Wild, and then I want to talk a little bit about natural variation in early sensory experience. So changes in peripheral morphology, this is a really old study, but it's still one of my favorites. This is a duck-billed platypus. It has an extreme morphological specialization of the bill, and it has electrosensory receptors that run in stripes along the bill, interlaced with mechanosensory receptors, so they're striped like this. And when it's doing anything important, when it's navigating, when it's feeding, when it's mating, it closes its eyes, its ears, and its nose. So it's one big, huge, enormous bill. And if you expose the neocortex and do electrophysiological recordings and explore a lot of the neocortex, what you find, if you look at the platypunculus, and this is just a rendition of a lot of electrophysiological recordings, is you have a huge representation of the bill. So let me just point this out to you. This rostrals to the right, medials to the top. This is somatosensory cortex. This is a representation of the body, the hind limb, the forelimb. This is a representation of the bill. And the green and blue are interdigit representations of mechanosensory receptors and electrosensory receptors. So about 90% or more of S1 is devoted to processing inputs from the bill, and then you have additional representations of the bill. So I'm not going to, like I said, I'm not going to talk about changes, genetic changes that occur during development that account for like some of these sorts of changes that we see in the platypus. So the question, of course, is to what extent is this enormous magnification of the bill in the platypus the result of genes intrinsic to the neocortex that are programming this, genes that are extrinsic to the neocortex that are involved in specialization of the bill and the sensory receptors on the bill, the environment in which the animal or the environment in which the animal is immersed. So what I do want to show you is that there is some good evidence that some of the changes we see in cortex may in fact be due to changes in the body. And this is a really beautiful developmental study by Kritikos and his laboratory looking at early forelimb development in the forelimb of a mouse versus a bat. And you can look at this forelimb morphology and say, okay, wow, that's really different forelimb than this. Here's D1, D2, D3, D4. You have interdigit membranes. You have an elongation of the forelimb. You have the part of the wing attached to the hind limb. So it's a very, very different morphology. And people have actually worked out genetically how something like a forepaw can be transformed into a limb. And one of the things that has to occur is this elongation of the forelimb. And this is just PRX1 is involved in this forelimb elongation, and the spatial location or the spatial extent of the expression during development is very different in the mouse versus the rat. Or, sorry, the mouse versus the bat. I'm a little nervous here. But what's really cool too, and it's a good story to look into, you would think that there are many, many genetic changes that have to occur, and in terms, there aren't. There are about 5 or 6 major players that can transfer a forelimb into a hind limb. But if we look at the cortex, what we see, and we look at the forelimb representation in the somatosensory cortex of a mouse versus a bat, and this is a microcoropteran bat, not one of the big bats, but one of the smaller echolocating bats. What's really nice about this comparison is that the neocortex is about the same size. And if we look at the representation in the mouse, it's really very small. The forelimb representation is very small, and you have this enormous representation of the forelimb in the bat. So the question is this due to simple changes in body morphology and use, something intrinsic to the neocortex, or some combination of both? Okay, so experimental, and so we're trying to get at this with experiments. This is experimental manipulations of peripheral morphology. And what we want to do is something very radical. We want to change the ratio of incoming sensory inputs to the developing brain. And one of the ways we do this is an animal model we use, which is a Monodelphis domestica, or short-tailed opossum. We do bilateral enucleations very early in development. And if you look at what's happening here in terms of this developmental timeline, you see that many of the events that we'd be interested in, like eye-opening or when thalamocortical afferents reach the cortex, when retinal ganglion cells reach the diencephalon, occurs prenatally. And in opossums, this occurs postnatally. So we can physically do a lot of manipulations in these guys that we can't do, that we would have to do in utero or in other species. And plus, and this is what they look like on the day of birth. They literally look like little embryos. They have no hind limbs formed, just their forelimbs. So we bilaterally enucleate on P4 well before thalamocortical afferents have reached the cortex, well before the retinal ganglion cells have reached the diencephalon. So what we've done is we've hugely changed the ratio of incoming sensory inputs to this developing neocortex. And what we find if we do electrophysiological recordings, and these are older studies, is that these blue dots equal recording sites where neurons responded to visual stimulation, red where neurons responded to somatosensory stimulation, and yellow where neurons responded to auditory stimulation. And what we see is that all of what would be visual cortex in the bilateral nucleate has been taken over by the auditory and the somatosensory system. We also see if we do an architectonic analysis, this is, I have a better slide, but here's V1, and this is architectonically defined V1. Just like the blind mole rat, we couldn't get rid of it. We couldn't get rid of V1. We made it smaller, and it was co-opted by other sensory systems. If we look at the normal connections, and this is just, there are more connections to primary visual cortex than I'm showing here, but this is just some basic pattern where mainly visual cortex is receiving inputs from other visual structures of the cortex and thalamus in normal animals. But in bilateral nucleates, visual cortex is now receiving inputs from the somatosensory system, both cortically and subcortically, and the auditory system. And a recent study by Jimmy Dooley, who was a graduate student in my laboratory, has shown that not only are there changes in the targeted system, which is the visual system, there are also changes in the somatosensory system. In fact, those changes are really quite remarkable in terms of connectivity. So both there are differences in cortical and subcortical connections. And this is just a sort of a connectome showing, a weighted connectome showing those differences. So if we look at the receptive fields in this reduced visual cortex, or what would have been visual cortex, what we find is that almost all of the receptive fields in that reorganized visual cortex are on the snout, the face, the vibrissae, and the head. There are very few on the lower body. So basically what I would contend we did is we sort of made a huge cortical magnification similar to what we see in the platypus, and we didn't have to do anything to the cortex, to the developing cortex itself. We just changed the ratio of incoming sensory inputs really early in development before the nervous system was hooked up. And we also changed, we also saw some remarkable changes in the somatosensory cortex, and this was work that's recently done by a graduate student in my laboratory, Deepa Ramamurthy. And she did single unit recordings in the vibrissae representation in S1 of these bilaterally nucleated animals. And what she found, and these are just the study is really beautiful. She actually found the data analysis is much more extensive than this, but she found that receptive fields were smaller in the early blind animals, and that individual neurons had a better discriminability, which means they can detect stimulation of one whisker versus the surrounding whiskers better than in normal animals. But so all these changes to the brain are interesting and cool, but really what we want to know is it's about behavior, because neurons aren't the target of selection and genes aren't the target of selection. They co-vary with the target of selection, but morphology and behavior are the targets of selection. So if this animal has a huge amount of cortex devoted to somatosensory processing, if we see changes in connections of both visual and somatosensory cortex, if we see changes in neural response properties of neurons, what does this mean for behavior? So this was some of the behavioral work, and it's just about ready to be published. We trained the animals on a very simple Y-maze task where they had to discriminate between a rough and a smooth surface, and then obviously that would be quite easy, and they would always move to the rough surface. And what we did then is we had a base grid of sandpaper. Yes, this is the Fred Flintstone part. We used sandpaper. This was probably held together. This was probably held together with paper clips and super glue. We MacGyvered that. But anyway, what they did then is you would slowly change the grits of the sandpaper, and they would have to tell which one was rougher. And so if you had two very, very rough ones that were slightly different, that was a very hard task. If you had a smooth one and a rough one, that was an easy task. So if it was a very easy task, smooth versus rough, both sided controls and bilateral nucleates did very well. If it was a really hard task, where it was the same texture as the base panels, they didn't do very well. But all the in-betweens, the bilateral nucleates perform significantly better. We also have other tests where we looked at natural behavior. And this is a really nice result. When we trimmed the whiskers of the bilateral nucleates, they dropped, their performance dropped significantly, although not the sided control, suggesting they're actually using different strategies. Okay, so in that portion of the talk, what I showed you, and I took out the slide, and this is, I think, an important observation, that being blind is not simply an absence of light, right? Because if you're congenitally blind, your entire nervous system has rewired itself. Not only has your visual system changed, but your somatosensory system has changed. And we didn't look at auditory systems or motor systems, but I would guess there were changes in those as well. So for the next part of my talk, I want to talk about our laboratory rats gone wild, and it was done in conjunction with Dylan Cook, who's now at Simon Fraser University, Mackenzie England, and Danielle Stolzenberg. Okay, so this is a question that has bothered me for a long time, and the reason it's bothered me for a long time is because I've worked on a lot of different species of mammals. I think of over 45 different species of mammals, and many of them I caught. When I went to Australia to work on platypus, I had to catch them, and it really changes your perspective on the body-brain sort of gig, right? That it becomes really important, because it's hard to catch an animal. They have specializations. They do things we can't do. And so the notion that almost all of our research is studying an animal that is highly deprived in a highly invariant environment was something that we were able to investigate and at UC Davis. And the reason we can do this is that we have these field pens out it's the Puda Creek Riparian Reserve, and this is an aerial view of the field pen. They're 3,000 times the size of a normal rat cage. And now we feed them, we water them, but they still have access to these sorts of things. And so I should explain the experiment that we do. So we take pregnant mothers out into the field pens, and they give birth out in the field pens, and then the young are reared in these field pens. And then we look at a lot of different things. And what's interesting is this is the range of environmental conditions, temperature 43 to 12. So for those of you who have to do IACUC inspections and they're looking at the thermometer in your animal rooms, and it's like, oh, whoa, it's off by 2 degrees. You know, like the heat of the summer, which was like 105, and the cold of the winter, and they were fine. Range in humidity. We even had an earthquake out there. Well, not just out there. It was, affected Davis. They actually have access to predator cues, wild turkeys, raptors. Obviously, the predators can't get in the cage, but their auditory experience is very different. So I just wanted to show this, and we have a number of other behavioral tests that we do. Every single thing that we looked at in these field pen animals was different, including when they opened their eyes, when they began orienting their body, when they started walking. Their nesting behavior was radically different in summer versus winter. So that was really cool. These are ongoing. How they perform in a Barnes maze. They look like 2 different species. Okay. So we were interested in motor cortex. Well, starting to examine motor cortex. We ultimately like to look at visual cortex and auditory cortex, obviously in pigmented rats. But the reason we started with motor cortex, we thought this would be changed quite a bit. I want to show you a little slide here. Oops. Oh, here we go. Okay, here we go. So this is cool. It's not going to run forever, but obviously the motor options available to developing rats are huge. They can move They can move vertically. They use their trunks a lot. They use their hind limbs. They use their tails to stabilize. They're doing a lot of things that laboratory rats just can't do, or don't have the option to do. They're pretty cute too, huh? Anyway, I like rats. Hold on. Okay. So, all right. So what we did is, this is intracortical microstimulation experiments. Each one of these dots represents an electrode penetration, and we put in a small current, and we'd see which sorts of movements we could elicit from the body. And this is a laboratory rat, and this is a field pen rat. And what I want to show you is that you have a relatively large representation of the portions of the face, the movements of the vibrissae, neck, and forelimb. Not as extensive in the laboratory rat, but if you look at the trunk representation, the hind limb representation, it's relatively small, and you don't see a tail representation. In these guys, you see a really large representation of the hind limb mixed with representations of the trunk and the forelimb. And this particular case doesn't show it, but we've actually have a few cases where we were able to elicit movements from the tail, which you could never elicit in a laboratory animal. But this might be a little more telling, this slide right here. If we looked at the percentage of sites in which we can elicit movements of the hind limb and the forelimb, as if the animal's using both in conjunction, we see many, many more in our field pen rats. and very few in our laboratory rats. And if we look at movement of the forelimb and the whiskers in conjunction, we see very, very many in the laboratory rats and very few in the field pen rats. So literally, the laboratory rats are truncated, right? This is what's represented in motor cortex. Okay, and like I said, what we'd like to do is we're going to start looking at connections and looking at organization of sensory cortex. So sure, we changed the phenotype, and this was a more subtle change of the phenotype, but we still changed the phenotype. But there's a bigger question. Can rearing in a highly variable environment alter the ability of the animal to meet a wider range of sensory motor challenges, right? So lights on and lights off, same time every day. They get fed and watered the same time every day. They have the same temperature. The big excitement of the week is to get their cage changed, right? That's it. That is their life. So if you rear in an environment where things aren't expected, or there's a lot of variation in light and sound and tactile experience, do you have a more plastic brain? And if so, there must be alterations to the neocortex that are probably at the level of the synapse. But that's not a question we're working on this second, but I think that model is there to answer that question. Okay, then the final part of my talk is natural differences in rearing conditions or cultural transmission of rearing styles and cortical connections. And so I went from here's what nature has produced, something extraordinary like the duck-billed platypus, and you can see these kinds of cortical magnifications in a lot of different species. Ken Catania's shown some beautiful ones for star-nosed moles, naked mole rats, and you can even look at the supralaryngeal tract of humans, including the tongue and oral structures, and you have a huge magnification in S1, motor cortex, premotor cortex, and that specialization is, people have called it Broca's area, but it's still following the same rules of construction. Then I showed you something really radical, taking a sledgehammer to the system, removing both eyes to radically change the ratio of incoming sensory inputs. Now I just showed you something a little more subtle, which was these laboratory rats gone wild, where we're changing the environment, but it's a radical change in the environment. And this is more subtle still. And all of those, I showed you changes in the phenotype. So I don't know if you know about prairie voles, but they are monogamous, and biparental. Only 3% of animals are monogamous. And so both parents rear the young. And what's really cool, like anything in the natural world, the way the parents rear the young falls under a normal distribution of a variety of different behaviors that the parents produce. One of the behaviors that the parents produce that falls under this natural, bell curve is the amount of tactile contact that the parents have with the young. And the reason I like that is because you can measure it. It can be quantified. That you can quantify the number of touches. And most of those touches are around the face and perioral region. And so we have low contact, high contact, and all those animals in between. And we just wanted to look at the high contact and the low contact offspring of these parents. So here's what's really cool. You can read that yourself. But here's what's really cool. The cross-fostering is really cool. So if you're a high-contact baby, you become a high-contact parent. And if you're a low-contact baby, you become a low-contact parent. But if you swap the babies on the day of birth and you put a low-contact baby with a high-contact parent, they become a high-contact parent. So this is a cultural transmission of a rearing style that we can quantify, which is really cool, right? Okay, and the first thing we wanted to look at was somatosensory cortex. So this is electrophysiological recordings in somatosensory cortex. Each one of these, well, she took the dots out. Anyway, what we did is we focused on the representation of the perioral structure, and this is how we mapped it out. And this is really early days yet, so we have 4 animals in each group. But what we're finding is sort of what we expected to find, that in terms of body representations and receptive fields, we had a much larger percentage of S1 and S2 devoted to representations of the face and this specialized perioral region in the high contact versus the low contact. Okay, so we looked at connections, and this was, I wish I could show you, more of the data. This was published by Adele Selke about 3 years ago, but this is a really hard study because we wanted to match the injection sites in the same representation and make the injections of a similar size so that we can quantify these connections and compare them. So if you look at the overall pattern, this is a high contact, this is a low contact, and you say, okay, overall these look relatively similar, but if you look a little more closely, you can see that there are differences in density of connections. And you can see this, and and some are very extreme. And Larry, our lab does quantify connections, but I think we might be the only, I'm serious, there are very few people who quantify connections. And there are ways to do it, but it takes a lot of time, and you have to do full reconstructions, and you can't just do photographs of the brain. And that's why I think a lot of people don't do it. Anyway, if you quantify these differences, this is the percent of connections, intrinsic connections of S1. I'll just give you this example in a high contact versus a low contact. So how to read this is that the high contact has about 12%, 12 to 14% more intrinsic connections of S1 than low contact. Low contact has about 10% more connections with motor cortex. If they were really similar, this line would be, they both would be about right at this line right here at 0. So you can see that you can actually measure some of these differences. Okay. So I'm going to end here with a little summary. What factors contribute to the cortical phenotype? Because, the question used to be, how does evolution produce a complex brain? But evolution is about genes. And so genes are just a part of the story. So genes, and they can contribute to cortical field size, cortical sheet size, cortical field size, cortical connections, peripheral morphology. Very important. Genes can contribute to cellular mechanisms involved in plasticity. So what's not inherited is the phenotype, but this ability of the brain to match the environment in which it finds itself, which then allows the environment to impact phenotype. And what's cool is that the environment can change the same thing as genes can. It can change cortical field size, it can change cortical field connections, it can change peripheral morphology, and I gave you the example of a bill of a platypus. And I talked a little bit about the tongue larynx lips of a human. And if we want to start talking about things like social learning and language and culture, I think that we should treat these as simply as complex patterns of physical stimuli that are impinging on the developing nervous system throughout development, and they vary over 2 time courses, the very long time course of evolution and the shorter time course of individual development. And this is a sort of history of humans, where morphology is at the top and environmental and social context is at the bottom. And there's some beautiful work from Chet and I can't remember the other guy's name. Chet, what's it? Yes, yeah, you got to give him the shout. That shows that the aspects of the human hand have been around for a really long time. So this baby's been around for a really long time. And we weren't doing very extraordinary things with our hands. We had large cutting tools. We started a tool use tradition. The modern hand has been around for about 700,000 years. And a big, huge human brain, and this is work done, Marta Florio showed this in a really nice review recently. The big, huge brain's been around for a huge time. So we had the hand, we had the big, huge brain. Where's the serious tool use culture that we have today? Modern humans have been around for 300,000 years. And so I want to blow up on this. So we had the stuff, but we weren't doing the behaviors, the sort of sophisticated behaviors we're doing today. 300,000 years ago, we had spears and knives. 70,000 years ago, we had, flake tools and relatively unsophisticated technologies. This is less than 300 years ago was the Industrial Revolution. 300 years, that's extraordinary. And then all this stuff snowballed. Automobiles, electronic computer, 1950, 1990s, World Wide Web. And today we have daily and prolonged use with machines. So if you believe that all behavior is generated by the brain, and I believe that all behavior is generated by the brain, and the behaviors changed so radically in the last 300 years, or certainly the last 50,000 years, then the brain must have changed. But it didn't change by changes in DNA sequence. It has to be these other sorts of things. And so I'm going to end with humans, even though I don't work on humans. And I'll read this because we are combinatorial creatures constructed by genes, bodies, behaviors, and environmental context. And what I think makes humans really extraordinary is our capacity to construct our neocortex, or reconstruct our neocortex, or create our neocortex over successive generations throughout a prolonged infancy. So that, what I mean by that is if you took my context now and you put me back 300,000 years ago, I wouldn't be Leah Krubitzer using a spear, right? It would be a totally different brain. It would be a totally different brain if I were born 20 years later. It'd be a different brain if I were born 2 days later. Okay. We have a remarkable fluid brain-body interface with the environment such that tools and machines can extend our embodiment in peripersonal space and expand the loop between our brains, our bodies, and the world. And I think this has made us into unique biohybrid creatures whose brains adapt and bootstrap themselves with the technology they have given rise to, and with whom our futures, for better or worse, are increasingly entwined. Okay, so this is my lab. I think I showed you individuals throughout the talks to give them proper credit, and thank you.
In the case of the within-species manipulations you did for the opossum, and to some extent also for the rat in the different circumstances, when you see the major reorganizations in visual cortex, for example, is that, do you think, all attributable to elimination of exuberant projections, or is there new growth or something else?
So that's some of the stuff we're working on now, because I don't know. It could be either. It could be that you fail to prune existing connections, or you've generated brand new connections. So we don't know. That requires us to do our dye injections and stuff like that, which we're doing.
Yeah. So along those lines, it makes me wonder if you're losing brain space for other traits as you gain, like in this case, greater tactile stimulation on the or contact on the oral facial area. And even taking it further to well, in that study, have you looked at whether other brain areas aren't changing their representation or losing? And the other is, if taking it further into human evolution, you can think further, are we losing something by gaining all this additional technology or ability to process all this additional technology?
So we did do brain measurements in the opossums, and somatosensory cortex actually gets bigger, and visual cortex, or architectonically defined visual cortex, gets smaller. The answer to your question is a little bit more, okay, you're not changing the size of the cortical sheet. I should have said that. The size of the cortical sheet hasn't changed, but how it's functionally organized has changed. So yes, some things are being gained, and some things it's not just this addition of stuff. And so, in terms of all this technology and how it's changed our cortex, there are two things I'll say to that. One is, I think, the portions of the cortex that are actually really, really more built during development are things like posterior parietal cortex and prefrontal cortex and temporal cortex. And there's I think, some really good evidence that suggests that's so. And those areas, depending on what your context is, are going to change their functional organization. I don't know if that's better or worse. So when you say things get subtracted or things get added, it sort of puts a value on it. And evolution doesn't have values, right? It's not good or bad. I mean, we might go extinct, and I guess you could think that's bad. But as evolutionary biologists, it's not bad. It's that's what happens. Yeah, I didn't answer your question, but I hope maybe. Okay.
So I'm right in front of you.
Oh, okay.
Hi. That was cool. I've a question that was prompted by the way I think you formulate the patterns you see, which is that essentially you can have peripheral changes in the body that are driven by selective pressures that have promoted those new body features that are then driving changes in brain structure. And I'm wondering if you could think of, pushing the analogy further, the brain itself as a physical structure. And are there examples, can you conceptualize the disproportionate increase in the primate brain relative to body size as itself a kind of body part that's expanded that is then causing changes in brain function, as it were?
That's cool. That's interesting. I may not be the best person to answer the disproportionate increase in the size of the cortical sheet. We're doing some of that stuff, and there's no question. It's not just that the neocortex has expanded relative to the body in primates and in humans, but relative to the rest of the brain.
Right.
Yeah. And you're saying, does that physical change in the size of the brain affect how the brain functions? I think, one maybe good example is, vascularization of the brain. So if you have this enormous expansion of the cortical sheet and you look at cortical vasculature in lissencephalic versus gyrencephalic brains, what you find is that major blood vessels are straddled between the gyri, right? So if you have a larger brain, you have to feed it, you're going to have the mechanical pressure of blood vessels pounding on the neocortex. So a physical change I think is changing the vascular, and if that in fact changes aspects of how neurons work, in terms of blood supply, I don't know. I mean, I'm just
sort of imagining more
saying
like
stuff.
You have a new big chunk of brain that develops that causes changes in other bits of brain as a result of that new brain.
Oh yeah. Oh yeah.
And in that sense, you can think of these consequences, sort of connections and so on, as not carved between brain and body, but as sort of interactions between body parts. It's like, the part of the brain and another part of the brain that's driving. If you have a new brain structure that evolves, and that new brain structure causes a functional change in another brain structure, you could have evidence of that. So, for example, you have a bit of brain that evolves to handle the muscles that have formed in the larynx and throat and so on. As a result of that, you got a brain change, and that brain change then brings about a change in cognition to drive those structures, like
right.
for language.
And when you have that sort of change, you're gonna retain aspects of the cortical networks that are homologous, but that's probably going to also generate changes in cortical connectivity and changes in network function and changes in cognition. Yeah, sorry, I thought you were asking a different question.
Hi, beautiful talk. Just a question about when you talk about after removing the visual input, the V1 shrink to a much smaller cytoarchitectonically defined region. So I'm wondering, what do you think about the kind of developmental mechanism? So is that there's a peripheral region that is more flexible, but the central region is more pre-specified?
Right. Yeah. So and this is just speculation about primary areas and secondary areas versus sort of these higher order areas. I think they're more genetically specified. So I think, and the reason you can't get rid of it is the way genes are deployed during development. If then it sets off a bunch of cascades, and you can't get rid of it without bad consequences. I think regions like posterior parietal cortex are less specified. And what's interesting, too, is if you look at really early brains or really small brains, you have visual cortex, somatosensory cortex, and auditory cortex are all bumped up against each other, right? So that's like this core region that I think is much more genetically specified. So you can muck around with them a little bit, but you can't get rid of a lot of aspects. So, that serves as the way genes are deployed during development serves as a huge constraint in how you're going to change your brain. But then with the expansion of some of these regions of cortex, I'm not suggesting that there's no genetic specification, because there are general principles of organization, but you're able to modify them much more radically during development than these primary fields.
We'll have the last
over here, Leah. So my question is, what do you think does it mean to have a larger cortical representation? Particularly when, even in the lifetime of an individual, learned skills, as you and many others have shown, increases representations of one region, but it's a zero-sum game, and I think this refers to an earlier question. It's not that other skills are being lost, which is a counterpoint to the idea that size doesn't necessarily equate to skill or even the acquisition, or certainly the retention of skill. What does it mean to have a larger representation? Also, if you take rodents, and I'm mortified to be quoting Lashley here, if you remove the cortex, what is lost?
I know.
So there's a number of questions that are merged in, but I think we need to think a little bit more, going back to Larry's talk of not only cortex, but his relationship to other areas and the networks and nodes.
No, I agree.
I
focus my talk on the cortex and cortex-thalamic relationships. But I will say, and I'm not 100% certain what having a larger cortical field means. I mean, people have speculated on this. I will say, though, that if you look at expansions and contractions of representations throughout a lifetime, those are different. I mean, they're on a much smaller scale than what you see, for example, in the bilateral nucleate experiments, where you have this really, really huge expansion of the face representation in visual cortex. Or in the natural world, you have this huge representation of the bill. I mean, the bill occupies about 80% of the cortical sheet. I mean, it means more neurons are processing inputs from the bill. That's probably all I can say about that. But I agree. We still don't know fundamental questions like, what does a bigger cortical field buy you? What does changes in connectivity, cortical and subcortical, mean? And we also know that, small changes in connections at low levels of organization can manifest as really huge changes in the cortex. So, yeah, it's a good point.
Okay, let's thank Leah again, and then... We'll have a coffee break now, and we'll be back to the room 11:10 on time. Next speaker is Eric Jarvis from Rockefeller.
Well, thank you for the invitation. And I'm really happy to be here and be among such I think, a high-profile group of researchers and familiar faces. So without further ado, I'll talk about the topic I've been studying for many years and sort of daring to say, we are generating some insights from non-human species into understanding the neurobiology of language in humans. And to really address this question, we also need to address the question of what is language. And as many labs are studying this particular trait, you'll get as many definitions. And the definition that I adhere to most is thinking of it as a multi-component trait, where it's a totality of different things like context-dependent inference, theory of mind, speech perception, production, and vocal learning. And a number of these traits are actually present in many species, maybe amplified in humans, or maybe the similar, like something like auditory perception. But others are actually specialized to humans or to a small group of species. And the one that is most specialized, that gives language its most unique component for spoken language, is what we call vocal learning. It gives us that ability of speech production. And what I'm going to tell you today, and I think it's important to get this right, a model of trying to define what it is and how to use animal models to study it, because 1 in 10 people are affected with speech disorders at some time in their lifetime, which is a large percentage of the population, anything from stuttering to autistic deficits in children with, speech deficits in children with autism. So what I'm going to do is tell you about what we've learned at 3 different levels, the behavioral, the anatomical, and the genetic, and then trying to test some of the hypotheses that comes out of these findings in mice. So first, on vocal learning itself. As I said, it's a pretty rare trait. It's only found in 5 groups of mammals, us humans, of course, dolphins and whales, bats, elephants, and seals. And it's found in 3 groups of birds, parrots, songbirds, and hummingbirds. All of these species have close relatives, like chimpanzees to humans, that don't learn how to imitate vocalizations. And so it's thought that each of these 5 groups of mammals and birds evolved the trait independently of a common ancestor. And this is what I mean when I say vocal production learning. I mean the actual production of novel or imitated sounds. Another component, though, that is necessary for language but is more ubiquitous is auditory learning. And this is common in many species. So dogs can understand the word sit, or siéntese in Spanish, oswari in Japanese, come here, get the ball, and so forth. You can teach a dog to learn how to understand 400 words and different combinations of words. But a dog can't say, okay, you got it, I will sit. All right? Instead, a dog goes woof. And a dog can learn to use woof in different social or other contexts and even attach meaning to the word woof or sound woof in particular objects of food. But that's what we call vocal usage learning, but not production learning. Now, conversely, there are a number of specialized traits that are associated with this vocal production learning behavior, including some disorders that you only find in the vocal learning species. We all depend upon auditory feedback not only to imitate the sounds, but when we become deaf, we lose the ability to produce learned vocalizations, learned speech, unless we undergo some kind of therapy. We all have critical periods for this trait where we, songbirds, parrots, and others, will learn how to imitate sounds really well early in life, before puberty, but afterwards it's difficult. And some things that affect critical periods also affect, like in autism, also affect specific language development in humans. We all culturally transmit our vocal repertoires from one generation to the next. unlike what happens with the species that produce innate sounds. And so when you take a conspecific zebrafinch or a human away from their conspecifics, you lose that speech. You lose that language, that culturally transmitted repertoire of sounds. So now, not all vocal learners are equal. Humans are by far the most complex, as you might be able to tell, but just because we're the most complex doesn't mean there are differences. Everybody else is different, or let's say simple. And a good example is parrots. And here's a case of Disco, a parakeet, a small parrot who's raised with humans for 4 years, and here's what Disco can do.
Never shake a baby bird. That would surely be absurd. Never shake a baby bird. That would surely be absurd. I am not a crook. My name is Disco. Again, meow, meow. You mean yes? Chili kid has cheeseburger. Are you a little stork or a stormtrooper? Disco, I used to be a spy. Not a parakeet. Sure, I'm a doctor, not a parakeet. Ideally, I'm a doctor.
See, and the dog just goes woof. So there's a pretty big difference there, all right? But every once in a while, people get excited with the video's not happening here? I guess it didn't get uploaded. All right. Well, I encourage you, some of you go online, you'll look at it, and you'll see a gorilla named Coco. What? It's going?
I said just go on the fold.
Okay. So you can do that for me?
Play from your fold.
Okay. This is what happens sometimes when you translate. So where it says Coco here? Okay, so every once in a while, a study comes out where they're saying a non-human primate is learning how to produce some imitated sounds. This will be video.
How about when you're
Okay.
coughing? That was good. You did a sneeze and then a cough. Excellent.
Okay. So yeah, that is excellent, but it ain't like Disco saying that's a cat, meow. Or, well, although it was a dog, we'll go ahead and put the and so so what's going on there is that Coco, who's raised with humans for 49 years, has the ability to understand human speech, as you can see, and respond quickly, and even understand some syntax or rules from the perceptual domain, but has difficulty actually producing novel sounds. And the issue is that Coco is able to voluntarily blow air up through the larynx, but not able to modulate that air in a way that we do to produce new sounds. All right, so that's prompted scientists like myself, many sitting in this room, to ask the question, why can't we get our closest relatives to say something as simple as apple, where those who are 300 million years removed from us from a common ancestor like these parrots can go one further and say golden delicious. So I've come up with a theory of why that difference exists, and I call it the motor theory of vocal learning origin, but the theory that I have is not the only one that's around. Some of these have become assumptions. One is the presence versus absence of a forebrain vocal learning pathway. The vocal learners have it, the non-learners don't. The other is that many species actually have this pathway, but actually in vocal learners you get a direct projection from the cortex to the motor neurons that control the vocal organs, and the vocal non-learners you get an indirect projection. And whenever you have a direct projection from the cortex to motor neurons, you have greater fine motor control of your behavior. The other is that there's a difference in the presence or absence of a direct versus indirect connection between auditory regions and vocal motor regions, like Wernicke's area projecting to Broca's area. Or coming from the linguistic community, there's a language module in the brain, or a language module circuit that humans have that interacts with these external auditory or speech circuits, and that other species may have, but they don't have a language module to synthesize it all together to give complex language. Or that there's a general difference in the brain, in the circuit or the number of neurons. Bigger brains just allows you to have speech. Or that it's not all in the brain, it's actually in the vocal organs themselves. Like in the larynx. That's where there's an extended drop in the larynx in humans that would allow greater production of different sounds. So I'm not going to have time to go over all of them, but I'm going to show you some data that I think will support one or two of these. And in my own work, it came about when we found that when canaries sing their learned songs like this,
That act of singing is associated with a robust increase. Can I use the arrow here? Oh, great, yes. Associated with a robust increase of immediate early gene expression in white here in certain areas of the brain that we know as song learning nuclei, like HVC, area X, and L-MAN here. The white signal is an mRNA product for the immediate early gene EGR1, and the red is crescent violet staining of the brain. And the more an animal sings, the higher expression you see within a half-hour period. Pretty robust response here. And so, and this is motor-driven. That is, it occurs in the absence of auditory feedback, in the absence of somatosensory feedback. Even if the bird tries to sing in the dark, you'll get this, or is mute. And he tries to sing, but actually doesn't produce sound. So we use this molecular mapping approach to identify communication circuits in all different kinds of bird species, and we find that in all of them, whether they're vocal learners or not, in blue here, they have this forebrain auditory pathway, which would be consistent with all of them having auditory learning. But only the vocal learners, the parrots, hummingbirds, and songbirds here, had brain regions that were active, forebrain regions, that is, cortical and striatal regions, that were active in the production of vocalizations. There were exactly 7 brain structures in each one of these vocal learning bird groups with similar connectivity when it was mapped out. The red pathway here is responsible for learning how to imitate the sounds, and the yellow is responsible for producing those learned sounds. The gray is for innate vocalizations. So we summarize that either, according to this new phylogenetic tree, we actually thought the tree could be wrong and they all had a common ancestor. Well, we did a whole genome scale tree, and they are showing independently evolved lineages here of hummingbirds, parrots, and songbirds. So either there are 3 independent gains of this trait with similar brain circuitry, or there is a common ancestor back here around the time of the dinosaurs, and there were 10 losses. All right, so we're supporting the idea of most likely of 3 gains. What about humans? You can't propose a common ancestor here, I imagine. It has to be independent. And here we start to ask the question, well, if songbirds or parrots and hummingbirds can come up with a similar pathway over the last 65 million years, what about us humans? Are we going to be any different? And here is a songbird brain, a zebrafinch to scale with a human brain. You can fit 3,000 of them in a human. That is, brain size doesn't matter. What about cortical folding? Well, obviously that doesn't matter either. Here's a smooth surface telencephalon, here's a folded one. And chimps have folded cortex, and they still can't do what these parrots can do. So what is going on here? To actually compare humans and songbirds, we first had to get rid of an old, outdated understanding and terminology of the avian brain compared with mammals or vertebrates in general. This old view, going back over 100 years, is that the non-mammalian brain is mostly basal ganglia. In this case, in birds, it was thought to be all purple here at striatum and globus pallidus. And even up until the time I was a beginning assistant professor at Duke, we were using this terminology. I organized a consortium of scientists in the early 2000s with others, where we reevaluated all the evidence, did some more experiments ourselves, and came up with a different model where all of this territory above the purple region here is actually cortical or paleo territory. And one hypothesis started by Harvey Carton is that the different cell populations here as clusters are homologous to the different layers of the cortex. And another hypothesis is that they're homologous to the clostrium and amygdala, and only this more dorsal region up here, the hyperpallium, is homologous to the 6-layered cortex. Even though this was a great improvement over the last 100 years, we did some more work in my own lab to argue that we even got it wrong there. We argued that in the bird pallium, there were just 4 major cell populations above and below where this ventricle space once was during embryonic development, and that these cell populations here are homologous to the cortex and clostrium and amygdala as fields. And so further, we found that they function as semi-columns in their organization, including in the song learning nuclei, where they are located in these brain regions. And so you'll hear from Zoltan that some debates about this and whether or not this is homologous or not, but it's interesting that it functions similarly and semi-organized similarly as it is in mammals. So with that new vision, I've argued that we humans have independently evolved brain pathways for production of spoken language that involves the laryngeal motor cortex and Broca's area and other regions that is convergent with these regions in songbirds, parrots, and hummingbirds. In particular, the HVC and RA song nucleus argue convergent with different layers of the laryngeal motor cortex, L-MAN convergent with layer 2 and 3 of Broca's area, and area X of the striatum convergent with a part of the anterior striatum in humans involved in speech production. and that the laryngeal motor cortex region and the analogous regions in these birds makes direct projections to the motor neurons that control vocalizations, and you don't find such direct projections in the other species, as I mentioned. But you do find an indirect projection, at least in non-human primates, with a part of the premotor cortex where some have argued where the laryngeal motor cortex exists. So to explain how all that could have happened convergently, we didn't have an answer until this finding here, where not only the brain regions that are controlling the production of learned vocalizations become active with these immediate early genes when the birds sing, when they actually perform well-controlled movements, in this case, hopping in a rotating wheel, we have 7 regions of activation surrounding all 7 saw nuclei, as the saw nuclei themselves, independent of auditory, visual, and somatosensory feedback. That is, it's motor-driven. And the connectivity of the surrounding brain regions is similar to the connectivity of the song learning nuclei themselves, except with some differences, like a collateral that goes to the striatum here in the surrounding motor pathway, but not in the song learning nucleus itself. Actually, this is more similar to what you find in mammals. So we wondered, well, if this is the case for birds, what about humans? It turns out, if you get, in this case, dancers to do learned choreographed sequence movements in a PET imaging instrument done by a study by Steve Brown's lab, the brain regions that show highest activation when doing learned choreographed sequence movements are the brain regions directly adjacent to those that control speech production, like we see in these song-learning birds. So putting all this together, it came up with this motor theory of vocal learning origin, where I argued that all species have this innate brainstem circuit for production of innate sounds, a motor learning pathway for producing learned motor behavior, like learning how to fly, learning how to walk, and so forth. And I argued that this motor learning pathway is replicated multiple times during embryonic development, getting hooked up to different muscle groups. But in vocal learning species, humans and parrots and songbirds, I believe it duplicated one more time, and that duplicated copy, like a gene duplication, got hooked up to the motor neurons for vocal behavior down here and taken over this innate brainstem circuit to get this emergent vocal learning system that is now similar in all species because it has a deep homology with the surrounding motor pathway that you can find in all vertebrates. So how we're going to test and prove that? We started to try to do so at the molecular level when we started sequencing the genes of many songbird and other species. And so this third part of my talk is about those discoveries. And what we did here is laser dissected out the song learning nuclei from each of the vocal learning species and the surrounding motor areas, and the equivalent regions in non-learning species where we'd expect to find song learning nuclei, even though we don't think they existed there, profiled them to microarrays. Allen Brain Institute was doing something similar with thousands of human brain samples from 6 people and samples from 6 monkeys, including different cortical layers in monkeys, and profiled them to microarrays. And folks in my lab did a lot of the computational work and wet lab work. They have their own labs now. Asked the question, can we find specialized gene regulation in these song learning nuclei that matches any place in the human brain that you don't see in a non-human primate? And the answer is yes. We found that the HVC saw nucleus had specialized gene expression profiles of similar to layer 2 neurons in laryngeal motor cortex, RA similar to layer 5 neurons and matching laryngeal motor cortex. The L-man song nucleus involved in song learning, with more recent data from Greg Jedman in my lab using RNA-seq, found a profile that matches Broca's area. And area X, a part of the anterior striatum, not quite where I predicted, but still in the anterior striatum. And we did not find any correlation of up or down regulation of these genes that match what we see in these songbirds and humans, in chickens or quails, really, and macaques. So what's interesting here is that this is like 50 to 70 genes per speech brain region that shows convergent gene regulation with songbirds, parrots, and hummingbirds that you can't find in these other species. So it's not just 1 or 2 genes. I'm going to give you an example of one of those genes because many of them we found were involved in neural connectivity functions. In this case, SLIT1, an axon guidance ligand, was downregulated in the song production learning nucleus of each of the vocal learning bird groups, but not in doves or quails, vocal non-learners. And here with the heat map on the human brain, you can see the white downregulated in 2 regions, this ventral laryngeal motor cortex and this more recently discovered dorsal laryngeal motor cortex. And these 2 sites here in the human brain happen to be the locations of hottest neural firing from Eddie Cheng's studies when people undergoing surgery are producing speech, and they put electrodes there. So the coordinates correlate precisely to where we see specialized gene regulation. So one asks, well, what is causing these specializations, and what are they causing themselves? Why downregulate these axon guidance molecules? Well, it turns out SLIT1 and a number of other genes that we found downregulated are genes involved in repelling connections from forming. And so I call this repulsion hypothesis, where in a vocal non-learner, you have connections coming from the cortex projecting down to the brainstem, and where it has low expression of the ROBO1 receptor, it can bind, but where it has high expression in the motor neurons for speech, we think it's preventing those connections from forming. Whereas in songbirds, parrots, and hummingbirds and humans, it's downregulated, and we think that then allows for direct projection from the cortex to form onto those motor neurons. And so that's what we'd like to test in songbirds, like upregulating SLIT1, see if we can make a direct projection indirect and prevent vocal learning, downregulated in a non-learner, take over an existing motor learning pathway, and see if we can make a indirect projection direct, maybe even get a chicken to imitate, and do the same thing in mice. And there is actually, after we proposed this hypothesis, a study came out that Nenad, who's here, collaborated on, showing that a receptor of SLIT1, an alternate receptor, plexin A1, also shows differential expression in the mouse and human brain. It's downregulated in human motor cortex. And when they upregulated or downregulated in the mouse cortex, they got greater projection to the motor neurons, as well as greater a number of neurons in the motor cortex that project to this region and greater dexterity of the paw movements in the mouse. Can we do something similar to the vocal circuits in mice, is the question we were asking. But we also have to characterize the vocal behavior. And what shocked many of us in the field back in 2005 from Holly and Go's study is that mice have these ultrasonic vocalizations that when pitched down to our human hearing range sound like this. You would think that's a bird, right? Here's a sonogram of what we just heard, the sound, the time and the frequency here of the sound. And male mice, like birds, produce these courtship songs, now we call them, while they're trying to court a female, and she likes it, she'll mate with him. If she doesn't, she'll run away. And different siblings will produce different syllable repertoire compositions, indicating there's some level of plasticity here. We don't know if that's genetic or learned, but is it learned? There are 6 studies that came out since then that tried to test for learning, and several of them, including from my lab, said there's a small degree of vocal plasticity, but not much, not like you see in songbirds or parrots, and others say there's absolutely none. So those two groups are debating with each other, but both can agree we're not getting something like Disco the Parrot from these mice. So even though it sounds like a song, and that's some work done by folks in my lab. But what was interesting and surprising is that when we got these mice to sing, we found a region of the cortex, in the motor cortex, that had high vocalizing-driven gene expression, like what we saw in songbirds. Such a region of the cortex was thought not to exist in non-vocal learning species outside of humans or these songbirds. So when we start to map out the neural connectivity of the circuit, injecting transsynaptic tracers in the laryngeal muscles, we found that that same exact region had layer 5 neurons, here, nicely labeled layer 5 neurons, that when placed tracers there made a direct projection actually to the motor neurons for vocalizations, but it was a very sparse direct projection. Here in black, you see axons coming from these layer 5 neurons. There are only 1 to 3 axons per motor neuron, whereas in songbirds and a little bit of tracing data from postmortem human brains, they look like they're hundreds, if not thousands. So this could be a difference of degree as opposed to absolute difference. Well, if we lesion that area, we also find that the mice can still produce their don't say that. They still produce their songs, but they lose some modulation of their vocalizations. So what about the genes themselves? Well, the ones that we found like downregulated in humans and songbirds in the vocal production areas, we did not find it downregulated in the mouse motor cortex in this region here. So what we think is that maybe instead of trying to induce a brain pathway in mice, maybe it's already there at a very rudimentary degree, and we can just enhance it by downregulating these axon guidance molecules, like what was done with the plexin A1 gene for the forepaw. And so we started doing some experiments, and we've been able to upregulate using human SLIT1 in a songbird RA song nucleus here. and asked what happens to the bird's song. And guess what? The higher the overexpression of SLIT1, the more the bird's song starts to deteriorate from one song bout to another. And here is an outlier where the injection was outside of the RA cell nucleus and the bird song stays intact. What about mouse? Using an RNAi molecule, we were able to downregulate SLIT1 in that area of the motor cortex, hooked up to a GFP molecule. And in this case, what happened is the mice just sang less, all right, which was interesting, that any vocal behavior difference occurred at all. We didn't see changes to the acoustic structure of their sounds. This is the folks who worked on this project. But with new syntax analysis tools that we developed on songbirds and then used them on mice data, we found that the sequences, the darker colors here, mean difference in the syntax before and after SLIT1 downregulation changes, such that this multiple pitch jump M syllable type sequence to another multiple pitch jump syllable is actually occurs less often after knocking down, occurs more often after knocking down SLIT1. So it's a complicated graph. I just want to the take-home message is that the syntax changes, or the sequence, not syntax that it has meaning in humans, but we don't know if it has meaning here. What's interesting is that SLIT1 is regulated by another famous gene, FOXP2, a transcription factor that when mutated in humans causes a speech deficit, such as this one here. Let's do that. I didn't get transferred over. Sorry about that. But what happens here is that children with heterozygous mutation of this transcription factor have difficulty producing learned speech, but they can understand speech quite well, not as good as the normal population, but much better than their ability to produce it. And what is really happening is that there's a problem with the sequencing of phonemes to make complete words. A lot of people put the human mutation in mice and studied the vocal behavior and said hardly anything happens except for just some less vocalizations like we found with SLIT1. But then we used tools that we developed for songbirds and applied it to the mouse data like the syntax analysis, and we found the following. In wild-type mice, going from smelling female urine, they love female urine by the way, to a live female, they switch from more simple sequences and syllable types to these more complex ones here. Whereas the FOXP2 mutant mice that have the KE family mutation have difficulty switching to these more complex sequences of vocalization like humans do. It's not as a dramatic as effect as you see in humans, but it does exist in these mice. What about those layer 5 neurons that I said that make a projection down to the vocal motor neurons? They exist in these mice with the FOXP2 mutation, here labeled with GFP, but unlike wild-type mice where they're localized here in the primary motor cortex, in the FOXP2 mutant mice, they're more spread out of the cortex. So this FOXP2 gene is necessary to localize these neurons that are projecting to the laryngeal motor neurons in the brain. So summarizing, I have 5 slides to summarize here. What we're coming to is that maybe vocal learning, this trait, this component of spoken language is not binomial, but a continuum, where you go from simple to more complex ones. Mice are more limited. Let's say songbirds are more complex, and parrots and humans are the high vocal learners. And maybe it's not just that trait, but other traits. And how does this figure into these various hypotheses I talked about in the beginning? Well, instead of presence or absence of a brain circuit or presence or absence of a direct or versus indirect projection, maybe there's some species that are completely absent, but others that have it to various degrees, and it's enhanced presence or enhanced direct projection in the more advanced vocal learners. What about this projection from Wernicke to Broca's auditory to motor regions? Well, actually, we find a pretty robust projection in mice in the analogous areas. So I'm going to rule that one out for the moment. Language module in humans or any other species that are vocal learners, we don't find evidence that there's a separate one that's controlling the speech production pathway. Rather, the rules for syntax and so forth we think is inside of the speech production pathway, not separate from it. There are general circuit and neuron number differences actually in the vocal learning species, like SR-GAP2 mutation that causes a general brain circuit difference in humans. Vocal learning birds have many more neurons packed per brain region space than non-vocal learning bird species. That was a discovery in the last few years. And there's no data showing that can support that there's a vocal organ difference. In fact, some vocal non-learning bird species have more vocal organ muscle complexity, more muscles and so forth, than the vocal learners, we think, to compensate for not having vocal learning. All right, and so explain all of this, I'm going to, this continuum hypothesis. I think there's an innate brainstem pathway in all these species, and there's a forebrain vocal motor pathway that gets duplicated to become a vocal motor learning pathway. In the advanced vocal learners, it moves outside of the regular vocal, the motor learning pathway, and then it gets enhanced with downregulation of SLIT1, genes involved in neuroprotection and plasticity, and then it gets duplicated one more time or even more than twice in humans and parrots. And that this could happen to multiple traits, not just for vocal learning, where there could be a longer continuum, and all of them in human may be more advanced to give us the more highly advanced spoken language we have today. And this will be useful to understand or translate understanding mechanisms of vocal learning, let's say even in birds, to humans, whether they're homologous or convergent. We can now predict, according to our analysis, the different cell types that these cells in the songbird brain would function like in the human brain for speech production and perception and so on, including cortical areas, basal ganglia regions, as well as brainstem regions, and in the thalamus. And one day may be able to figure out, yes, why is it difficult for this animal to say something like apple, or even get it to say apple and say, what are you thinking about, by manipulating these genes in the brain. So I will go ahead and stop there, and you can read the summary here and take questions. Oh, and also thank my funders, including NIH here, by the way. Oh, okay, go ahead. Yes. David? Oh, I thought you raised your hand. No, okay.
Eric, great talk. I'm wondering for that SLIT1 manipulation, was that experiment done in the young? What stage was it done in the adult?
Good question. So we initially did it in an adult. We didn't see any change in the vocal behavior, and then we repeated the downregulation in pups, and then we saw a change in vocal behavior when they became adults. Yeah, which is perplexing to me because all the human data and the songbird data that we had was from adult humans. All right, so the gene is developmentally downregulated during critical periods, but then it stays that way for the rest of our lives. Yes.
Hi. I remember a study about birds and FOXP2 being related to differential partitioning of the striatum. In some way, do you see that as well? The idea was that a greater proportion of the striatum was dedicated to plasticity and quick learning versus habit strength?
Say it again? FOXP? Oh, yes. In birds, you mean? Yeah. So I didn't have time to present that, but yeah. So work from Constance Scharf's lab, Rich Mooney's lab, our lab, has shown that if you manipulate FOXP2 in the Ariex striatal song nucleus of songbirds, you actually impair the ability for the birds to imitate usually the acoustic structure of the sounds properly as opposed to the sequence. And what's interesting and paradoxical about that is that in humans, the sequential production of phonemes is affected more than the actual acoustic structure of the sounds. And so why manipulate it in the striatum region, you have an effect mostly on the acoustic structure, is interesting. And it indicates that maybe the effect of FOXP2 mutation in humans is different when it's prevalent throughout the body or throughout the brain than just localized in the striatum. Jonathan? Somebody?
Yeah,
okay, yes.
I was wondering whether your theory could also be applied to more the temporal aspects of language, like grammar.
Mm-hmm. Yeah, so one thing that's been hard to do is understand what these birds, non-human animals, are saying and meaning when they produce their learned sounds. Most of the time, they're producing their learned sounds in more effective states to produce a high variation of sounds to attract a mate or defend a territory. There are a few species out there, including some parrots, like you just saw here, that will actually produce more semantic information in their learned sounds, and you can start to try to ask the questions about grammar and so forth. But I think we're really far away to even, some people think that's a sacrosanct area, that these birds aren't even having anything close to grammar. But it's just really hard to understand what another animal is saying. And think about it. If you grow up with just speaking English, and you only heard English in your entire life, and you hear some other language, it just sounds total gibberish to you. And that's what these songs sound like to us.
I was just curious, you said, if I understood correctly, that you saw 55 to 65 genes change, but you only tested one of them. I was wondering, when you look at networks of those genes, do you get clues of what other mechanisms may also be involved? You proposed one, but what are those other genes doing?
Yeah, so we found out, we tested 7,000 genes for the convergent expression analysis and found 50 to 70 per brain region. But actually genetically manipulating them, that's hard to manipulate multiple ones at the same time. We've been trying to see if there's an upstream transcription factor that specializes them and maybe hit multiple genes at the same time. Other than that, we're thinking about ways of using CRISPR to affect multiple genes at the same time, and then we might get a more specialized trait as opposed to manipulating one. But yeah, that's an issue.
Eric, one thing I'm trying to get a handle on is a question that was raised earlier about evolution of language and syntax as opposed to vocal production. And so, some recent experiments I think you're familiar with from Bob Bulding's lab suggest that there's a difference between two vocal learners, say zebra finches and budgies, such that the zebra finches don't care about the order of a syllable. They care about the fine structure, but not the order. So there you have an automatic difference between sequences and encoding of sequences and production of sequences. And I wonder if you've thought about, just pushing you a little bit on this, how either vocal memory or auditory memory or how sequence encoding might play a role in development of more complex communication skills.
Yeah, so sequence encoding, whether it be sequence encoding in the auditory domain or in the motor circuit itself, is something that we don't know a lot about, but we do know that as you go up higher levels of the auditory pathway, those neurons are more and more sensitive to the sequence of the sounds that they fire to. And in the vocal motor pathway itself, it's more of the anterior pathway regions. That would be analogous to Broca's and anterior striatum in human, that is more involved in the sequential production of sounds. And they have different gene expression specializations from one species to another, which we think might be responsible for some of them. And so that's an area that needs to be investigated more. I do want to say one thing before you get me off here, that I talked a lot about the basic biology of these circuits here, and I know this session is also focusing on disorders. But I want to say that what I'm learning more and more as I saw with our bird brain studies, you really got to understand that basic biology of that species really well, of one species versus another species, before you can compare the two. Because if you have errors in one versus errors in the other, then you actually propagate those errors. But with that said, in this anterior pathway and other parts of the circuit, you can lesion some of these brain areas and get stuttering like you do in humans. You can get sequential deficits like you see in some these vocal learning deficits in these FOXP2 mutant humans and so forth. So some of those deficits are similar, but the mechanisms of how they work, we don't know if they're similar or not.
So, Eric, nice talk. So humans, of course, can also produce language using sign language. And I wondered if you've given any thought to the pathways in monkeys and in humans compared to the birds on that side.
And yes, I put a lot of thought into this, and because people keep asking me, like yourself. And I'm going to do a little bit of hand waving here, but I think it might be true. Is that I think the computational mechanisms that give rise to our language communication, whether they be productive, like with signing or voice, or receptive, is already there in the neural circuitry. It's already there in the motor learning pathways. And what had happened in human evolution, I believe, is that when we evolved this vocal motor speech pathway, it inherited that ability to produce complex syntax and learn sequential movements. And this is why, actually, I think even as I'm signing here, when you do speech production, we tend to learn gestures, because I think there's an evolutionary relationship there. So I don't think the complex rules that involve syntax and so forth and grammar came about with learned vocalizations. I think it existed beforehand and then became enhanced after vocal learning occurred.
Okay, let's thank to the Eric.
Thank you.
So our next speaker is Susanna from Vanderbilt.
Good morning, and I want to start by thanking the organizers for the invitation to first listen to really amazing talks and then see if I can give one myself. I'm gonna tell you about work that we've been doing on the larger issue of where do humans fit in. We're going to listen a whole lot more about what is so particular about the human cortex and the human brain as a whole and how did it develop. And of course, the mouse is the other extreme that's really interesting because it's so approachable and amenable to manipulations. But the problem is that when we do only that, whilst we learn a whole lot and a lot of details about these two creatures, we really fail to grasp what is the general picture, where do mice and humans fit in. So what I'm going to tell you is I'm going to give you first a very quick summary of work that we've been carrying out over the last 15 years, ever since we figured out a way to very rapidly and reproducibly count cells in brains or really any fixed tissue, anything that you can dissect, you can count the number of cells that compose it very, very rapidly, and that I won't give you more details other than we affectionately call this method brain soup, because that's really what it is. You dissolve the tissue that you dissected, and it's because it's very, I don't have a pointer here. Because it's very easy to use, we've been, and thanks to an awesome network of collaborators all over the world, we've been able to analyze a very large number of mammalian species and also bird species now. For the rest of the talk, I'm going to be showing you all graphs color-coded in the same way. And let me point this out. That if you remember one color, remember red. That's the color I'm going to show all the primate data, including humans, in. Then rodents in green, and each mammalian clade in its own particular color. Now, before I show you what we have for where humans fit in data on all these different species, let me show you what we have for the human brain counted the same way. So first, it's not 100 billion neurons and 10 times more glial cells, please. It's on average 86 billions and not a single more glia cell than that. That's an average number for 4 male Brazilian men of 70 years of age or so. But and if you would like to protest that, 100 billion, 86 billion, what's the difference really? The difference is a full baboon brain. That's what 14 billion neurons amount to. But what I really want to point out to you is where the human data points fit, where they fall compared to all the other species. So here you see already one major trend that I'm going to be alluding to. You see all the red data points, they fall on a different slope. They form a different arrangement from everybody else. And you typically always see that. And that illustrates already in one single picture that a primate brain is unlike other mammalian brains in many, many ways. There is one thing that makes human brains and the human cortex in particular stand out very much from all the other species, and that is the sheer, so absolute number of neurons that you find in the human cerebral cortex. And let me just point out that this is the only graph that I'm going to show you on a linear scale for the rest of my talk, and I do that simply because this is how we're used to thinking about numbers and quantities, and this allows you to appreciate the distance between the second-placed species in numbers of neurons in the cortex, which are gorilla and orangutan. And then you get chimpanzee with about 7 billion. This is 16 billion neurons. Here you get gorillas and orangutans with about 8 or 9, chimpanzee with 7, elephant 5.6, cetaceans, we have a minke whale with 3 billion neurons, and we can predict that all other cetaceans are going to fit in the non-primate curve. So this is what we get. Apart from that, there seems to be nothing otherwise particular to the quantitative neuroanatomy of the human brain. And let me really make this clear. I am not at all saying that nothing else is different about the human brain, the human cortex, or human neurons. I'm speaking strictly in terms of quantitative neuroanatomy, all those things that people used to think that, of course, had to be completely different in particular about the human brain. They're really not once you have numbers of neurons to bring into the picture. So let me just walk you quickly through these. You see that the human cortex has about the mass that you would expect for its number of neurons, or the other way around. It has, like all other primates, it has a much higher density of neurons in the cortex compared to what you get in other mammals, and that's not because primates became different. It's not that primates gained more neurons or smaller neurons. It's what we've proposed is just that over the course of evolution, as the first mammals were probably here because the rules still apply, as more neurons, as animals appeared with more neurons, the average size of the neuron probably increased, except when primates branched out, one thing became, so more neurons became dissociated from whatever it is that makes neurons bigger. You also see that we fit the relationship for cortical surface area for the number of neurons in a primate brain. We have a similar constant number of neurons per square millimeter of cortical surface for a primate, which, by the way, is a whole lot more than you find in other species. We have about the volume of gray matter in the cortex that you would expect for the number of neurons. Same for the white matter. If you look at the prefrontal region in particular, so all cortex anterior to the callosum, you find that we fall exactly on the spot for the relationship between the number of prefrontal neurons and the number of non-prefrontal neurons, or the volume of the prefrontal white matter as a function of the number of neurons in the prefrontal region in other primates. These curves, by the way, were plotted without the human data point, and you see that the human data point really falls right on the line where you would expect. We have a cortex that's about as folded as you could expect for a primate cerebral cortex with our number of neurons. That's different from non-primates, but actually we are like any other mammalian species in the degree to which our cortex is folded, given its combination of surface area and thickness, which is what we have found to describe 99.8% of the variation in cortical folding across species, according to a very simple physical model of minimization of free energy. That's a whole other story. What about the body? Even there, it's not humans that are the outliers. What we've proposed is that, probably for metabolic reasons, it's the great apes, it's the largest great apes that are the outliers in the relationship between brain size or number of neurons and body mass. You see that we have, first, let me point out that there's a lot of diversity that's acceptable, let's say, in terms of the relationship between the body mass and the number of neurons in the brainstem and diencephalon that operate those neurons. But you see that humans are really no different than you could expect in that range of diversity. And you see that we also have the number of neurons in the cortex that you could expect for a generic primate with our body mass compared to, again, non-great ape primates. And you see, let me just point this out already because I'll come back to it very soon. You see here that there's a clear pattern where for a given body mass, let's say a 1 kilogram warm-blooded animal, primates have more neurons in the cortex than any other 1 kilogram mammal, but birds will have even more neurons than a primate. A 1 kilogram bird will have even more neurons than that primate, and I'll come back to that very soon. Let me just finish this part here by pointing out that there's also no clear differences in the quantity of glial cells that you find in human brain structures for the size or for the number of neurons that you have there. The glia to neuron ratio in human cortex, cerebellum, brainstem is also exactly what you would expect for the density of neurons. So for the average size of the neurons in the tissue. So also nothing completely different there. And finally, you're probably aware that one of the last ways in which the human brain might be completely different is that, well, our brain is extraordinarily expensive. It's just 2% of the mass of the body, but it uses 20% of all the energy that we have. Well, yes, except that that is exactly what you would expect for a mammalian brain that has the number of neurons that we have. So it turns out that there's a almost perfectly linear relationship. Yes, there are only 6 data points available so far, but still, you get this nearly perfect linear relationship between how much glucose the brain consumes per day and how many neurons that brain has. The slope gives you a rate of about 6 kilocalories per billion neurons per day, which doesn't sound all that expensive, but if you do the math in comparison to how many calories different animals can consume, also based on their size and how many hours they are awake per day, it amounts to a whole lot. It amounts to 500 calories of your average 2,000 just to keep your brain alive. And you see how that is, again, exactly what you would expect for a primate of our body size that consumed as much energy as we do. So you put all that together and you get that, again, in quantitative neuroanatomical terms, you get that the human cerebral cortex is really just a very large primate cortex, meaning it has all the characteristics that we can measure in this scope that you could expect for its number of neurons. What is really unique about it is the absolute number of neurons that you get in the cortex. So it's the extent to which whatever it is that regulates how numbers of neurons get added to brains, it's the extent to which that has come. So what I want to propose is that yes, of course, we all want to understand what makes a cortex human, but I'd like to propose that, yes, pairwise comparisons are useful and they're very interesting and they're important, but there's a problem with that that I'll get to in a moment. And maybe a more useful or efficient approach would be also to look at not that one question, what makes a cortex human, but actually a combination of 2 questions. First is what distinguishes a primate cortex from others, non-primate cortices. And second, how do cortices as a whole gain neurons? Because what we've learned from looking at all these different brains is that there is, you can predict all this quantitative and neuroanatomical variation from just fiddling with 3 parameters, 2 that regulate how many cells, how many neurons you get, and 1 that regulates what is the average size of the cells. So you can have a set of primate-specific changes that apply to how primate and not other mammalian cortices are built, and there probably is a whole set of other set of genes that regulate numbers of neurons. Now, the important thing is that we know already from the literature that there's not a single gene that does this. There's many of them, and there's probably, once you start running the pairwise comparisons, you're going to find as many differences across one species and other species as there are pairs of species to be compared. And where I would expect us to find what really distinguishes the human species is whatever lies at that intersection, what the differences that make us a primate and the differences that make us a primate with a particularly large cortex. All right, so what? So we can think of there's all this diversity, we're just one more data point. What does that mean? Well, we're used to thinking in terms of adaptation. Everything that exists out there must exist for a reason, must be good for something. So there certainly must be pros, advantages to having tons of neurons in the cortex. And sure, this is the most boring one. I'll say that with more neurons, you get more cognitive capabilities. You can imagine the various ways in which that would bring about increased fitness. I'm not going to go into that. In terms of evolution, there's one really interesting pro that is, and this is a whole other story. I'm just going to give you the 10-second version of it. It turns out that the best predictor for how many hours a species needs to sleep, a species of a mammalian species needs to sleep, is the ratio between the density of neurons in the cortex and the surface area above that cortex, above that density of neurons. Presumably, this is what I propose, presumably because that is what determines, that's what limits both the rate of production of sleep-inducing metabolites and the rate of clearance during waking and sleep. Important point is, it turns out that varies highly predictably with the number of neurons in the cortex, depending on whether you're a primate or a non-primate. And in general, the more neurons you get in a cortex, the more waking hours, the more hours you get to spend awake, which is very important, especially considering that the first mammals we know were very small, with very tiny brains, with very few neurons, and they must have slept most of the day. When you sleep most of the day, there's very little time that you can spend eating, so you can only be so small. So the more neurons you get, if that buys you more hours of waking, then the larger you can become, simply because you're awake for longer periods of time, you can eat more. And one of the benefits that come with being able to afford a bigger body is that you go from being food to being the one that eats other creatures. Well, that's fine. So then if nature and evolution work in terms of adaptation and things getting ever better, you could ask, if that's so cool, then how come everybody hasn't developed a much bigger cortex? How come only us? There must be cons. So of course, one, like I mentioned, is a high energetic cost. But then again, you could say, well, that's taken care of because the very same larger number of neurons that cost more, they also allow you to stay awake and feed them. You could say there's a larger body that's required to come along with that because the larger brain has to fit in a larger body, however that happens. But then again, that is also afforded maybe by the very increase in number of neurons. So this is what started me thinking that maybe there's something else. Maybe there's a time component to this thing here. Maybe the larger you get, well, the more time it's going to take you to develop, to reach that body size, although my Great Dane has just shown me that that's not true at all. He's my size and he's only 7 months old. So it could be something like it's the number of neurons that you have in the cortex. Maybe through some mechanism that I'll be the first one to admit, I have no plausible, reasonable explanation for how exactly that could happen, but it sounds intuitive that the more neurons you have in a cortex, the longer it would take to assemble that cortex. So I'm going to move on to a completely different story now, which is, does the number of neurons in the brain, or maybe it is body mass, is that related to life history? Is that related to how long it takes for an animal to develop? So I first thought of that question in terms of how long does it take an animal to become independent, which also felt like something that should intuitively come with having more neurons. The more neurons you have, the longer it'll probably take you to fine-tune them, assemble them, and that's that way that gets you ready to go without your parents. How do you measure that? How do you figure out when a species has become independent? Closer I could come up with was the time to reach sexual maturity. And then something really interesting happened, which is that the time to reach sexual maturity is the one really, really best predictor that was known for how long it takes an animal to eventually no longer live, let's put it this way. And that is also, by the way, the origin of another theory for something that makes humans special. We humans, perhaps, thanks to our grandmothers who started somehow living longer, that would have dragged our childhood, and human childhood would have become longer as well. So that's our data point. Now, let me just show you the standard explanation for what limits longevity is the idea that it's a very slow computer. Let me start over. So it's the idea that aging occurs as the body accumulates damages over time. And the rate of damage accumulation is expected to be proportional to, I tried to jump to one slide and then it crashed on me. But it's okay, I'll keep telling my story. It's opening already. So you could expect that the slower an animal uses energy per gram of tissue, body tissue, the more slowly it should accumulate damages, and therefore, the more slowly its metabolism runs, the longer it should live, because the longer it's going to take it to accumulate all those damages that eventually make you no longer viable. Now, it so happens that there's a very good correlation that will appear soon between how large your body is and the rate at which your body uses energy. The larger your body is, the slower that rate is, and therefore, the prediction that larger animals should live longer. As simple as that, simply because they accumulate damages at a slower pace. It turns out that when you look at that, it's a reasonable correlation. It's an obvious correlation, but it's a correlation with an R-square of just, I think it's around 0.3. So about 30% of the variation in longevity is accounted for by the variation in the size of the body. Now, what happens when you, what happens when you color, no, let me jump to the, thank you. This. I know I'm being mean to the computer. My presentations are like 200 megabytes. Yeah. So when it appears, you see that when you color code the data, you realize that for the same body mass, it turns out that systematically, yes, thank you, there. So that's the distribution of longevity as a function of body mass, and then you color code the data, and that, by the way, is the NH database that I first found out about last year. You realize that for a same body mass, let's pick again that 1 kilogram warm-blooded animal, you find that birds systematically live longer than primates, which in turn systematically live longer than non-mammalian, than non-primate mammals. Does that sound familiar? When I saw this the first time, I could not unglue myself from the computer for the next 10 hours until I had an answer to what the questions that came after, because what this suggests is that it's not the size of the body. This is the pattern that you get for the number of neurons in the cortex. So this is just to show you that this is the relationship between number of neurons in the cortex and body mass that I showed you before. You see that for that body mass, you do have some differences in brain mass across these species, but it really doesn't look that remarkable. But it's when you look now at the same data point on maximal longevity as a function of the number of cortical neurons, and the wheel will stop turning at some point. What you see is that there is a very, very good relationship between, oh, come on. there's a very good relationship between longevity and the number of cortical neurons across species, and that the disagreement, the discrepancies across birds and primates and all other mammals just goes away. And you can actually explain 74% of the variation in longevity by simply the number of neurons in the cortex as a whole. So there you have it. That's that 74% alignment, let's say. And you don't get that if you look at body mass. It's just to confirm that we get exactly the same thing with our own data. You don't get that also if you just look at number of neurons in the cerebellum or in the brainstem plus the telencephalon. It has to be the cerebral cortex, which was unexpected in a few ways. Now, you could say, well, that's 74 species, that's already pretty good, but that's nothing compared to the hundreds or thousands of species that you have in the NIH database. So, and we certainly don't have data for 2,000 species, but we have understood already what is the relationship between the size of the brain and the number of neurons in the cortex of different species, of the different clades. So we can use these relationships to predict from data in the literature on brain mass. We can predict how many neurons those species must have in the cortex. And if that's not related to longevity, you should have an explosion of data points because you're really just introducing noise, unless it actually has something to do, and then you see the data become compact into one distribution where, again, you get that 74% of variation explained. The striking thing, too, is that once you account for body mass, the residual longevity is still there to be explained by the number of neurons in the cortex, but the converse is not true. Once you account for the number of neurons, there's the residual longevity, so longevity, variation in longevity that's not explained by the number of neurons, there's nothing really to be accounted for by body mass or by metabolic rate, which pretty much means that it doesn't matter what the metabolic rate is. What matters is how many neurons you have in the cortex. And that applies not only to longevity as a whole, it applies to how long it takes a species to reach sexual maturity, male or female, and it also applies to how long a species lives after it's reached sexual maturity, and lo and behold, that's us, exactly on the mark. So it takes us exactly as long to develop as you would expect for a warm-blooded species, a warm-blooded vertebrate with our number of neurons, and after that we live exactly as long, that's maximal longevity, by the way, as you could expect. So what does that mean? The more neurons that we have, the longer time it takes us to mature, to reproduce, and the longer we get to live. Is that a pro or a con? Well, you could argue, well, that's definitely a pro, because with increased time, we get more and more learning opportunities. So you can really increase enormously the amount of information that you can assimilate during your lifetime, and you can do that exactly because you had more neurons to begin with, so you have what it takes to process all that information. So great, all is good, except that you might remember then that we still have that same problem. Why do small cortices still exist if all is good? Well, turns out that it kind of sucks to have a lot of neurons, because that means that if your whole lifetime slowed down, if your whole life slows down, it means that everything is going to take a long time, which means that everybody else who's out there with fewer neurons will reproduce faster, will leave their progeny faster, will consume food faster than you. That's not to say that I believe this story. This is another small parenthesis. I think natural selection is overrated. I think what we have is really more and more a picture of diversity in terms of whatever works. There's multiple ways of putting a brain together. There's multiple ways of getting that in the body. And whatever is good enough to function until you reproduce, that will be fine. But coming back to the cons. because this is one way to look at it. This is what I started thinking, and this is how I think the two things tied together. It looks like metabolic rate in the end is really irrelevant. What matters is how many neurons you have. How could that be a key thing to how long you live? Well, if damages accumulate over time regardless of your rate of energy use, then in 1 year you have one damaged neuron in your cortex, in my very childish cartoon here, no matter how many neurons you have as a whole. In 2 years you get more neurons damaged. I don't mean dead, I mean just functionally damaged enough that function and integration of the body through your cortex is compromised. Now, you see how having more neurons would make you more resilient to those damages that accumulate with time, not with metabolic rate, not with age. And that's a key, because in 1 year of damages, that first brain is pretty much gone. In 10 years of damages, now more damage has accumulated to both a medium size and a large brain with a large number of neurons in the cortex. And maybe that middle one is approaching its limit, but the one on the right is still fine. The one on the right still has enough viable, functional, let's say, healthy neurons to keep functioning, and it will carry on until maybe 100 years of age, accumulating a hell of a lot of damages, but it's still hanging in there. It still has enough functional neurons that can keep the body running. So this is what I'd like to propose that we consider just as alternate hypothesis, that maybe it's not that humans are uniquely prone to some diseases like Alzheimer's or Parkinson's disease and so many others. Maybe it is simply that humans are fortunate to have enough neurons that we get to live enough to develop these diseases. And we can think of evolution also in those terms, and in terms of how much time we've added to our life over the course of evolution as our ancestors gained more neurons. That's a very significant increase. And I'll wrap up here. I just want to point out to you that an old mouse at 2 years of age may be equivalent to an 80-year-old human, yes, but they're certainly not equivalent in the amount of damage that they will have accrued over time. And also, you can look at comparable young adult species. They're never going to be equivalent in experience, in information accumulated, simply because of their biology. So cognitive comparisons across species will always be mismatched for that reason. So I'll wrap up here. What I'd like to point out to you is simply that we should add to this whole picture of how do humans stand out from other species, not just the biology, but the cognitive capabilities that we get from having more neurons, but also the difference that it makes, that with that comes a slower timeline, a longer life, and many more opportunities to learn, and then get together, overlap with other generations, and pass it all on like we're doing here. That's why I've come to really treasure meetings and the opportunity to exchange information. So I'll finish here and thank the people who made the work possible. Thank you.
Sorry for hogging first questions, but it's just so interesting, this conference. It makes me wonder if it's cortical increased number of neurons mostly in the cortex as opposed to other brain regions. You know, the cortex doesn't act alone, and as Larry's work has shown, the entire cortex projects to the entire basal ganglia practically. And so has it really been shown that the basal ganglia doesn't have as big a difference like this? And if it doesn't, how do you imagine more neurons in the cortex are surviving damage, but the basal ganglia might not over time?
I can say exactly nothing about the basal ganglion separate from what we are. highly precise anatomical denomination, the rest of brain. The striking thing so far is that was my initial expectation, that if you're going to find a correlation between life history and number of neurons, it should be the number of neurons in the brainstem and the hypothalamus. No, it's very clearly no. Even the diencephalon, which in the birds includes the hypothalamus, and there we have them separately.
So the thalamic regions have been sorted out, but the basal ganglia versus cortex hasn't been looked at yet in these
Correct.
analyses. Okay.
Yeah, the next question, of course, is it one particular part of the cortex, or is it just that the cortex acts as a wonderful proxy for something else that really matters?
Hi, thank you for a wonderful talk. I was just curious if the idea then is that perhaps the issue with the human aging-related diseases is just simply that we're fortunate to be older. Is there strong evidence that species that live longer accumulate these diseases as well, excepting humans? Like I know certain species of parrot have been reported to live to like ages of 70 in captivity. Do parrots develop Alzheimer's disease?
Wouldn't it be wonderful to know? I don't have an answer for you, but that's exactly the idea that I think it would be really important to make a concerted effort to look for these age-related diseases, which I would really prefer to call time-related diseases, in the long-lived species like elephants, whales, and the long-lived parrots and other birds. Yes, definitely.
Hi, Susanna. So you have done these wonderful cross-species comparisons, but I wonder what you might make of the within-species variation in size. For instance, dogs have the largest range of body size and brain size of any extant animal because they have been bred for over a 10-fold range of body size. And it seems, in fact, it is clear that the selection by dog breeders has been for a single gene, IGF-1.
Exactly.
So do dogs show the range of longevity that your cross-species comparison proposes? And second,
Legend
is
has it that it's the
IGF-1
other way
such a
around.
critical gene for brain size?
Legend has it that it's the other way around in dogs. I don't know how much of that is really true, but it makes sense that exactly because of the IGF, a lot of what we've bred, or people have bred dogs for, is gigantism. And IGF is exactly the key to the pathway that is one of the few ways to tap into longevity experimentally. You manipulate the growth hormone pathway. So one of the things that come with gigantism is shorter lives. So maybe that's something that's going on in dogs. But let me just be clear. This applies across species. I can tell you zero about whether or not this translates directly to differences across individuals. There's so many other factors that come into play, especially as you start living that long. So many buses out there to run you over.
I just have a quick question. Because I looked at the curves and you have humans living up to 80 years old, and that's a relatively recent phenomena. Like the Egyptians were lucky if they lived to 30, from what I've read and stuff like that.
This is maximum longevity, maximum recorded longevity. It's not average.
Okay.
None of these measures would be perfect. For these goals, I think maximum longevity is the least bad.
So basically you believe that humans could not live infinitely, so if we got rid of...
Yes, not indefinitely, not infinitely. At some point, even we lose the ability to swallow, to control our pharynx, like C. elegans does in just 2 weeks. So that's one way. Okay, I am that undying optimist, and it gets unnerving at some times, but you could look at it this way. We could just go down really fast like so many other animals. The fact that we have so many neurons allows us to hang around for long enough that we can actually sustain, we can still live pretty decrepit lives, but still be functional, which you can choose whether you want to see that as a blessing or a curse, and we can, of course, all decide to do what we want to give everybody better lives. But that's the best of problems. Sorry.
So if I understand the argument correctly, which makes sense, this is a potential explanation for degenerative diseases in aging populations of humans. But I'm wondering, in this context, how this relates to psychiatric disorders where there's often an onset around sexual maturity, or certainly much earlier in life or in childhood, and whether you think these types of relationships in any way explain the timing of psychiatric disorder onset.
I think I agree with you that the obvious link is with any kind of disease or disorder that has age as a risk factor. But even like you mentioned, you have psychiatric disorders that the first symptoms will only appear when people are in their 20s maybe, or in their 30s. You consider that again, you're never going to get a rat or a mouse to live that long. We don't know at this point, I think, how much of a psychiatric disorder that appears only when you're in your 20s or 30s is something that surfaces only because now you have a system that is complex enough, that has enough neurons, that has enough connections, that has a working that is so incredibly intricate, and also that has benefited from all those 20 or 30 years of fine-tuning, that now you get to see problems that you would never see otherwise. I think that could explain a lot of why we don't see those things in other species, and why it takes a while for many problems to develop.
There we have a last question, and
Yeah.
then hopefully to hold your question.
No, thank you for a really great and really interesting talk. I was thinking about, as you're talking about risk for disorders, especially aging disorders, it seems to be, let's take something like Alzheimer's, where the vast majority of the genetic risk, which is a kind of, gives you a causal anchor, sits in neuroinflammatory cells and neuroglial cells, especially monocyte-derived lineages like microglia. And some of the data that you showed early on, you were mentioning that the glia can't account for things, but I was wondering, since there's a 4-to-1 ratio roughly, or of certain kinds of glia to neurons in the brain, there's a kind of established ratio. If the neurons are increased and the glial to neuron ratio stays the same, then the same thing would be happening, could be applied to glia, unless I misunderstood the way the calculations were done.
Yeah, it's very clear that the same does not apply to glial cells. With glial cells, if you want to look at numbers of glial cells, an excellent proxy for that is the simple size of the brain, or the size of the structure. So glial cells in numbers explain no more of the data or of the variation than brain size already does. And it's just to add to that, the ratio between glial cells and neurons is really not that high. And it's also, astrocytes are not the majority.
But it's constant.
It's not constant. It's highly variable across structures and across species, and it depends simply, apparently, on the average density of the neurons in the region that you're looking at. I'll be happy to tell you much more about that.
Let's thank you to
Thank
Susanna
you.
again. So we'll have 3 short talk from trainees. The first speaker is Joel Han.
Can you give me a second to say hello? Can you give me a second to say hello before you start playing? Yeah. Thanks. So thanks for everyone who's still here. I expect you're all fairly saturated and possibly hungry at this point. So I also want to thank David Leopold for slotting me in here. I'm not technically a trainee, in case you're wondering why I have so much grey hair. But I'm going to try and talk to you a little bit about some work that I'm doing. So Larry Swanson's keynote, for those who are here for that, acts as a sort of primer for this. So as the quotes at the bottom of the screen indicate, we still know little about hypothalamic development. So that being so, as a counterpoint to the developmental focus of this symposium, I'll talk briefly about my recent research to determine the network architecture of the mammalian hypothalamus. That may also guide future investigations into both hypothalamic development and psychiatric disorders. Hypothalamus is a relatively small part of the brain. Well over a century of research has revealed that it plays a critical role in the control of multiple functions related to body homeostasis and behaviors that are fundamental to survival. But to date, we have not had a model of its global network architecture. So I sought to construct such a model using data of monosynaptic connections between hypothalamic grey matter regions, macro connections in the rat, with reference to a current postulation scheme that identifies 65 regions on each side of the hypothalamus. So after extensive review of the primary literature, I was able to extract data for 88% of the 16,770 possible intrahypothalamic macro connections. The data indicated about 8,000 of these existed, giving a high overall connection density of nearly 50%. Connection matrix on the left represents these data, with each pixel representing data for a connection between two regions. The data is independent of left-right side and is arranged here according to the results of a recently developed algorithmic community detection method called multiresolution consensus clustering that Larry mentioned earlier. This method analyzes directed weighted connections between network nodes, regions, across multiple network resolutions, and generates a hierarchical nested network solution, the co-classification matrix and dendrogram on the right. The results indicated the existence of two top-level intrahypothalamic subsystems or modules mirrored on either side of the brain, each composed of two second-level subsystems. Here, the results are represented on a on a flat map for the bilateral hypothalamic HY2 network. The upper half here shows in blue two second-level subsystems, and the lower half indicates regional arrangement based on earlier structure-function model, actually in a 2000 review paper that was held up here at the start from Larry. And what I'd like to draw your attention to here is that one of the second-level subsystems in darker blue on the upper half corresponds mostly to the regions coloured yellow and red on the lower half, while the other, lighter blue, corresponds mostly to the pink, green, and grey coloured regions on the lower half. It turns out this division corresponds to a general division of hypothalamic function between control of body homeostasis and behaviour that I mentioned at the start. Bring this all together into a novel hypothalamic network model. The whole hypothalamus is richly integrated and has two major subsystems, one primarily associated with control of body homeostasis, the other with behavioral control. Moreover, the most prominent network nodes, indicated by the stars here, are associated with homeostatic control, suggesting network primacy of body homeostasis. Last but not least, the model reveals previously unrecognized subnetworks that offer many new avenues for further research. To close with a single example, these three regions form a subnetwork that includes the ventrolateral part of the ventromedial hypothalamic nucleus, VMHVL, and two adjacent regions of the LHA, lateral hypothalamic area. Recent research on the VMHVL has highlighted its apparent involvement in control of both aggressive and sexual behaviors. Given network association of these three regions, it suggests the roles may be related. Additionally, a few years ago, I determined connections of these two LHA regions, both of which are highly connected, and notably received direct substantial cortical input. This organization has potential relevance for research into psychiatric disorders affecting aggressive or sexual behavior because it raises the possibility of targeting intact components of a behavioral control network at the level of the hypothalamus to circumvent or counteract cortical dysfunction. With a view to eventually obtaining a network model for the nervous system, we're currently working collaboratively to obtain region-level subconnectomes for the other parts of the brain. Thank you very much for your attention.
If you have any question, we can take one. And
so I don't know whether you contain the cortical connections. So my question is whether your matrix contained the direct cortical connections, because it is known, and I also recently did some monosynaptic rabies tracing from different cortical cell populations, that the lateral hypothalamus does have a direct connection to frontocortical area.
Yes, absolutely. So in fact, if I can get this right here. So in fact, this illustrates some of that. So the connection matrix that I've described is just for the hypothalamus, so it's intrahypothalamic. So one of the future goals is to look at the network for the telencephalon, or cerebral hemispheres, with the hypothalamus and the other parts of the brainstem. So that's certainly a future goal. But you're absolutely right. There are plenty of direct connections between the cerebral cortex and the hypothalamus.
Thank you.
Thank you.
Our next speaker is Lindsay Cantine from Rockefeller.
Hello. So I'm a graduate student in Eric Jarvis's lab at Rockefeller University, and my project is mainly focused on studying epigenetics and gene regulation in vocal learning species. And today I'm specifically going to talk about the epigenetic specializations in the zebrafinch song system. So as Eric had previously talked about, in a study that we did when we were comparing the songbird song system and the human speech regions, we found gene expression correlations between different regions of these two circuits. One of the more significant ones is the RA to the human laryngomotor cortex. So this actually, it makes sense because the RA is and the LMC are found in the same positions within their circuits, and they also have similar functions within their circuits. So my project is mainly focused on trying to identify what is causing these gene expression specializations through identifying gene regulatory regions. And the data I'm going to show you today is going to be focused on the RA data that I have, and then comparing that to the surrounding motor region that Eric was talking about before that we use as a control. So my method is using ChIP-seq, which with an antibody against H3K27 acetylation. So this histone mark is associated with active regulatory regions in open chromatin. So I can pull down regions of the genome that are associated with this histone mark, and then combining this with next generation sequencing, we can look to see where this mark is present across the genome. So to do this, I also did microdissections of the RA and the surrounding region. And then when I look at my ChIP-seq data, just overall, I see about 100,000 H3K27 acetylation peaks total for RA and LAI each. LAI is what we call this surrounding archopallium motor region. And we also find that there are about 70% common peaks between them and 30% unique peaks, so differential peaks. And I also find that the peaks generally are enriched around transcription start sites, which are associated with promoter regions. So now I'm just going to go in and focus on a couple of the genes that we're really interested in right now. So here we have a track showing the SLIT1 gene, and then here is the RA H3K27 acetylation peaks and the LAI H3K27 acetylation peaks. So SLIT1 is a repulsive axon guidance gene, and it is downregulated in the RA and RA of songbirds and the LMC of humans. And what I find is a number of unique peaks within the LAI region that are not present in the RA region. So these could represent putative regulatory regions that are involved in driving the expression of SLIT1 in LAI, but not in RA. Another thing that I should have included that I just want to note is when I look at genes that are expressed, that are not specialized in these two regions, so they're expressed the same within RA and LAI, I don't see any differences in peaks. And then another gene that I wanted to talk about is NeuroD6, which is a transcription factor that has previously been shown to regulate SLIT1. This also has a similar expression pattern as SLIT1 does in zebrafinch and humans. And again, we're seeing these unique peaks within LAI, so these are going to be putative regulatory regions that we'll look at further to see which sorts of transcription factors are binding there, which may be leading to the specialized expression. Another thing that I wanted to comment on is some of these peaks within the NeuroD6, within or near the NeuroD6 gene, are actually associated with nucleotide mutations that are found specifically in vocal learning species, but not in non-vocal learning species. So it's possible that these mutations are potentially causing different transcription factors to bind, and that's definitely something that we want to look at further. So in the future, my plan is to then repeat these experiments with human brain regions, such as LMC and the surrounding region, as well as looking throughout development within the zebrafinch to see how the activation of different regulatory regions are changing throughout development, leading to this specialized gene expression that we're seeing across the species. Yeah.
Okay, so then we'll move to our last speaker, Lin Hezhu from the Vanderbilt.
Good morning. So we're interested in diversity. There is about 1,000-fold variation in neuronal density across brain structures and species. But just like neurons come in various shapes and sizes, but glial cell types are much more homogeneous in density and morphologies across species. How does so much diversity appear in evolution only for neurons? Our hypothesis is that neuron-specific genes are less conserved. That is, they're more variable than glia-specific genes. And to assess the level of conservation, I compared the NDS scores for neuron-specific genes and glia-specific genes across species. So first of all, I defined the top 500 neuron-specific and glia-specific genes according to the Barris data set. And then I did a data mining through the Ensemble 93 database for the NDS score. Here we see a full distribution of the DNDS scores. We have expected, as our prediction goes, that the DNDS value to be higher for neuron-specific genes compared to glia-specific genes. However, what we see is that the distribution of DNDS is shifted to lower value for the neuron-specific genes compared to other non-neuronal specific genes, which means neuron-specific genes actually turn to be more conserved, which is the opposite of what we expected. So, the differences in the distribution is even more clear in a frequency histogram of the DNDS value for genes specific to each cell types. The significance of this difference is also confirmed by a Wilcoxon test. There are more neuron-specific gene than glia-specific gene with very low value of DNDS, indicating highly conserved neuron gene. Likewise, there are more endothelial cell-specific genes and the glia-specific genes with higher levels of DNDS, indicating highly variable glial and endothelial cell-specific genes. Then what we wonder, what are those genes? So I look at the biological gene ontology terms for each and every single gene that we analyzed. And we found that the neuron-specific gene with low DNDS are likely to be related to transport or the response to stress geoterm. In contrast, the glia-specific genes that have high DNDS scores are related to immune system process, and we find that those are particularly enriched in microglia. The answer to our question of whether the higher diversity of neuron type, morphologies, and sizes is related to a higher variability of coding sequence of neuron-specific gene, the answer is no. So, and also, so far the most diverse gene category across species is immune system process in microglia cells. So, where the differences in variability between cell type come from? There are multiple possibilities. It could be just the developmental sequence that's different. There could be a small, particularly limited set of genes that are contributing to this, or maybe glial cell diversity is just much higher than we previously has suspected. And thank you, guys.
Great. Any question? Two questions. Okay.
One of the major, kind of tenets in kind of modern evolutionary genomics is that it's non-coding regions that are really the most divergent, and it's also very well established that brain genes in general are under purifying selection, which is what you see for the neuronal genes. Very well established. And so I'm wondering what the motivation is for looking kind of at DNDS in terms of diversity when really the main areas that are driving the differences have to do with driving regulation of gene expression and splicing.
Yeah, thank you very much for the question. So to be honest, before I start looking at the DNDS, I really expect to not see a difference because exactly as you said, we expect it to be the non-coding region. But what we see here is that there is a significant difference in the DNDS scores.
You're right, but that's been shown before. Brain genes are under purifying selection. There's dozens of, I don't even know dozens, it's well known. I guess, so I'm not sure why you were surprised. So it's like, I guess, a two-part question. One is, that, as far as I know, that's pretty well understood, but are you planning to look at the non-coding regions?
Yeah.
Yeah, no, I had exactly the same question, and my prediction is you're going to see the reverse result in the non-coding sequence.
Oh, you mean the level of conservation will be the opposite? Yes. Okay. That would be a next step for me to look at. Thank you.
Okay, so this ends our first session. So that I would like to thank all the speakers one more time for giving great talks.
Welcome to the second session of this today's symposium, and I'm Quan Hong Wang from NIMH, and to continue the exciting conversation we started in the morning, which is on the brain architecture viewed through the lens of Evo Devo. In this second session, we'll focus a little bit more on the mammalian brain circuits development, particularly using mouse as a model system and combining the amazing breakthroughs in the modern mouse genetic manipulation, and to examine how the mammalian circuits develop in space and time. And we are for a treat from four really pioneers in this field. And they are the first speaker will be John Rubenstein from UCSF. The second one, Zoltan Molnar from University of Oxford. And the third one, Takao Hanch from Harvard University. And the fourth one, Mariganka Sir from MIT. Followed by three data blitz from trainees, Jen Fisher, Jeannie, sorry, I cannot now see the name very clearly here, but you can read from the slides here. Then after this data blitz session, we'll open up for a half-hour discussion, and the speakers today will come to the front, sit in chair, and there will be an open panel discussion. With that, I would like to welcome the first speaker in the afternoon section, Dr. John Rubenstein from UCSF.
Thank you, Quan and David, for inviting me to your fantastic assembly of diverse experts in various aspects of the development of the brain. And as many of you may know who know me, know that my work is interested in generally early molecular mechanisms of cell fate specification in the developing basal ganglia and cerebral cortex, But the title that I gave to Kwan and David a year ago evolved a little bit since we had some progress in a project that Siavash, who's sitting out there, succeeded in getting published. And I'll be telling you about a story that goes back to the beginning of when I became a professor, the function of TBR1 transcription factor. And for those of you who don't recognize that view, that's the view looking out the Golden Gate. And it's poignant because that picture was like 2 days after the rains came and we stopped being the most polluted city in the world and finally had resolution of this horrible disaster. That part, at least in San Francisco, not in the Sierras. Okay, so we started working on TBR1 around 1987. And when I was searching for transcription factors that were regionally and temporally expressed in developing telencephalon, thinking that those would be good entry points to understand the genetics of cell fate specification in the telencephalon. And we isolated these telencephalon embryonic subtraction genes called test genes. And this was work done by Matt Porteous, my first grad student. Ted Hudson, I don't know if Ted's in the audience, but he works in this building. And I wanted just to shout out thanks to Ted. And Roland Ciaronello, who was my mentor at that time. And our hope was that some of these genes, or the pathways that they regulate, would eventually be implicated in neurodevelopmental disorders. At least that's what we wrote, probably in the first or second line of the grant application. And I know many people didn't believe it would happen. And also I had fun naming the genes. The number 56 gene was TBR1, and the first gene was DLX2. And so the test genes were also the name of my daughter, Tess. And the test 56 is named after Thomas Braden Rubenstein, who's in that picture there. Okay. So these transcription factors, TES1 and TES56, or DLX2 and TBR1, turned out to be interesting molecules because they are very specific in regulating GABAergic cells or glutamatergic cells in the developing telencephalon. So it was among the first evidences for distinct genetic pathways for these cardinal differences in cell fate specification. And just a little history of TBR1. So we first published it in, I guess, 95. Work by Alex Bulfone, who was my first postdoc, and he showed that it was expressing in glutamatergic neurons. In fact, the earliest born neurons, the cortex, the Caja cells, the subplate, and layer 6. And then shows these Caja cells, the preplate, and layer 6. And then Robert Hebner came to the lab and showed that TBR1, by virtue of its function in the subplate and the layer 6, controls the outgrowth and directed growth of cortical thalamic axons, and the axons get stuck in the internal capsule and don't make it to the thalamus. And then Robert went on to show that if the axons don't get to the thalamus, that Zoltan Molnar was right, that then the thalamic axons won't go to the cortex via this handshake hypothesis that Zoltan proposed. And then Ben Chen's lab at UC Santa Cruz kind of took the leadership and let me stay involved. And she showed how TBR1 and FESF2 have complementary functions. TBR1 for specifying layer 6 identity and FESF1 for specifying layer 5 identity. And then our hope came true that one of the genes we worked on would become a disease gene. And this is through work on the de novo mutations in autism. And Matt State and other laboratories found that TBR1 is a high confidence autism gene. Here it is circled by red. And so
now it became interesting again for me to get back in the game and do more work on TBR1. Matt State with Jeremy Woolsey provided evidence with Ninad Sestan that TBR1 is part of a co-expression network in mid-fetal life in deep cortical glutamatergic neurons, suggesting that there's a cluster of genes that all affect the development of these projection neurons that contribute to autism. So the objectives of our current study with with TBR1 was to elucidate later developmental functions of TBR1, because in our first studies we used a constitutive mutant that removed TBR1 function in the germline, and so the phenotypes we would see were phenotypes that we were accumulating beginning around embryonic day 10.5. So to understand what the phenotypes would be at later stages, we needed to generate a conditional mutant. We were also interested in phenotypes that might be related to autism, contributing to autism symptoms. And in this regard, we needed to pay attention to phenotypes that one would see in heterozygotes, because autism patients typically have a change or loss of function allele as a heterozygote state, not as a homozygote state. And then we wanted to get into the genomics of how TBR1 works and to understand transcriptional networks downstream of TBR1 that might link TBR1 function with the expression of other autism genes. I can see I'm one slide behind in all of these. So we've generated a conditional mouse mutant. This was done in collaboration with Matt State and Bin Chen. It's a typical Floxed allele where we recapitulated the deletion of the exons that we'd made in the constitutive mutant. And Siavash Darbandi went on to show that the mutation behaves as a null mutation by virtue of loss of TBR1 RNA and protein. So now that we had a conditional allele, we could investigate its function in different cell types. So we started by deleting it only in layer 6 and the subplate using a Cree called NTSR1 Cree. And Siavash showed that this Cree turns on around embryonic day 17.5. So the deletion basically removes TBR1 function about 7 to 8 days after it already has begun. So we'll find out what happens when we lose TBR1 almost 10 days after its initiation, whether or not TBR1 function is required at these later stages. Siavash began by doing a transcriptomic analysis of the changes of RNA expression in these layer 6 cells, and the computational work was done by Jeremy Woolsey and Chihao Chi. The cells we were working with were fluorescently labeled by an endogenous T2 tomato allele that was recombined by the same Cree. And Siavash would purify the cells and then use fluorescence-activated cell sorting and then generate the RNA and then do batch RNA-seq on them. On your left is the volcano plot showing that there are quite a few genes whose expression are either up or down regulated. And computational analysis showed that, in general, the effect of the mutation led to a modification of the fate of layer 6 cells such that they now had a mixed fate. They lost a lot of layer 6 identity and they gained a lot of layer 5 identity, but they also maintained some of their layer 6 identity, and that's what those two rectangles to your right are showing. So for instance, if you looked at markers of layer 6 by in situ RNA hybridization at postnatal day 3, I'll find it. This one here, FOXP2, is a good example. You have nice expression in layer 6, and in the homozygote mutant, it's greatly reduced. And the same can be said for all the markers that are shown here,
to varying degrees. Now, with regard to markers of layer 5, BCL11B, FESF2, and FOXP1 are all expressed in layer 5. Let's look at the FESF2, which is crystal clear result. In the wild type, layer 5, and in the mutant, you still have layer 5, but now the layer 6 cells express high levels of FESF2. And one of the fascinating things that Siavash found was that even in the heterozygote, the ectopic expression of FESF2 in layer 6 is very easy to see. So that means that dosage of TBR1 is exquisitely powerful in controlling the properties of layer 6 neurons. So now we know that even in heterozygotes, layer 6 neurons have some layer 5 properties. And so let's go forward and see what other properties change in these heterozygotes and homozygotes. But based on the based on these RNA expression changes, we have a slew of genes that are positively regulated by TBR1 and another group of cells that are negatively regulated. And those genes that have asterisks to their left are autism genes. So there's a lot of autism genes that fit into this pathway of being targets of TBR1, or I shouldn't say targets in this case, genes whose RNA levels are changed in the TBR1 mutant. A brief aside is that even cortical interneurons are affected in the TBR1 mutant. The cortical interneurons do not express TBR1. In this case, we're looking at somatostatin interneurons in the control, wild type, heterozygote in the middle, and homozygote on this side. And you may be able to discern that there is a change in the distribution of these blue interneurons, these somatostatin positive interneurons in the mutants. And this was quantified by Siavash for the superficial layers, layer 5 and layer 6. And in most of these layers, there are coherent changes in both the heterozygote and the homozygote in the distribution of interneurons. So much as Paula Alarta showed some time ago, that when you change the position of a cortical pyramidal cell or the properties of a cortical pyramidal cell, cortical interneurons may also follow, and that's what we think is happening in this case. But we don't know what the molecule or molecules are that have led to this interesting laminar change in the interneurons. Okay, so now we looked at the ability of TBR1 to bind to the genome in vivo. We used chromatin immunoprecipitation and whole genome sequencing to determine where TBR1 binds. And this was done by Siavash, and our computational collaborators were Alex Nord and Ronaldo Catapreta at UC Davis. TBR1 is a very robust chromatin binder. It's not binding 1,000 places, it's binding about 20,000 places in the genome, which makes the interpretation quite complicated. But we do get very beautiful peaks of binding that are highly statistically significant. This is the binding site that TBR1 binds, and it binds to the transcriptional start site. It binds to locations within the gene or outside of the gene, and so it's all over the place. But once you start looking at interesting loci, you can see that this binding does not seem to be random. There's binding to promoters, and there's binding to known enhancers. So these are some of the genes whose expression changed in the, expression change in the in situ. I know you can't see it, so we just picked one of them, FOXP2. And so here's the FOXP2 locus. And the green and yellow places are peaks that Siavash cloned to test whether or not these candidate regulatory elements could in fact change gene expression.
And so what Siavash did was he, for these group of genes, as well as for including FOXP2, cloned about 15 to 20 of these candidate enhancers and tested whether or not TBR1 can regulate them in primary neocortical cultures. So he built a construct where he would put these candidate enhancers upstream of luciferase, and then he also built a construct that has TBR1 being expressed. Okay. I thought I was going to show one thing else. So in these assays, the neocortex from neonatal mice, grew them in culture, and then transfected them either with the enhancer construct or the enhancer construct plus TBR1. If it just has the enhancer construct, it's the brown bars. And if it's the gray bars, it has the enhancer plus TBR1. And it shows that in the cases of these four enhancers, TBR1 can amplify the activity of these enhancers, providing evidence that they are, in fact, positively regulated enhancers. And these four enhancers come from loci of the genes that were downregulated in the TBR1 mutant. So these would be cystic regulatory elements that through which TBR1 increases the expression of the target genes. And so they would be for genes like FOXP2 or BCL11A that have decreased expression in layer 6. On the other hand, when he did this with enhancers for genes that were downregulated in the TBR1 mutant, he found in each of those cases that adding TBR1, the gray box, reduces the level of activity of the enhancer, providing evidence that TBR1 is acting as a repressor in this case. And that was the case for FESF2 and FOXP1, genes that were very upregulating the mutant and therefore are normally repressed by TBR1. So we found evidence that TBR1 can be both an activator or a repressor, and that information is encoded by the enhancer element and not by TBR1 itself. Next, we looked at the phenotypes of the dendrites of the layer 6 cells at postnatal day 3, postnatal day 21, and postnatal day 60 in controls, heterozygotes, and homozygote mutants. For those of you who can't see it, normally layer 6 axons grow to about the junction of layer 5 and layer 4. But in the TBR1 null mutant, these layer 6 axons grow all the way to layer 1. And that growth to layer 1 is a phenotype of layer 5 neurons. So this is further evidence that the layer 6 neurons are changing fate towards a layer 5 type of pyramidal neuron. And also what Siavash found is that the heterozygotes show an intermediate phenotype here, that there are ectopic apical dendrites in the heterozygote that go to layer 1. So we think that the model that there's a very strong bias of the mutant layer 6 cells to take on layer 5 properties is correct, and that heterozygotes show this phenotype. Another cardinal phenotype of TBR1 mutants is that their layer 6 axons and their subplate axons cannot grow to the thalamus. In this mutant, however, those axons actually do grow to the thalamus, and that's probably because we don't remove TBR1 until those axons have already gotten there. However, within the thalamus, the ability of those axons to arborize and synapse is greatly diminished. So this is a control. On the top are controls, and the bottom of the mutants, and you may be able to notice where these white arrows is that in the medial parts of the thalamus, there's much reduced axon arborization of the layer 6 neurons. Then we turned our attention to electrophysiological analyses, and these were done with Vikas Sohal and Sarah Robinson at UCSF.
we also looked at synapses by immunohistochemistry, and we concentrated on the synapses in layer 5 of the apical dendrites because the controls would have layer 5 apical dendrites, but not further, more superficially, like the mutants that grew up. So we did our control layer 5 as well as the mutant. And Siavash found by immunohistochemistry, looking at excitatory synapses, and I'll just show you the quantification. Wild type is red, green is heterozygote, and blue is homozygote. There is a decrement in both the heterozygote and the homozygote for the number of excitatory synapses. Consistent with this anatomical finding, Vikas Soha's lab found a decrease in the spontaneous excitatory postsynaptic currents in the heterozygotes and the homozygotes. So both by electrophysiological assays and by immunohistochemistry, the pyramidal cells have less excitatory synapses. And the same is true for inhibitory synapses. So whatever it is that the TBR1 mutants don't have for making synapses, it's a general problem for both excitatory and inhibitory synapses. That's the quantification for the inhibitory synapses. And electrophysiology bears that out as well for the inhibitory synapses. So I'm going to briefly say that we searched for mechanisms that underlie the reduction of synapses in the TBR1 mutants by carefully looking at the genes whose expression was changed in the RNA-seq analysis. And one of the genes that Siavash found was Wnt7b, whose expression in the wild type is very strong in layer 6, and in the heterozygote, and especially in the homozygote, its expression is greatly reduced. And tomorrow, you're going to find out from Siavash, when he gives his 5-minute talk, whether or not Wnt7b or any of the other candidate targets of TBR1 are the magic bullet that controls their synapses. So in summary, I'd like to just recall to you that TBR1 is required to maintain fundamental aspects of layer 6 identity. The transcription tractor code of those cells is altered toward layer 5 identity. Direct TBR1 gene regulation on the chromosomes does this through binding to regulatory elements, enhancer-like structures, for which we've identified several now. The transformation to layer 5 identity is very clear when one looking at the dendritic morphology as the apical dendrites now grow to the superficial parts of layer 1. I didn't tell you about this, but the H current, which is a characteristic of layer 5 cells, is now found in the layer 6 cells. And the corticothalamic innervation of the thalamus, while partially present, is still greatly abbreviated, showing that the layer 6 cells are not themselves. So a timeline of the phenotypes of TBR1. If you, I'll put my glasses on, I'm sorry, I can't even see this. Okay, so we've deleted TBR1 at E17.5. And then we were able to find that even 10 days after its initiation, its maintenance function is totally required in order to keep these cells in the right fate. And that wasn't always clear. Often one thinks of a transcription factor and a fate change being somewhat permanent. Well, that's not the case of these layer 6 cells. They require consistent TBR1 presence, at least through the stages that we studied. Morphogenesis of apical dendrites. That is plastic as well under the conditions of this experiment. Here I go again. This is very common, the reduced density of synapses on the apical dendrites. I have a feeling that this may be a big part of what's wrong in the autistic children who have the TBR1 mutation. They really have a huge decrease in the amount of synaptic input onto their pyramidal neurons that depend on TBR1. They have this increased H current, which is an intrinsic physiological characteristic of layer 5 cells. Now that's in layer 6 cells. And I didn't show you the data, but these animals have increased anxiety, perhaps related to autism, who knows. TBR1, we're beginning to get a circuit diagram for what TBR1 does in terms of activating and repressing genes, many of which are autism genes, including SCN1A, BCL11A, FOXP1, Wnt7b, NUOC1, and SPARC1. This is a high confidence and this is a low confidence autism gene. So TBR1 is required for maintaining the normal levels of excitatory and inhibitory synapse numbers on layer 6 pyramidal neurons. And you'll see tomorrow that TBR1 may do this through its regulation of Wnt7b and perhaps other genes to control this process. And I'd like to thank Siavash and the other people who I've already mentioned.
Differential reduction of the corticothalamic feedback to the thalamus. It seems like that particular image that you shows, for example, has BPM has more or less still the feedback axons, but not the medial part of the thalamus. Is it related to their development, temporal development, so that before P7, I forgot when the TRB1 kicks in, so corticothalamic feedback for a more lateral part, for example, like BPM, already has their axons back there.
Yes.
Versus the one projected back to the more medial part of the thalamus comes later, and that's why you have a greater reduction there, or is it something else?
I think that's right. I probably Zoltan may know more, but I think the ingrowth of the thalamic axons is from lateral to medial, and that would explain how you have kind of they stopped.
I see.
Yeah, I was wondering more about the behavioral changes you're seeing when you say anxiety and aggression. Overall, I'm a little surprised that the animals survive without a normal layer 6 connection, although I guess you can decerebrate some animals and they survive. So I'm just wondering if you think if there's a gross overall, let's say, behavior or types of behaviors that layer 6 is controlling that you be able to use these
I'm kind of a nihilist about behavior, and at least these kinds of behaviors in mice, mutants. I don't actually feel like I understand it, except to say I think the animals are confused, and I think that when one's confused, one gets anxious. So I would say that, and with regard to the aggression, I have no idea, because we did not mutate TBR1 in the amygdala, nor in the prefrontal cortex. So it's very hard to understand that particular phenotype.
I was looking on the list of affected things for something like axonal pathfinding.
Yeah.
And I guess what I'm wondering is for the constitutive case where the axons don't make it at all to the thalamus, and then for the conditional where they make it but don't seem to go far enough, do you think that's for the same reason? And what is the reason for that?
I can only speculate. We don't know what the molecular mechanism for axon pathfinding, axon growth regulated by TBR1 is, although
Sivash and I are working our way through some of these target genes. One of them, the NUOC1, which is an autism gene, but is a gene that Franck Pelot works on. And Franck has told me that that gene is quite important in axon growth. And so one could speculate that at least a component of how TBR1 controls at least generic growth of thalamic axons is through NUOC1. The TBR1 mutant, constitutive mutant, I think those cells not only have a problem with growth, but also with pathfinding, with sensing and turning, because there's quite a bit of gymnastics as they go through the internal capsule and loop back into the thalamus. And I don't know what part of the pathfinding parts, molecular pathfinding mechanisms, would be TBR1 dependent. A bientôt.
Do they sleep?
They sleep. I would have to pass that on to Sivash, who lives in the mouse room. Do they sleep? Yeah, they sleep.
So the reason why I was asking, we silenced some layer 5 populations using conditional SNP25, and they actually sleep much less, and they also don't have a nap in the active period, which is the evening. So basically, tinkering with cortical circuits can actually alter the sleep, which is, for me, was very surprising. But we didn't do it with layer 6 so far. 6B, they don't care, unfortunately. They sleep just as much. Yeah.
Sivash is doing the layer 5 mutant as we speak, so you can ask him about that next year.
I think it's particularly exciting that you can see a phenotype in the heterozygotes, because that's sort of been challenging in this field. Have you been able to determine in those layer 6 specific knockouts what the actual protein level of TBR is? Is it really 50%, or is it more or less?
I don't think we've done a careful job on quantifying the protein concentration in the heterozygote, and I think we should. Mark it down. Yeah. Yeah. We haven't done it, and we should do it. We haven't done it, we don't know.
I have a question.
Yeah.
For the conditional one, have you compared with the initial? Compare this conditional one with the initial 3 knockout. What is the major difference in phenotype? Yeah.
Well, the initial phenotype basically died of day of birth. So phenotypes such as the dendrite and the synapse, things that we discovered we would never have found. With regard to the cell fate change, it's very related, although not as severe as in the null.
Thank you for that great talk. It was really, really interesting. And I guess I have a comment as much as a question, which is, it seems as though your research is showing us that in some ways it's difficult to identify the causal link between the up or down regulation of a gene and the downstream effects. Because throughout your talk, it occurred to me that we really couldn't tell whether it was due to the direct effect of the gene on its downstream genes that you pointed to, or whether there was some event in the extracellular environment which in turn fed back on the neuron to alter its gene expression.
What do you mean by that?
Well, I guess what I'm saying is that well, what am I saying? I guess
we're looking at the gene. We're looking at the gene expression at P3 before there's a major amount of synaptic information.
Could you repeat that, please?
Well, with regard to the environment feeding back on the cell, at least with regard to synapses, electrical information, we're looking at P3, which is before there's, I believe there's a lot of synaptic information coming in. So I feel like we're mainly looking at intrinsic properties of the cells, although there could be effects of, at a cell biological level, of contacts of adjacent cells that are responding. But I don't think there's so much
with regard to the environment via synaptic information.
Okay, fair enough. Thank you very much.
The next speaker will be Mona.
Can you give me my power supply, please? Abento. I'm working on this. You want to use mine? No, it's okay.
First of all, I would like to say thanks to the organizers for the wonderful meeting and your hospitality. I'm going to compare development of very different species. If you look at a kangaroo and you slice up the brain and you stain it for fiber, as I did, you have a 6-layered cortex. You don't have corpus callosum. You have a huge anterior commissure. But it is still very similar to all other mammals. If you have a look at the brain of a turtle, you notice that you only have a very thin layered dorsal cortex, and you have this bowl protruding into the lateral ventricle. If you stain the brain of an iguana and stain for fiber, you see that you have an even bigger bowl protruding into the telencephalon, and the crocodile an even bigger one. So the question is, what is this bowl of cells which is protruding in the lateral ventricle, which is called the dorsal ventricular ridge, or woost, in birds? So, and what is this bowl doing? So if you want to compare the development and to look into the forks of evolution, you have to compare several species. And I am going to compare species which live today, but I don't want you to think about it that I think about evolution like this, that, you know, a turtle brain became a mammalian brain, or there was an expansion of a rat brain into a monkey. You know, they evolved just as much, and their development diverged. And this is how we ended up with these arrangement. So it was Dobzhansky who said that nothing in biology makes sense except in the light of evolution. And apparently it was Leah Krubitzer who said that nothing in biology makes sense except in the light of development, which I copied. And apparently it was my student, Thomas Lickis, who said that nothing in biology makes sense. and the reason why I emphasize this point, that we have to look at development, because this is how the brain is evolving. You have to change the development so that you will have a different adult structure, and then you will get selected out whether that structure is working better or not. So if you look at the adult brain of a chick and the mouse, the anatomy is very difficult to, you have to be Luis Puelles to understand what is what here. And basically nobody is arguing about the subpallium, that this is probably corresponding to this part of the brain, and nobody is arguing about the medial pallium. So these structures are considered homologous. But once we get deep into this bowl of cells, the woost, it's becoming very complicated, and we have no idea what is homologous to what. You heard very nice talks today about the function of this bowl of cells, and I must say that I personally believe that with this bowl of cells, the DVR, you can do a lot. And with Paul Menger in Leah's lab, we recorded from iguanas. And if you look at the visual representation in the iguana brain, it's a very reputable visual field. You have at least 3 different visual fields. There is a mirror reversal within the bowl of cells. And if you compare these receptive fields with the ferret visual field, then it's extremely similar. So it's a very reputable visual system. So these cells do act like cortex. and they can do quite a bit with it. Now, how about the connectivity? So Harvey, Carter, and many others looked at the connectivity of these structures, and indeed the DVR is receiving connections from the thalamus, which is very similar to the connections the cerebral cortex would receive. But I want to point out even here that the amygdala is also receiving similar, lateral amygdala is receiving similar connections from the thalamus. So, so this bowl of cells could get very similar input to the cortex. And this really, these functional similarities, some gene expression similarities, and the connections prompted Harvey Carton to suggest that you have an equivalent circuit in the sauropsids, so the bird and the reptilian brain, and these cells don't line up into layers, but you have this group of cells, but basically you are dealing with a very similar equivalent circuit. And this is a very powerful suggestion because indeed there are lots of similarities. So in mammals, the thalamus is projecting to layer 4, and then it feeds into the supragranular layers, and then they project back via the layer 5, 6 to the brainstem, thalamus, spinal cord, basopons, colliculi. And the same is true for birds. The thalamus will target certain cells, and then they interconnect with neurons which don't leave the brain, and then you have output. So what I'm going to do today is I shall revisit some of the literature, some of that is old literature from me and others, and I look at gene expression patterns of these regions, and I try to compare them. Then we will look at single-cell data, because now there are some single-cell data in mammals and also in reptiles. And then I will look at the specification of cortical progenitors and ask the question that how would a chick progenitor in that part of the telencephalon differ from a mammalian progenitor? And do you have more specification in the mammalian cortical progenitors? And then finally, I shall ask the most important question for a developmental neurobiologist. Homology means that whether that particular cell group is originating from that particular segment of the neuroepithelium. So that is what we call homology. And that's what I will look at in the last part. If you look at the neural tube, so all nervous system in all vertebrates, they start from a tube. And the rostral end will enlarge, and you will have a telencephalic vesicle. And this part of the telencephalic vesicle will become the cerebral cortex. And if you look at this zone with time lapse, you can see that most of these cells divide here in the ventricular surface. So this is where the PL surface is, and then the cells will migrate out. And Rosalind Carney, when she was a graduate student in my lab, she stained some of these brain sections with phosphohistone H3, an epitope which is only expressed in dividing cells. And then you can see that only here in the ventricular zone you have most of the divisions, but you also have this subventricular zone, so you also have divisions here. And the same is true in actually the developing human. While these cells divide, they express certain transcription factors, and you just heard from John, and in fact this is his figure, that during the division you will have these waves of transcription factors expressed in the ventricular zone, and some of these transcription factors, they will be expressed also in the postmitotic neurons. And that's very handy for us, because then we can label these cells in the brain, and we know that OTX1 is staining layer 5 projection neurons, ER81 also another subgroup of layer 5, ROR beta, layer 4 thalamic recipient cells. So the question is, if we use these markers, can we identify homologous regions in the avian and reptilian brain? So when subplate neurons are born, they have SOX5 expression, layer 6 TBR1 and others, of OXP2, layer 5 CTIP2, and supragranular layers, they have SETB2. And what I want to show is that between these transcription programs, you have a cross-suppressing effect. So if you start moving out some of these transcription factors, then you will change the patterning of the brain. So if you remove SOX5, then the CT2 program is going wide, and you will have layer 5 cells all over the cortex, because there is a cross-suppressing effect between these transcription factors. And then if you remove SETB2, then the layer 5 program will last longer, because SETB2 is suppressing CTIP2. And there is also COOP-TF1, which is a layer 5. So this is how you change the proportion. And I will come back to this. This transcriptional program is not present in this format in the avian brain or in the reptilian brain. And this will have implications in the projection neuron specification in those cortices. So let's have a look at some of these gene expression patterns in the avian and the reptilian brain. And two groups did that. They did exactly what everybody wanted to do, is to clone up some of these transcription factors in turtle and have a look at the expression. So I would have selected also the same transcription factors, but these were restricted to these. And then they looked at the gene expression. And indeed, some of the genes suggested that Harvey Carton's ideas were right. For instance, some of these thalamic recipient regions, they expressed ROR beta, and you could make an argument that these genes actually revealed an equivalent circuit. And you heard from Eric his very extensive studies when he looked at immediate early gene expression after certain stimulations and looked at also gene expression, and he suggested that you almost have a columnar organization within these bows of cells, and the cells come from different parts of the telencephalon, medial pallium, ventral pallium, subpallium, but nevertheless, they form a functional unit which is very similar to the columns of the dorsal cortex, which is, of course, they develop very different manner. So we wanted to approach this in a different way. We wanted to dissect different layers of the mammalian cerebral cortex from the adult, and also hippocampus, striatum, amygdala, claustrum, endopiriform nucleus, and then get some markers for these and compare all the transcriptome with the transcriptome of the avian brain. So I did these experiments with Grant Bellegarde and Chris Ponting. So Grant was a NIH Oxford joint student. So this is a really successful program. They can spend 2 years in Oxford and 2 years at the NIH, and then you usually have complementary skills. So Grant came to us and said, what would you consider the biggest challenge where I can use my bioinformatics? And by the way, Grant then went with Dan Gershwin to UCLA after his studies, and now he has his own company. So what Grant did was he used all sorts of techniques to predict new layer-enriched genes, and he also used the LN database, and then we had over 3,000 layer-specific markers after his studies. And we discussed quite a bit today about susceptibility genes and their associations to certain layers. So Grant predicted that Alzheimer networks are more prevalent in layer 2, 3, and also in layer 5 in his studies. And we use this mammalian database, which is still alive, to compare it with the avian database. And so we had to drive out to a chicken farm in Oxford, and I seriously contemplated to become vegetarian after that experience. And we dissected hundreds of chicken brains. And from these regions, we dissected different regions, and these were sequenced here at NIH. and Grant looked at the transcriptome and looked at the similarities and differences of the transcriptome. And he found that the avian transcriptome very, very different from the mammalian. He did 4 types of comparisons, so specificity comparison, marker comparison, compared the networks, and also looked at the preservation of modules within these networks. And we managed to identify several genes which were specific for certain parts of the pallium, so hyperpallium, mesopallium, nidopallium, striatum. And now we could work with hundreds of genes which were enriched in these and could be distinguished from other parts of the avian brain. And we asked the question, what parts of the mammalian brain will be similar? Which one will cluster together? And what was immediately obvious, that the striatum clustered with striatum, hippocampus with hippocampus, oligodendrocytes with oligodendrocytes, and the only other clustering which we could see was that the layer 4 of the neocortex was clustering with the nidopallium. Exactly it's the same region which is receiving the thalamic input. Then Grant did the network comparison. So he was looking for cross-species overlap of the gene co-expression modules. And then he looked at also the preservation of the modules. And the striatal gene co-expression modules were very similar. And we validated many of these genes also with in situ. The hippocampal was also very apparent, and the layer 4 nidopallium. And again, you can see here the ROR beta. And this was one of the genes which the other groups selected for the in situ. So coming from the other end, we could also pull out these similarities, but the rest of the brain, we did not have more similarities. So we couldn't, for instance, relate subplate to a certain part of the avian brain, which was one of my main goals with this, or we couldn't find the layer 5 projection neurons. So therefore, we were a bit disappointed. The only similarity was with these yellow regions. The ROR beta network was present, but the rest we didn't find. Since then, things moved on, and now people do this with single-cell level, especially the Allen Institute, Bozilka Tasik and Hongkui Zhang, and they also combine this with retrograde labeling. And for instance, here, you can pull out different labels for subplate neurons which project to the frontal cortex or to the thalamus. And you can now have a much better job. And with the sequencing, which was done on the reptilian brain by Maria Toscas in Frankfurt in the Max Planck Institute, you can actually now compare the different single cells. And this was published recently. I just summarize you what they found, and this was very surprising. The gene networks, which I talked to you about, the SOX5, CTIP2, SETB2, and others, which cross-suppress each other, these networks were expressed in the same cells. So they did not differentiate into these subclasses of projection neurons. And the success story of this paper was that they actually identified all the GABAergic interneuron markers. So basically, they were very similar in the reptilian and also in the mammalian brains. So the GABAergic actually interneurons that were present, almost all types. And they didn't see this diversification of the projection neurons. So how did the progenitors change in these brains? Because you have to produce a bigger brain, so you don't produce just the dorsal cortex. You really have to produce more cells here. So how did the progenitors change in the mammalian brain? If you stain some of these developing reptilian and mammalian brains, the first thing you notice that in this turtle, you don't have subventricular zone. Most of the divisions occur sporadically here in the ventricular zone. in chick, in the dorsal cortex, you only have ventricular zone divisions. In the wolves, you have a bit of a subventricular zone as well. I also looked at marsupials, and Leah was talking about Monodelphis domestica. So this animal is born very early, and but even if we compare equivalent ages, like an embryonic 15-days-old rat would look like this, you still don't have subventricular zone division. So the intermediate progenitors appear much later in the marsupial brain, and in a wallaby also there is a delay. And if you look at the cell numbers in a marsupial, like a Monodelphis domestica, the neuron number in an arbitrary unit column is half of a mouse. The glia cells are roughly the same. So they are not in a hurry to produce these neurons, so they get away without intermediate progenitors. So it was John Rubinstein who, was TBR2 also named after your son? Yeah. So Buffone identified TBR2, and this gene is expressed in the intermediate progenitors. And if you knock out this gene, like we did in the dorsal cortex, in a conditional knockout, then you have less intermediate progenitors. And at the time when we did this experiment, the belief was that these progenitors selectively contribute to the upper layers. And indeed, the divisions and the gene expression changed in the subventricular zone. Now, the cell numbers were affected in all layers after this knockout, so we just didn't believe that the intermediate progenitors only contribute to the upper layers. And it took several years when we had the tools, finally, the TBR2 Cree, when we could check the lineage. And even if you just cross this line with a reporter gene expressing line, you can then count the cells which express the reporter gene in proportion to all the cells. And we noticed that a large proportion of the cortex was generated through these TBR2 intermediate progenitors, including subplate, which is the earliest generated cell. And that could explain why we found the reduction at the time in all these cell layers. So TBR2 intermediate progenitors, they contribute to all layers. We also developed a new technique called clone, where we electroporate flock-stop fluorophores together with transposase, and then you have a unique random sequence expressed in the progenitor, and the same label is expressed in all the derived cells. And we looked at the size of the TBR2 clones, and depending on the age, you could actually end up with quite a large clone coming from the TBR2 progenitors. But once these clones leave the TBR2 cells, they are actually quite radially organized. They don't mix after that, so they don't disperse. So we concluded that these intermediate progenitors, they contribute to all cortical layers in mammals, and not just to the upper layers. So can then the radial glia have some subtypes which selectively producing upper layers? And this notion was suggested by Uli Müller's group in this paper, where they noticed that some progenitors, they hang around a bit longer before they start generating upper layers. Whereas most of the literature is suggesting this model, that the same radial glia progenitor will produce all cortical layers. So Fernando Garcia Moreno, who now he has his own laboratory in Bilbao, started looking at this. In fact, it was an accident. He was doing something else, but he noticed that when he used the 5' and the 3' EMX2 enhancer in his electroporation work, then usually you just label the germinal zone. So you label the progenitors, but they did not start neurogenesis. And if you just put in a KEG-CRE promoter, they already started, which is not selecting between progenitors, it's infecting all the progenitors, then you already have neurogenesis. So he took this further, and he discovered that you have these EMX enhancer selected progenitors, which actually produce the callosally projecting cells, and these cells are not present in the avian brain. If you use the same construct, you don't get this dissociation. And he did it in all these brain regions, in the hypopallium, mesopallium, nidopallium. You have no delayed neurogenesis. So it's a mammalian-specific feature that you generate this. But of course, a lot more has to be researched in this area. And how about the neuronal lineage and cell migration? So what Harvey Carton proposed was that in the ancestor, probably some cells were generated here in mammals, and they somehow migrated into the cerebral cortex. And he was speculating about this kind of migration, tangential migrations, at the time when there was very little evidence for this. So you can imagine how excited Harvey was when John Rubinstein, Juan de Carlos, Tamamaki, and others started looking at these tangential migrations, because they were coming from a region which he postulated that these cells were coming from. But unfortunately, all of these cells are GABAergic interneurons, and they are not exactly from the region where he postulated the cells. And moreover, this tangential migration was present in reptiles. So I looked at this in turtle. In those days, we didn't use this piggyback electroporation. We used just a cell tracker, which we put on organotypic turtle slice cultures, and we established the hotspots where these interneurons were coming from. And basically, it was the same DLX expressing zone as in mammals. Moreover, we also did some chimeric transplant experiments, which is really wild. We put turtle ganglionic eminence into mouse, and mouse ganglionic eminence into turtle. And we looked at the released GABAergic cells, and they knew exactly where to migrate. So they were communicating. So it's a highly conserved feature of the reptilian brain. And now Fernando used these more fancy electroporation techniques with piggyback transposon. And then you can label not only the progenitors, but all the derived cells in the avian brain. And basically, Fernando also confirmed this observation with this technique, so that the GABAergic interneurons, they come from this zone. So there is no difference in here. The GABAergic cells are born here, and they migrate to the dorsal cortex. So how about the rest of the bowl of cells? It was Alexandra Pierani who discovered that the paleo-subpaleo boundary has a zone which is expressing the gene DBX1. And this region, according to Alessandra, is producing cells which migrate dorsally to the cortex. So she based this observation on reporter gene-expressing lines, mouse lines. And there are some other cells in the mammalian brain, the Caja-Rechu cells, and also the subplate. Some of the subplate cells, they come from the rostro-medial telencephalic wall, and these are glutamatergic cells with tangential migration. So with Fernando, we decided to have a look at these cells. So just to remind you, the subplate cells come from the rostro-medial telencephalic wall, and they migrate in tangentially to the subplate. And they express subplate markers. Now, is it present in the bird? If you electroporate the rostro-medial telencephalic wall, no cells leave the wall. If you electroporate the ham, no cells leave the ham. You will have cells, progenitor cells. They also divide, but the cells which they generate, they stay put. So then we wanted to see this ventral pallium region in the cheek, and Fernando kept electroporating small regions here, but no cells were leaving to the dorsal direction. They were only heading straight to the nidopallium. And they had, some of them, they had nice radial arrangement. So then I told Fernando, okay, before we publish, let's do the control experiment with the same technique in mammal. So electroporate into the ventral pallium, and it turns out that nothing leaves the ventral pallium. Basically, the DBX1 is expressed in the cortical progenitors, so they generated the DBX1 cells. So one of the best candidates for this migration is actually had to be eliminated, and we just published this. So the bowl of cells is coming from the ventral pallium, and now we are beginning to understand the lineage and the similarities with claustrum, endopiriform nucleus, and lateral amygdala. So we don't have this stream, and in birds we don't have any of these glutamatergic migrations. They are just not present. So in fact the model which we proposed with Ann Butler without knowing any of this is correct. These bowl of cells, they migrate out laterally in mammals, and they stay put here in sauropsids. This is not a new proposal. Luis Puelles had a very similar proposal before us. The only difference was what we also added these cortical cells from the ventral pallium. And Luis did not have this in his original model. The last 5 years, we both changed our mind. I eliminated these cortical cells, and now he added them to his model. I tell you why I added them originally. I looked at the expression of latexin, and because of these cells, I suggested that they might be also from the same zone. And the reason why Luis incorporated them, because he looked at some of the subplate marker which we identified, NUR1, and you can see the expression in a normal, in a P35 knockout and reeler, so it's marking these cells. And if you look at the NUR1 expression, then you have some lateral cortical expression, and that's why Luis is suggesting some lateral cortex and subplate might also come from that ventral pallia regions, which I don't particularly believe because of the lineage tracing experiments, but the gene is expressed. So it seems like it's a fascinating problem how to move these cells out in mammals. And I just want to show you one interesting finding we had a couple of years ago, that you can have a mutant where this is actually going wrong. And this is the PAX6 knockout. With Anastasia Stoikova, we looked at the PAX6 slug Z mouse. And in a knockout, if you follow the development of the paleo-subpaleo boundary, you will have a protrusion of cells in the lateral ventricle, just like you have in the reptiles. So just deleting one gene, you can actually change the course of development. And this, instead of migrating out, these cells stay here and protrude into the lateral ventricle. Now, there is some other evidence for this. If you look at the platypus brain during development and the adult, they don't have claustrum. The claustrum is not segregating from this rest of this field. And there is also a human pathological study with band heterotopia. You can see that here you have a bigger claustrum, and also you have this band heterotopia continuous with one another. So basically, during evolution, this region could have segregated more or less, depending on the migration of these cells. So I showed you some evidence that based on the gene expression patterns of regions, or even in single cells, it's highly unlikely that all the cell types are present in the sauropsid brain than in mammals. Probably the development also changed. Nevertheless, it's surprising that there is some overlap, and they use the same molecular logic for certain steps. And this is fascinating. But the produced cells and the lineage and the cell migration studies suggest that the dorsoventricular ridge has more to do with the endopiriform cortex, claustrum, and the lateral amygdala than with the dorsal cortex. But this is not taking anything away from the ideas that the two systems operate with similar principles. And in fact, Harvey had an amazing insight so early on about these systems. So it will be very interesting to see why is it that these exactly the same gene networks are used in these brains, and why is it that, for instance, a thalamic recipient cell is expressing ROR beta. So most of the work was done by Fernando, who has his own laboratory in Bilbao. Navneet is in Copenhagen after a postdoc in Edinburgh. And I'm also very grateful to all other members of the lab and to you for your attention. And I think this NIH Oxford joint student program is fantastic. So it would be good to get more students who are interested in this kind of work. So thanks for your attention.
So I think because some of our results are different than what you got. Like we were able to get archopallium to show, find a molecular profile similar to primary motor cortex layer 5 neurons, for example, and I think maybe some of the differences between studies could be due to how do you dissect the tissue before doing either microarray or transcriptome profiling and so forth. And I'm wondering, let's say if you actually dissected a piece of tissue that was actually mesopallium and nidopallium together, right? What would be the prediction in those findings? Or is there a way that you make sure you and other people have done analysis that can dissociate that out? Because we don't always really know the exact organization of the brain in which we're actually, within that species before even comparing it to other species.
So, Eric, I was worried about exactly the opposite when we did the experiment. So rather than going in and dissecting the entire region, we just sampled inside to avoid exactly what you suggested. But then you can have concerns that maybe you were too conservative and you don't have some parts of the nucleus. So we were worried about all this. But then how do you explain the single cell results? Because the single cells surely they should have showed some of these. And maybe, okay, it's just one species so far, one reptilian. Maybe if you do it in a songbird and you do a single cell level, maybe it will cluster. So
yeah, we've been seeing some single cell stuff cluster now as well. And I think it depends how you cluster the data, the gene expression data in the single cells and whether the cells are clumped together or not.
So I was really expecting that some of the earliest generated cells in the mammalian brain, these are the subplate, the preplate. So I really wanted to know whether these cells are present in the dorsal cortex of the reptilian brain. Because, if it's conserved, then it would be nice. And I was very disappointed to see that some of the genes which determine subplate, they were actually co-localized with other layer 5 markers in the same reptilian projections neuron. So maybe they just didn't evolve and they don't have this kind of cell types, or they lost it, or yeah.
Can I ask you to summarize briefly again what you think the relationship is with the colostrum? You spent a lot of time on it, and I'm sorry, but is it too simple to say that the dorsal ventricular ridge is sort of an unmigrated claustral component that kind of builds up into the nuclear structure into the ventricle? Is that approximately correct?
It's I think what Luis Puelles, the original, not the revised version, I really like his idea that as a field, unsegregated field, you have the claustrum, endopiriform nucleus, and lateral amygdala. And those cells did not migrate out ventrolaterally in the sauropsid, they stayed behind. And in mammals, they go through that zone, and that is causing quite a bit of a developmental problem, for instance, for thalamocortical projections. So that is was my main research interest, looking at how thalamic fibers negotiate through that zone. And if they are not in the right place in the right time, and they don't have these guidance cues, corridors, corticofugal projections, they don't make it through that zone. So it became very vulnerable, that ventral pollial region where these cells migrate out ventrolaterally. So yes, so that's, in one sentence, yes. So there is a field homology with those structures.
So one follow-up. So then if you have a similarity between the entopallium and layer 4, which is unexpected, is that then conferred by thalamic afferents as opposed to a protomap or something like that?
Very, very good question. So we are looking at that in a CSR3 conditional knockout, which was developed by André Goffinet So this mouse, they don't have input or output from the cortex. They have a servo isolé. And Mriganka mentioned that some of these animals, they run around, swim, walk without cerebral cortex. They do just that. Without cerebral cortex. It's better not to have a cerebral cortex than a developmentally altered cerebral. If you change the interneuron population slightly, you might end up with a worse phenotype than not to have a cerebral cortex. So we are looking at the cerebral cortex of these animals with the servo isolé, and we want to see whether this ROR beta network is expressed there or not. Because if it is induced by the thalamic input, then that could explain why these cell groups with very different lineage, very different developmental origin, they express the same gene network. And then it would be very interesting to see the primate. And then why do you have these subcompartmentalizations of in the primate? You have like 10 layer 4, no, in primate or more. Yeah.
I haven't given a thought to this in a very, very long time, but now that I heard your talk, and I go back to very early work that I did with Fernando, I don't know if you remember, we described the ventral and the dorsal hotspots for neurogenesis in the adult brain. Whether these correspond to any of these migrations early in the embryo, it would suggest that the ventral hotspot might correspond to interneurons, and the dorsal hotspot might correspond to this insular migration. But those cells do migrate, and they migrate very far away, even in the adult brain.
Did you look at these hotspots in the embryonic?
No. I don't think anyone has done.
Because all this, what I was talking about, is the embryonic. So, but it is interesting that in some songbirds, George Streeter is suggesting that you do have compartmentalization of the germinal zone, because they pump out more neurons. So you have a thicker layer of intermediate progenitors. So I showed some of our data in chick, where in the dorsal part of the telencephalon you don't have this compartmentalization. But what George is saying, that in some songbirds during embryonic development, where they have to produce a bigger DVR, they do have. So it would be very interesting to study more species because there might be some differences.
But the dorsal hotspot is not in the pallium side. It's actually what would be ventral pallium, right after the fold. I don't know. I don't think anyone has looked at markers that might be.
But so it's above the DLX territory.
Yes. Yeah.
You showed one of the mutants that the cells fail to migrate into this colostrum insular area. So what is the functional implications in that mutant?
Yeah, they die at birth, so we don't we don't know. But I was so surprised to see this bowl of cells protruding into the lateral ventricle. I thought that. My lab manager was cutting a turtle brain when I just looked at the slides, and it was the PAC6 conditional knockout. So, but it was there was a striking resemblance with the reptilian brain.
Okay, so if no more questions, let's thank Zoltan again for the wonderful talk. Welcome back. We are starting the second part of the afternoon session. There will be 2 talks and data bleeds, and afterwards, the speakers and the speakers, including the data bleed speakers today, please come to the front at the end to sit in the chairs for a panel discussion. Now, let's welcome the first speaker, Takao Hensch from Harvard University.
Thanks very much, Kwan and David, for having me here in this very special symposium. As many of you are, and I, are painfully aware, brain plasticity changes dramatically with age. And it was already apparent for a long time. Here's Aristotle's thoughts that early life is particularly potent in shaping who we are and our behaviors. And fast forward to the modern day, philosophers like Calvin then put the onus on us, parents and caregivers, to provide the right environment. And so this is, of course, a critical time in life when a variety of opportunities present themselves, and careful observation of human behavior already 100 years ago by Maria Montessori started to identify these bulb-like swellings of malleability around birth and in the first years, actually before school age, and then perhaps a second opportunity during adolescence. And so we're well aware of cases of extreme performance emerging from experience during this time. But I think we're all here because of the dark side of these critical periods, which are equally windows of vulnerability. In fact, most, if not all, mental illnesses seem to have developmental origins, a theme in this conference, that need to be understood. And that's really why we call them critical periods. In the absence of certain kinds of stimulation or certain kinds of input or genes, our brains become malleable in a derailed sense, perhaps. And this is something that we would like to understand at a fundamental biological level. It's the classic gene by environment interaction question. And I call them critical periods, and I won't debate whether they're sensitive or critical, just to put this in the right context. You've heard a lot about gene programs and migratory processes which remarkably set up the nervous system. And these critical periods are opportunities for each and every one of us to reshape those circuits, to customize our brains, if you will, to the environment into which we are born. And so in the laboratory, we're interested in understanding when are these critical periods. And obviously there's not just one big critical period, but there are multiple such windows in a seeming sequential hierarchy. So from primary sensory areas to sensory motor integration to higher cognition, we see waves of elevated brain plasticity that allow our functions to develop. And so we can pose more specific questions. What turns on these windows? What determines their duration? And what turns them off? And by way of introduction, of course, the field turns to the visual system, which has the long history of study of trying to identify cellular mechanisms underlying critical periods, because from mouse to human, it's a relatively straightforward system in that its function is well studied, vision to see. It has two inputs, left eye and right eye, which arrive through the eyes, through one relay station in the thalamus to the back of our brain. And we know where to look for changes that are quite dramatic. So the work of Hubel and Wiesel and many others has shown that the arrival of thalamic axons serving the left or right eye in forward-viewing animals such as ourselves is typically well-balanced, as shown in these ocular dominance column maps of flattened visual cortex. But the simple act of a lazy eye or patching one eye, creating discordant input without frank damage to the retina, causes a massive physical rewiring of the system early in life, such that the open or more salient eye largely takes over visual cortex at the expense of the other eye input, with significant consequences for depth perception, visual acuity, and so on. And this condition of blunted vision or amblyopia becomes permanent and to this day has no treatment after the age of 8 or 9 in humans. And so, in fact, it's very important that any discordant visual input is corrected as early as possible to allow these critical periods to reconnect the eye that was losing its connectivity to the visual cortex. And so it's a bona fide neurodevelopmental disorder with impact on 3 to 5% of the human population, leading to untreated amblyopia having lifelong consequences for vision. But it's also an opportunity for animal experiments to test what are the factors that determine and restrict this plasticity largely to early life and make it harder to recover as we get older. So to make a very long story short, because I know the main purpose of my being here is simply to tee up Gervasio's 5 minutes at the end, we and many others have dug into the local circuitry of the visual cortex and come across striking findings that the plastic windows seem to be determined by the maturation of inhibitory cells, and perhaps even one particular subset of inhibitory neuron, these parvalbumin-positive large basket cells, so-called because they make these pericellular nests, as drawn here by Cajal, again, over a century ago. And these cells can be enwrapped as they themselves mature in a tight extracellular coating called the perineuronal net, also drawn here 100 years ago by Golgi. And so we've known these players for some time. We just didn't know how pivotal they were for determining critical period milestones. So now we can apply more modern techniques such as Brainbow, developed by Josh Sains and Jeff Flickman, my neighbors at Harvard, to label individual parvalbumin basket cell axons in different colors and allow us to map more precisely the maturational time course in different brain regions and in different mutations that might underlie neurodevelopmental disorders, and quantify the convergence and plasticity of these pivotal connections that seem to trigger or initiate these windows. And using super-resolution imaging techniques like storm imaging, you can map with nanometer resolution these perineuronal nets shown here in purple, as we've done in collaboration with Xiaowei Zhuang's lab, also at Harvard. And I'll talk more about them later. And so we have cellular bookends that we can use to look at the timing of critical period onset and offset, and I'll illustrate that in my talk today. Basically, just one slide of review. If you take a mouse and you study its visual acuity, there is a window of time when patching of one eye, the classic Hubel and Wiesel experiment, leads to a loss of visual acuity, and we call this the critical period. Mice, as you know, are born with their eyes closed, and so around postnatal day 12 or 13, they open, and patching of one eye actually has no effect at that early age. But around 22 days of age, for a few weeks, there's a peak of plasticity, and then the plasticity winds down. Interestingly, if you do something to delay the development of what turns out to be inhibitory circuits, such as raising animals in total darkness, this critical period can be divorced from the chronological age of the animal. So you can raise an animal in a dark room, and they will not enter this critical period until they're brought out into the light, even as a 3 or 4 month old animal. Conversely, you can treat a wild-type mouse with a GABA-strengthening drug, such as benzodiazepines, just after eye opening, and initiate a premature loss of visual acuity and plasticity before its time. And these results really were striking because it showed, A, that GABA was surprisingly driving this timing, and B, the critical period itself seems to be plastic. And in fact, this result has been replicated many times over, and I don't mean to intimidate you with this slide. I just want to tell you that we and others have now mouse models where you can take them out of the animal room and know that their critical periods are either shifted early or shifted later. We can play with the spine maturation on excitatory neurons and extend or narrow the duration of these windows, or create mice where critical periods fail to begin because the GABA system fails to mature or never closes, for example. And so since there's obviously not enough time to talk about all of these things, I just wanted to take my time here to tell you 3 short stories that relay some concepts that we are very much interested in and hope will relate to other talks in this meeting. So, for example, if critical period timing is so important, you might wonder if there's a clock or a timing mechanism in the brain that determines when these critical periods should happen. And so we can study this using molecular tools that are familiar to you. Clocks in the brain are very elegantly studied in the suprachiasmatic nucleus, and were rewarded with a Nobel Prize last year, the identification of circadian genes. And in fact, this is a very elegant system of a transcriptional feedback loop where BMAL and clock dimers drive downstream circadian genes, and those then dampen the BMAL clock action, and you create a circadian oscillation of gene expression well documented, even in an isolated piece of SCN in a dish. So Yohei Kobayashi in the lab wanted to know if circadian gene oscillations, which were increasingly appreciated to happen throughout the body and brain, would happen also in the visual cortex. And he dissected from adult mice the visual cortex at different times of day, and sure enough found that these circadian genes oscillate in the visual cortex. When he went younger, though, it was interesting to see that there was no such rhythmicity, for example, just before eye opening. And in an uncanny parallel with the onset of the critical period, this circadian oscillation of gene expression in the cortex emerged. And so this correlation was too good to pass up, and we started to study clock knockout mice. If you delete clock, the circadian gene oscillation is severely dampened, even though physiological circadian rhythms are intact. And so this is a way to illustrate how these experiments are done. We record visually evoked potentials from the primary visual cortex of mice viewing sine wave gratings. And at some point, when the sine wave grating becomes too fine, the visual evoked potential, or VEP, goes flat, defining the admittedly very poor visual acuity of the mouse. If you do monocular deprivation, or MD, for 4 days during the peak of this critical period in wild-type animals, there's a significant loss of visual acuity, which lasts beyond this time if you don't reopen the eye in time. But interestingly, in the clock knockout mice, there was no such plastic change. Instead, if you waited longer to a time when the wild-type mice no longer showed this loss of visual acuity in response to deprivation, the clock knockout mice now became plastic. And so based on what I've already told you, this is a situation that was quite familiar to us. It looks like a delayed onset of the visual critical period. And one way in which this could happen is if inhibition were to be slowed down in its maturation. So could we rescue these mice? The answer was yes. If we treat these animals with a GABA boosting drug like diazepam or Valium, then monocular deprivation at the expected age becomes effective. And conversely, if you treat the animal repeatedly with a GABA enhancer during this time window, then the delayed critical period plasticity is now lost. And so these results were very consistent with the other findings that I'd mentioned. And satisfyingly, if you look at where clock is expressed in the cortex, there's the highest colocalization with these parvalbumin-positive inhibitory neurons. And if you label the basket boutons that these cells make, they are clearly impoverished at the regular critical period age, and there is a reduced number of miniature inhibitory postsynaptic currents. And so instead of using the total clock knockout, Yohei made a conditional clock knockout where clock is removed only from parvalbumin-positive cells, and was able to completely phenocopy the total knockout situation, namely that the critical period plasticity was delayed. For good measure, he also made a pyramidal cell-specific knockout, the vast majority of neurons in the cortex being excitatory pyramidal cells, but disrupting clock from those cells had no effect on critical period plasticity. So these examples illustrate how we go about doing these studies and asking these questions, and led to the conclusion that clock gene expression in these pivotal inhibitory cells may actually, in a cell-autonomous way, contribute to the timing of these windows. And I went through that just to start thinking more broadly, because it has a number of immediate implications. So for one thing, if inhibitory circuit maturation turns on critical periods, then you might think it might be possible to reintroduce a second critical period by putting immature inhibitory neurons into the postnatal brain. And Arturo Alvarez-Buya is here. This is something he's been doing at UCSF with Mike Stryker, and found that, in fact, if you scoop out the source of embryonic inhibitory neurons of this type and transplant them into the postnatal cortex, you do, in fact, create a second critical period. But what was interesting from their results is that it doesn't happen right away. It takes a certain amount of time before the second critical period happens. In fact, it takes about a month, which is consistent with the maturational profile of these cells in the intact brain. So it could very well be that these cells have some kind of timing mechanism that contributes to plasticity onset. A second implication of relevance here is that circadian gene polymorphisms have been linked to a number of mental illnesses, and often these are interpreted as circadian consequences on cognition. Certainly anyone who's had jet lag will appreciate that. But our results suggest another possibility, that perhaps this kind of circadian mistiming has, over development, mistimed these windows in people who carry these kinds of polymorphisms. And so there could be an accumulating issue that affects timing across brain regions which should be experiencing staggered brain plasticity. And this is something that I won't have time to get into too much today, but we think might play an important role in neurodevelopmental disorders like autism spectrum disorders, which are often linked to excitatory-inhibitory imbalance. You could imagine that such an imbalance would create slight jitters in primary sensory critical periods, which then ripple through the system and then manifest as complex cognitive disorders as an accumulation of derailed timing. And one way to treat such a disorder might be to intervene as early as possible and to redirect or restore the proper timing of these windows before it escalates into these higher order areas. Of course, parvalbumin-positive basket cells don't exist only in the visual cortex, and so, as I said, we can use them as a biomarker to predict other windows of plasticity. And even within the same mouse brain, it's well known that these cells mature at earlier times, say in the auditory cortex than in the visual cortex. And so it makes a prediction that critical periods in the auditory domain should be at least 10 days earlier than in the visual domain. And this is something we can test. So another wonderful postdoc in the lab, Anne Takesian, had worked on the auditory system before coming to the lab. And just to remind you, this now is a somewhat more complex route from the periphery to the auditory cortex through multiple relay stations, but represents one dimension of sound frequency, shown here in this color map called tonotopy. This linear dimension of sound frequency is reiterated at all these relay stations and allows us to do an experiment of altered sensory experience like patching an eye. In this case, if you were to map the sound representation in auditory cortex using single unit electrophysiology, for example, a normally raised animal would show a nice rainbow spectrum of responses to low to high frequency sound input. But raising animals listening to particular sound frequencies at strategic times in development can distort this map. And so here's an example of a mouse that was raised under a 7 kilohertz repeated tone presentation. And lo and behold, this is effective only when the tone rearing happens at postnatal day 12 to 15, 10 days earlier, than the visual cortex becomes plastic. And so using this system, we can start to dissect the contribution of inhibitory interneurons because, in fact, there's a very nice opportunity to cut brain slices that are semi-horizontal that preserve the thalamocortical connection in the auditory system. And use this preparation soaked in a voltage-sensitive dye and using a high-speed camera could map topographic changes in the thalamocortical projection as a response to these sound experiences. And as a single scalar index of topography, we map the linear progression of response in the slice to stimulating different spots in the auditory thalamus. A typical normally raised animal will have a slope of 1 on this graph, indicating a nice normal topography. If you tone rear the animal, this topography breaks down and the slope goes down. And so she started to play with inhibitory neurons and their activity during this tone rearing. And in fact, she found that if you use, in this case, a dread approach to silence a subclass of inhibitory interneuron, then this tone rearing became ineffective. And despite being in this tone environment, these mice did not change their map. Interestingly, the subtype of neuron that she studied here was a group that expresses the ionotropic 5-HT3 receptor. And these mice, these cells, sorry, are predominantly upper layer cells, shown here in white, which extend remarkably narrow columnar palisades of axons down into the deeper layers of cortex. And using rainbow imaging, we found that these cells that are primarily targeted by these upper layer interneurons are parvalbumin positive basket type neurons in layer 4. So this was quite nice because it suggested that the plot thickens, but ultimately the inhibitory neurons that she found to be pivotal are inhibiting these cells that we had previously identified as critical period triggers. And in fact, she could fairly routinely find connections using paired cell recordings in these slices from these upper layer 1 cells of a non-late spiking type, making inhibitory connections onto these parvalbumin cells. What does this do to the system? It's a way to gate inhibition in the thalamo-recipient layer 4. And so in this example, she used an optogenetic approach to stimulate these layer 1 interneurons labeled with channel rhodopsin at different times with respect to thalamic stimulation. The thalamic input to these layer 4 interneurons is normally quite strong, often leading to action potentials. But if you concurrently stimulate the layer 1 interneurons, you effectively shut down the activation of layer 4 interneurons for a rather long window, as you can see in blue, at least 100 to 150 milliseconds, creating a narrow window of opportunity for thalamic input, which is much weaker, onto neighboring excitatory cells to be somewhat more effective. And across a number of cells, you can see that there's a window of disinhibition created when layer 1 cells are activated. And so we think that this might be one way in which perhaps spike timing dependent plasticity rules could be engaged for plastic changes to occur. She also noted, interestingly, that these layer 1 cells were receiving thalamic input directly. And that could be seen here in this labeling from the thalamus, very rich plexus of axons from the thalamus. And if you stimulate in the thalamus and record from layer 1, you get EPSPs the size of the ones you would find in pyramidal cells in layer 4. And so this led to the possibility that thalamocortical topography might also be seen in layer 1. And in fact, she went back and found that in all of her voltage-sensitive dye images of layer 4, when she analyzed layer 1, there was a beautiful topographic map from the thalamus living in layer 1. Curiously, when you tone rear the animals, so the layer 1 map has a slope no different from the layer 4 map normally, but if you tone rear animals, the layer 4 map shows this plastic change, but layer 1 does not. And so it's a new wrinkle on this topography that it seems the thalamocortical map in layer 1 is a kind of scaffolding that is immune to tone rearing, at least at this time, and might be coordinating plasticity below in layer 4. Why is this interesting for us? Well, of course, all of us experience something like this at birth. We're born into this world with a brain that developmental linguists tell us is capable of hearing speech sounds of any language, and then passes through a number of sequential critical periods to shape the language system in a kind of bootstrapping fashion through auditory experience. And so it could be that these kinds of very early discriminations of native versus non-native speech sounds is very similar in some sense to the tone rearing that I mentioned. And so if that's the case, would critical periods for early language discrimination also move like mouse critical periods do when you alter excitatory inhibitory balance? In collaboration with Janet Worker's lab at the University of British Columbia, who pioneered the findings of many of the earliest critical period windows in humans, we found this could be true, that in fact infants exposed to SRI antidepressants showed an earlier closure, a shift to the left of these very first phonian discrimination critical periods. These kids are now about 10 years of age, and so we're very curious how they are doing on the full-blown language acquisition. But already 2 years ago, a much larger powered study was done in Scandinavia. Jay Gingrich from Columbia, working in Finland, had access to the records of 850,000 people and identified on the order of 10 to 15,000 subjects who might have been exposed to SRIs around birth, and found that, in fact, at school age, they had a 37% increased risk of speech and or language disorders. There's a large gap between what happens in that first year of life and these tests. So this actually could be an underestimate, because if you detect a change in language acquisition, chances are a number of interventions would have been put in place. But this is just to say that we believe strongly the mechanisms we identify in mice are fundamental and could inform us to consequences of these early exposures. So I'd like to turn to the closure of the critical periods. The notion that these windows disappear has suggested that we might lose something as we get older, but it's equally possible that we gain something which acts as a brake to dampen excessive plasticity. And the reason we looked at these layer 1 cells was, in fact, motivated by a brake that we had identified a few years ago acting on the cholinergic system. So in moments of heightened attention or arousal, acetylcholine is secreted, which is very important for effective plasticity. And it turns out that as our brains mature, we create a protein called LINX1, which has a structure very similar to bungarotoxin, a kind of proteinaceous toxin that, in fact, dampens nicotinic acetylcholine receptor function. And so if you delete LINX1, what happens? In fact, Yuichi Makino has developed tests of attention in the lab where animals have to wait for long periods of time, or making the stimulus ever shorter to challenge the animal's attention, and found that compared to wild-type mice who miss these shorter stimuli because they're typically not on task, the LINX1 knockout mice are hyper-attentive and aroused. So even with the shortest stimuli, they get these tests right. And from trial after trial, they're always on task. Some of the best students in my lab. So in these heightened attention animals, we find that indeed the critical period plasticity stays open longer. At a single cell level, these upper layer interneurons, the 5-HT3 positive cells, have links and their requisite nicotinic acetylcholine receptor. And if you fact sort these cells, you find that in this case in the auditory system, LINX1 expression goes up significantly as these windows close. And if you record from these layer 1 cells and apply nicotine, they are robustly activated in the young age, but famously desensitize as the animals grow into adulthood. This desensitization is lost if LINX1 is deleted. And under this condition, tone rearing still is effective. From a therapeutic point of view, these animals are also able to recover from early life deprivation. Here in the visual system, Hirofumi Morishita showed that if you do monocular deprivation spanning the critical period, there's a beautiful loss of acuity, as in wild-type mice. If you reopen the eye too late, there is no recovery of acuity in wild-type mice, but the LINX1 knockout mice show a full recovery of vision, suggesting that this could be a druggable target for reopening a critical period. And so that's one interest, of course, but we already know how it works. It dampens cholinergic transmission. So another way might be to boost cholinergic tone with a drug that's commonly available, cholinesterase inhibitors. And even wild-type mice then show a recovery of visual acuity, which is quite exciting because, as I said, there is no cure for amblyopia in adulthood currently. And at Boston Children's Hospital, we actually have a pilot trial going to see if you can use this strategy of reversing critical period plasticity to restore function that might have been lost earlier in life. This is a drug approach, but maybe someday a non-invasive approach might be possible. It turns out that when animals exercise, and in this case it's driven by optogenetic stimulation of the midbrain locomotory centers in Mike Stryker's lab, copious amounts of acetylcholine are released in the cortex. And so if you pair this running exercise with visual stimuli, then he finds adult animals will show selective enhancement of response to that stimulus which they saw while running, but not to other not observed stimuli. And so this is a huge sigh of relief because reopening the critical period might not be such a great idea. This suggests that it does still take paired training to get a very specific kind of enhancement under these conditions. So to summarize, what we've been trying to do is a local circuit dissection of critical period plasticity using sensory systems, and increasingly as we move to the front of the brain, we think similar ideas are repeating, at least in the cortex. Long-range projections from the thalamus arrive onto an EI microcircuit, the stronger input of which goes to these parvalbumin-positive basket cells. And then there is no doubt that plasticity in the cortex involves pruning and regrowth of spines on these excitatory, far more numerous pyramidal neurons, including new players such as microglia, perhaps. But the timing of when all of this happens seems to be directed by the maturational state of the inhibitory cell, which acquires this perineuronal net as the critical periods close. And I told you today about layer 1 contributing to this. These cells are loaded with neuromodulatory receptors of a fast type, nicotinic, ionotropic, or 5-HT3 serotonergic receptors, live in layer 1 or send projections there, and can be strongly modulated by context or environmental enrichment as a stimulus that leads to secretion of such neuromodulators. And they may, in fact, be wired into this system to control plasticity. So if I am allowed another minute, I just wanted to end on a word of caution. Because essentially what we're doing is trying to dissect the mechanisms of this critical period and, as a proof of concept, reverse engineer it in adulthood, which is actually quite interesting from a therapeutic point of view to perhaps recover from stroke or traumatic brain injury, a moment in life when you might want to boost plasticity to juvenile levels. But of course, it's easy to go overboard and start thinking about other opportunities. And so I just wanted to suggest that this might not be such a great idea to do willy-nilly. And for that, I turn to the perineuronal net, this structure that closes and surrounds these basket cells, illustrated here by an artist's drawing, is kind of saran wrapping around the cell body and proximal neurites, perforated by synaptic boutons. And many people have been attracted to this interesting structure and have likened it to a punch card for memory, famously written up by the late Roger Chen. Nobody remembers punch cards, I suppose. But if you could read the holes in the net, you might actually know something about the experience of the animal. And that's something we're doing with the storm imaging. So in fact, with nanometer resolution, now you can map these nets. And using a trick from cartography, if you unfold the globe of the perineuronal net, you have a two-dimensional layout of the holes. We can now measure in detail the spacing, the size, who lives in these holes if you use 2 or 3 colors, and look at EI balance on these pivotal neurons themselves. But they're not just a structural barrier to plastic change. They're a chemical structure rich in sugar-bearing proteins that might attract non-cell autonomous factors. I told you about clock, but there are many other factors that determine and maintain the healthy, fast-spiking nature of these basket cells, which may be attracted to these nets. And in fact, one molecule we study, shown here, beautifully trapped inside these cells, is not made by these cells and has a sugar-binding motif in its structure, which we can use, exploit, to knock down in the adult and reopen plasticity by removing this one factor. The reason I'm telling you all this is that the nets also have a day job of protecting these cells. These neurons are extremely potent generators of reactive oxygen species. They fire at high rates and so are chock full of mitochondria. And if you have weak nets under conditions of oxidative stress, these are the first cells to show oxidative damage. And in fact, Alzheimer's was mentioned. Well before any of this was known, already in the last century, 1999, in human brain tissue, it had been reported that the chondroitin sulfate proteoglycans that are found in these nets are weakest in those higher order human associational areas that have evolved to be plastic longer. So it's an inverse relationship. We have more plasticity in these regions, perhaps because the brakes are off in these areas. And in mouse models and actually human tissue of schizophrenic subjects, we find that the perineuronal nets are impoverished and not nearly as beautiful as in neurotypical controls, suggesting that this may be some kind of hub for psychosis. And so closure of a critical period might be important as a neuroprotective strategy, and that in fact plasticity is great, but stability is probably just as important for a healthy functioning nervous system once the circuits have been sculpted early on. And so I just want to end by saying that we're very excited about this work because it allows us to bridge this gap between genes linked to mental illnesses and behaviors of mental illness at the local circuit level. And we're now launching a major initiative to link principles of circuit development with computational approaches to try and understand better how this EI balance is directing traffic. And finally, thanks to NIMH for refunding our Conti Center, which allows us to work with people like Paula and Jeff Flickman and Guoping Feng to look at how these local circuits develop in human, mouse, monkey tissues and under conditions that are linked to mental illnesses. So I'll stop here. Thank you.
Hi, thanks for a great talk. I found the clock data really kind of intriguing. So as you're thinking that it's a way that the neurons might know how old they are, essentially, by how many cycles they've experienced. And if so, what's counting the cycles?
Right. So that's a great question, and I won't have a satisfying answer yet because it's something we're very interested in. The motivation was, as I laid it out, maybe there are a certain number of ticks on the clock that allow the cell to mature fully and initiate plasticity. Well, then why is it that in the auditory cortex it's so much earlier? Is the clock running faster? Is it not counting ticks or doing something else? After all, clock has multiple functions. It could be simply a transcription factor. It's also a hat and has many other possible explanations. So our usual process of elimination is ongoing. But what we're interested in doing is speeding up or slowing down the clock in different brain regions and seeing what's the consequence of separating out the timing in that sense. My bet is this would wreak havoc, but yeah. So we have a lot to borrow from, say, fruit fly tricks to speed up or slow down. Doing something very non-stressful, removing an hour a day is too slow. It would take, you would pass the critical period before you knocked off one day or one tick on the clock.
Yeah, just the same question, actually, but a different part of it. I understand a lot of the critical period timing is experience initiated and self-terminating and some of the visual system stuff. I'm not sure if that's the case in the mouse, so I'm also really interested how to integrate some kind of clock advancement with that kind of effect. Do you have an idea on that?
Yeah, so the clock was related to the onset, but interestingly, in all of the manipulations where we moved the onset, the timing followed after a certain duration, suggesting that it's a trigger of some kind of gene program. And so, in fact, epigenetics was mentioned earlier. We have targeted epigenetic mechanisms as a way to keep the window open longer. We think that the EI balance, once it hits some optimal range, unleashes a program that ends with perineuronal nets forming and breaks coming up.
Yeah, maybe last question, then we need to move
my
on.
fault.
We can come back in the discussion session. Yeah, maybe just one last question.
No, she said I can do it. Okay, I get it. I was intrigued by the layer 1 findings, and because I really haven't seen, maybe it's in the literature, like almost looked at as like a stronger projection there as it was to layer 4 from the thalamus, and wondering if that's more specific to the auditory pathway. And the other is, I didn't get how the critical period difference or lack of it in layer 1 might be influencing layer 4.
Right. So, the second part first, I guess, it was a surprise to us. And we found that, at least at this age, the same manipulation has no rewiring of thalamocortical input. And so it could be a way to reset, keep a memory of the original proper map, so to speak. I'm thinking of Barnell experiments, for example, where maps could be shifted, and then there's something to go back to when the prisms come off. And so maybe this is some version of that. And layer 1, this was all done in auditory cortex for a number of interesting technical reasons, but also getting us into the auditory system. Now, of course, we're going back looking at visual cortex and trying to see what the relationship is to visual acuity and so on.
Okay,
Do you?
Need to move on. We can come back to ask at the end of the session. So the next speaker is Margarita from MIT.
Okay. Thank you very much, Kuan. Thank you very much, David, for this invitation. Wonderful meeting, and I've learned a lot. So I'm going to tell you today about some studies that my lab has been doing for the last few years, in which we have tried to examine fundamental mechanisms of plasticity and their relationship to genes of brain disorders, whereby these mechanisms can provide some insights. So, to remind you, from an article that John Rubinstein and I wrote a few years ago, it gets earlier and earlier, John, every year. The cortex wires itself using mechanisms that depend on developmental time. The earliest events have to do with development of diffusible factors that pattern the cortex, that lead to axon guidance and targeting, and finally the shaping of circuits via activity-dependent mechanisms. So we take it as a starting point that cortical areas and pathways form early. Synapses and circuits form progressively and consolidate later and are shaped by mechanisms of plasticity. Most psychiatric brain disorders, the theme of this meeting, don't mess up the brain royally. They mess up the brain subtly. And so we take as an important starting point that many brain disorders arise from deficits in synaptic development and plasticity. And I hope I'll convince you by the end of my talk that there are actually specific plasticity-dependent processes that might be attributed to some, at least, genes of autism. So I'll tell you about two kinds of studies. First, on a set of experiments that we have done to look at a very specific mechanism of activity-dependent plasticity of cortical synapses that shapes circuits. And we call this locally coordinated dendritic plasticity. We think it provides a window into both Hebbian as well as autoregulatory plasticity that shapes neuronal responses. And we think that ideas such as this, applied to an example of cortical plasticity, first discovered by Hubel and Wiesel, but then and you might think, what more can we discover? But in that grain of sand, we see the universe, and that's ocular dominance plasticity. And we and others have divided that phenomenon. You've heard a little bit of it from Takao, into two kinds of plasticity, and in particular, one kind, the response of a neuron to a change-inducing plastic phenomenon. Amazingly, I hope I'll convince you, has a very big role to play in how we might understand conceptually at least some subsets of autism spectrum disorder. So first, what do I mean by locally coordinated dendritic plasticity? We are motivated to ask this question by studies that my lab has done for many years. The first study published 30 years ago in which we rewired the brain. Normally, the visual pathway consists of retinal projections, the visual thalamus to the visual cortex. The auditory pathway consists of auditory projections through the auditory midbrain to the thalamus to the auditory cortex. By taking newborn ferrets, which are born at about this approximate stage of the first third of gestation in humans, and by removing the inputs that go from the inferior colliculus to the medial geniculate nucleus, we were able to make retinal projections go to the auditory thalamus, thereby making visual inputs drive the auditory cortex. It took us 20 years to figure out why this works in the first place. When you remove these inputs, you alter the molecular architecture of the auditory thalamus, which begins to express genes that are normally expressed in the visual thalamus. What does this do? It makes the auditory cortex, which is now driven by vision, to generate orientation-specific maps, responses and maps by changing the nature of its connections. While in the normal auditory cortex, there are connections along the isofrequency axis. And in the normal visual cortex, the horizontal connections link neurons with the same orientation preference. In the rewired auditory cortex, there is no addition because the auditory inputs have been removed. There is only visual driving, and the horizontal connections end up resembling the connections in the visual cortex, as also evident in the orientation map with pinwheels in the rewired auditory cortex. It's the same cortex, but it was never made to see, yet the nature of input activity induces this pattern of circuitry. How can this be? And this leads to the deepest question in our field. How is it that electrical activity, and specifically the pattern of electrical activity, alters synapses and neuronal circuits? A lot of evidence now indicates that between different cortical areas or between the inputs to them, there is not a difference in the amount or the mean amount of activity, but there are important differences in the patterning of activity, which leads us to ask, how is it that patterned electrical activity wires the cortex? And to do that, we have turned to a mechanism that is called spike timing dependent plasticity. This was discovered by both Henry Markram and by Mu-Ming Pu's lab in slices of hippocampal neurons, where they made the presynaptic neuron fire before the postsynaptic neuron, and that increased the strength of connections, shown here, pre, before, post, whereas if they made the postsynaptic neuron fire before the pre, that decreased connections. It's a particular form of Hebbian plasticity. In fact, this was Donald Hebb's idea that if a neuron takes part repeatedly in firing its partner, postsynaptic partner, increase the strength of connection. This is essentially what Hebb wrote in 1949. But then it's a symmetric rule. If you make the post fire before pre, they showed that at least in neurons in a dish, the strength of connections will decrease. And we took this idea. This has never been really proven in vivo, except for a couple of demonstrations. And two very talented postdocs in my lab, Sami El-Bustani and Jacques Ip. And Jacques is here and will follow up my talk with his own talk. They tested this, demonstrated it, and showed a mechanism for this as a way to shape receptor fields and circuits in the intact awake mouse visual cortex by examining one single neuron in great detail in which they could implement spike timing dependent plasticity. So pairing presynaptic activation with postsynaptic spikes can modify synaptic strength and hence a circuit that shapes neuronal responses. How did they do this? So here's the basic idea. A neuron in the visual cortex, or any cortex, gets inputs from many synapses, from many different neurons. Let's say there are these 4 synapses that arise from 4 different neurons. Each of these synapses has a receptive field, which is the receptive field of the presynaptic neuron. And these receptive fields are shown here in the haze. Okay? And the neuron's receptive field is the vector average of all these synaptic receptive fields, shown here in the white circle. If one puts a visual stimulus, a spot in visual space, and flashes that spot, you are driving those neurons whose receptive field overlies that X, right? And then you follow up that flash with a spike in the postsynaptic neuron. You force it to spike by inducing channel rhodopsin-driven single spikes in the postsynaptic neuron. You will have a pre-before-post pairing of only those synapses that were driven by the stimulus spot located on the X. And if you do this enough times, a few tens of times, and if the spike typing dependent rule is correct, you would increase the strength of these synapses that were driven by this X, shown here, by the flash on the specific location marked by the X, and you would change the receptive field of this neuron because these synapses have been strengthened, and the receptive field will shift to that location. Does everybody understand what I'm talking about? Okay, it's a simple idea. A neuron's receptive field is the sum of its synaptic fields, and you can potentiate some synapses by a pre-before-post pairing. And this is a schematic of how this is done. You can put a stimulus spot at many different places, but whenever the stimulus spot appears on one location, you induce a spike in the postsynaptic neuron. So this is how it's actually done. What Samy and Jacques did was that they filled a single neuron, a single one neuron, by intracellularly filling it with channel rhodopsin in order to make it spike when they wanted to, with GCAM6S in order to image the neuron's somatic receptive field as well as the receptive field of every one of its synapses, thousands of synapses in principle. In practice, they only imaged a few hundred. That's good enough for the point I'm going to make. And with a volume-filling dye called mRuby, because they wanted to study the volume or the anatomical size of a potentiated spine and correlate it with what else is happening. So here's the idea. So a neuron is being mapped, its receptive field is mapped, and then a spot is being put everywhere, and whenever the spot appears in one location, it is followed very quickly, within a few tens of milliseconds, by a blue flash of light that makes this neuron fire. Okay? So that's the pre-before-post. Pre being excited by a spot at a given location, followed within a few tens of milliseconds by a spike in the postsynaptic neuron, thereby potentiating that location. And if they do this for over just a few minutes, with up to 160 actually pairings, you can show that the neuron's somatic receptive field has now shifted from there to there, right on top of the stimulated spot. And that's because different synapses that are driven by that spot have been potentiated. And how do we know that? We know that by actually imaging hundreds of spines, which are the postsynaptic elements of a synapse, plotting their receptive field and showing that when there is a receptive field on the target, then shown in red always, that spine increases in size. When there's a receptive field off target, that spine decreases in size. And when you look at all the receptive fields that have increased in size, they all lie on the stimulated spot. And if you look at all the receptive fields of the spines that have decreased in size, they lie away, so that the difference explains to us how is it that the soma receptive field has now shifted from somewhere else to here. And where do the receptive fields of these fattened spikes or LTP'd spines lie? They lie right on top of the target, and the LTD, or the depressed or the shrunken spines, are away from the target, thereby underlying this receptive field shift of the cell. So it's a very reduced, very precise way to monitor, to manipulate, and hence to measure plasticity over stretches of dendrites. And what is that plasticity? So it turns out that potentiated or fattened or red or structural LTP spines and depressed or structural LTD spines are not randomly distributed. They lie in close proximity. Wherever you see a red arrow showing a spine that has grown, you see green arrows around it showing spines that have shrunk. And we can do this very precisely. We can measure it over hundreds and hundreds of spines, over many stretches of dendrites. And the bottom line is that surrounding potentiated spines, there are depressed spines within 50 microns. And so that whenever you have a fattened spine or a potentiated spine, very soon around it, there are spines that have shrunken in size. Now, when we published this paper, a reviewer, bless their heart, wanted to know, is this real? And can you not do serial electron microscopy in addition to the awake animal 2-photon imaging pre, during, and post in order to prove to us that what you have shown as potentiated or depressed spines are truly structurally enlarged or depressed? It's actually a valid question because we are working at the limits of what is possible by light. When you have a spine that is a micron or less in size and it's shrunk by, let's say, 25%, it's actually below the Abbey limit for optical imaging. So I remind you, the Abbey limit, the finest resolvable image, is limited by lambda, the wavelength of light, divided by the numerical aperture of your objective. And so the wavelength of light that we normally use is about 4 to 500 microns. Therefore, 4 to 500 nanometers. Therefore, changes below 0.5, 0.4 microns actually have to be proven to be true because the light microscope can fool you. So we were lucky to collaborate with one of maybe a handful of people in the world who can do this, Graham Knott, where we were able to do this imaging in an awake behaving mouse, the imaging of stretches of dendrite on a single neuron in the brain before and after, and then prepare the animal, prepare that slice of brain, that block of brain for serial electron microscopy. Here's about 100 micron length of dendrite imaged under the 2-photon microscope, sent to Graham Knott in Lausanne, imaged under his microscope and reconstructed, and every spine you see here can be seen in the electron microscope, every single spine. So this is the first time that Graham tells me such a long length of dendrite has been imaged in vivo and then reconstructed. And we were gratified that our size measurements match. So then, you never know till you know. So we have this phenomenon that if you induce spine-specific plasticity by a protocol that is a spike timing dependent protocol, very reduced, very precise. We know exactly which spines are being strengthened because we know their receptive field, we know where we put the spot, we know which ones should be expected to fatten, and they do. Surrounding them are spines that have reduced in size, and the two, therefore, cooperatively make the neuron's receptive field shift. It's not homeostatic plasticity. Classic homeostasis is a synaptic scaling mechanism where what goes up then over time comes down so that the total inward current into a cell is constant. This is cooperative in that what goes up stays up, what's around it comes down, so that over a stretch of dendrite, there has been a shift in the response. Okay? So that some receptive field locations have been strengthened, others have been weakened, and this makes the neuron's receptive field shift. How can this be possible? So a molecule called ARC is a historically activity-dependent molecule. It's been known for 20 years that ARC, or ARG 3.1, is upregulated under the influence of electrical activity, or in a classic experiment that Kwon did when he was a postdoc in Suzumoto Negawa's lab and collaborated with us, he made the ARC GFP knock-in mouse, whereby he was able to show that when there's only 50% of ARC expressed in certain neurons that also express GFP, these neurons had broader orientation tuning. And this was a kind of a puzzle. How can it be that a molecule that's upregulated by activity nonetheless may have a role in countering the effect of activity? And so it has taken the field a long time to realize a very subtle and a very beautiful idea. ARC is upregulated by activity, but its function is to counter the effect of increased activity. So ARC is endocytosis, AMPA receptors. It is expressed at the base of active synapses, and it has the function of countering hyperstimulation of synapses or of organizing other synapses such that the effect of a potentiated synapse can be countered by the surrounding synapses. And this idea is called an inverse synaptic tag, that ARC in slices or in vitro has been shown by Hiroyuki Okuno and Haruhiko Bito that it is targeted to inactive spines in cultured neurons in order to counter the effect of activation. So therefore, we hypothesized that ARC, when you potentiate a spine, ARC leaves that spine and goes to a neighboring spine and removes AMPA receptors from these spines such that the net strength of synaptic drive across these spines is preserved. AMPA receptors are inserted here, and they are removed from here by the action of ARC, which binds to inactive CAMK2 beta. So this model makes three predictions. ARC should be present in spines that have not been potentiated but are going to be depressed. ARC should co-localize with reduction of AMPA receptors. And when you block ARC, you should block this phenomenon. You should block the receptor field shift. I'll show you that each of these three ideas holds. We can measure ARC in a living, awake, behaving mouse by inserting an ARC probe. And we can also measure glutamate receptors or AMPA receptors by inserting a glutamate probe into neurons. And this just shows the imaging in vivo in an intact animal in which we can induce this spike timing plasticity. And here's what we find, that when a spine increases in volume, it has lower arc. When it decreases in volume, it has higher arc. And spines that increase in volume have, of course, higher AMPA receptor density, and spines that decrease in volume have lower AMPA receptor density. So there is this inverse correlation. Potentiated spines have less arc and more AMPA receptors. Depressed spines have more arc and less AMPA receptors. So surface AMPA receptors and arc density show opposite changes after plasticity. When you knock down arc with an shRNA and compare it with scrambled injection at a single neuron, remember, all our work is at a single cell, hundreds of spines on that one neuron. When you knock down arc, the distribution of AMPA receptors is destroyed. Otherwise, AMPA receptors go from potentiated to depressed spines over just neighboring spines on dendrites, over tens of spines. And so in the control, you have already seen this before, the LTP spines are surrounded by LTD spines, but when you knock down arc, this relationship is destroyed. And alongside the receptive field shift is destroyed, so that this arc knockdown prevents the spatial organization of structural LTP and structural LTD spines and the receptive field shift. So that arc is crucial for the synapse-specific organization of neuronal plasticity. And finally, if we knock down the CAMK2 beta, remember arc binds to CAMK2 beta in order to translocate from potentiated to depressed spines and thereby remove AMPA receptors. When we knock down CAMK2 beta, then the distribution of ARC is removed, arguing that ARC does bind, at least in part, with CAMK2 beta in order to be translocated and implement this locally coordinated plasticity. So far I've shown you that cortical circuits are profoundly influenced by electrical activity during development, patterned activity, which we believe is the driver of activity-dependent, synapse-specific plasticity that shapes circuits during development, while spike timing-dependent plasticity induces locally coordinated synaptic potentiation and depression, and such plasticity crucially involves ARC translocation to depressed spines and ARC-mediated AMPA receptor regulation. What does this have to do with brain disorders? We think it provides important insight into how we might think about brain disorders, particularly autism spectrum or classes of psychiatric disorders. You've just heard from Takao. How much time do I have? 5 minutes. Okay, great. Yeah, thank you. You've just heard from Takao that if you close an eye during a critical period of development, then responses in the visual cortex to the closed eye are reduced and responses to the open eye are increased. We have known this for 60 years. The issue is why. And so you've also heard that each eye's input drives pyramidal neurons and also drives inhibitory neurons. And the interesting thing, implicit in Takao's talk, but now I'm going to make it explicit, is that the drive to the inhibitory neurons due to the closed eye makes this neuron less driven because the eye has been closed. It makes this neuron less driven because the eye is closed, but because the same input drives the inhibitory neuron, therefore the activity of this neuron gets restored within a short period of time. Does everybody understand this? Okay? Therefore, the critical driver of change in this neuron cannot be the amount of activity. It has to be the pattern of activity. Okay? I want you to just remember that. That's why I'm telling you about ocular dominance plasticity close upon spike timing dependent plasticity, because it's not fully appreciated how important spike timing is in explaining and understanding ocular dominance plasticity due to closing one eye. People think it's because you have reduced the amount of activity. Not so. You have reduced the pattern or the quality of activity. Okay, so what does MD or monocular deprivation do? It does two things. It does what Hubel and Wiesel described as binocular competition, but we now have a much richer understanding of competition. The first thing in a mouse that when you close an eye happens is that the drive to a neuron from the closed eye reduces. No problem there. But then if you keep that eye closed for a few more days, something interesting happens. The drive to that same neuron from the open eye increases. Nothing has been done to that eye. That eye was open, has remained open, yet the drive from that eye increases. And we call this feed-forward plasticity. This is an example of Hebbian plasticity. You close one eye, the eye drives neurons less, therefore this response is weaker. But then there is this feedback plasticity, whereby the open eye ends up driving that same neuron more. And this is reminiscent of the locally coordinated plasticity that I've just told you about. Okay, and I'm going to argue that there are similar ideas that now sort of go across these multiple kinds of plasticity. So then, the idea is that AMPA receptors are likely removed from closed eye synapses, and AMPA receptors are likely inserted at open eye synapses, and the two together, feedback plasticity, is cooperative with feed-forward plasticity and acts on distinct synapses to implement response changes. And this is a much richer view of what Hubel and Wiesel described as binocular competition. The two eyes we thought were competing for synaptic space. They are not competing at all. The world writes into the neuron, and the neuron writes back. And the two together shape the response of the neuron to closure of one eye. So the model we have of ocular dominance plasticity is the induction happens due to reduction of inhibition, and Takao and Mike Stryker and Josh Trachtenberg have led our understanding of how parvalbumin neurons are critical players in inducing the effect of closing one eye. But then the implementation of plasticity, the fact that the closed eye drives a neuron less, has to do with AMPA receptor removal, and the fact that the open eye drives this neuron more has to do with AMPA receptor insertion. So implementation of plasticity requires AMPA receptor removal and insertion, and we submit molecules like ARC are absolutely essential for this process, similar to the locally coordinated plasticity that I've just told you, induced by receptive field-induced spike timing dependence. So then, what does the loss of ARC do? The loss of ARC prevents all of this plasticity. If you take a ARC knockout mouse and you compare it with a wild-type mouse, if you close one eye for a short period of time, you see a reduction in drive in the wild type, but none in the ARC knockout mouse. And similarly, there's no reduction of glutamate receptors compared to wild type. In wild type, there's a big reduction of glutamate receptors driven by surface expression of AMPA receptors at deprived eye synapses, but not so in the ARC knockout. And similarly, in the ARC knockout mouse, if you let the monocular deprivation go for 7 days, when there is this increase in response to the open eye, there is no such increase. There is no reduction nor an increase of response. A reduction to the closed eye, increase to the open eye. So that ARC is crucial for bridging feed-forward and feedback synaptic plasticity. And this leads us to ask, what does this have to do with brain disorders? Everything. And let me take you through in the last 2 minutes as to why I think so, and what do I mean. So psychiatric and neurodevelopmental disorders have strong genetic risk factors. You'll hear a lot about this tomorrow from Dan Geschwind. There is a major effort in multiple fields, in the field of schizophrenia, in the field of autism spectrum disorders, through GWAS studies, through exome sequencing studies, to screen larger and larger samples, and more autism risk genes are being identified. If you go to the Simons Foundation Autism Research Initiative gene list, which does the most up-to-date listing of genes, the listing from today is 901 genes have been associated with autism spectrum disorder with varying degrees of confidence, with 1 being the highest and 5 being the lowest degree of confidence. If you just look at the genes with high confidence, there are 86 genes. And we think, and Mustafa Sahin and I have speculated on this, and others have speculated on this, that there might be 400 or 500 genes that might be implicated in autism spectrum disorder. What are we going to do with these genes? What are we going to make of them? Can we form unifying hypotheses about their function? We think we can by reflecting on what these genes are. A very large majority of these genes are expressed as synapses. Not all. There are many genes that are involved in regulatory phenomena. There are many genes that are involved in membrane phenomena, such as SCN2As, the sodium channel genes. But a very large majority are expressed in synapses. And importantly, a significant number are induced by activity. If you look at the genome, maybe 3% of the genome is transcribed in an activity-dependent manner. 3% of the 20,000 or 25,000 genes. Maybe 10% according to Eric. If you look at the gene list of genes implicated in autism spectrum disorder, it's at least 50%, if not more. We think that feedback mechanisms are an important target of autism genes, and a very influential review by Dick Chen demonstrated the kind of thinking. When you induce change in a neuron, the neuron does alter its synapses, but doesn't just sit there. It has to alter many other things so that it doesn't fire away in the ceiling. So if you increase the strength of some synapses, the neuron senses that change, and just like a thermostat in this room that is keeping the temperature of this room at a certain level, there are thermostats in neurons that dial down other activities such that the firing rate of a neuron is kept within a certain narrow, astonishingly tight range. And these are called feedback mechanisms. And there are many such feed-forward, homeostatic feedback mechanisms. And we think that I've just shown you that one such mechanism is ARC, acting on local stretches of dendrites. And I'm going to show you very quickly that several autism and neurodevelopmental disorder genes also are part of such feedback mechanism. So it's an interesting thing. They don't respond, these genes form the very subtle but very profound response of the neuron to a change-inducing stimulus. What do I mean? There is a lot of evidence that ARC itself is connected to multiple neurodevelopmental disorders characterized by synaptic and circuit defects, and ARC demonstrably is part of such a feedback mechanism. Here's a gene called STAT1, and it is regulated by monocular deprivation. It's also one of the autism risk genes. STAT1 is upregulated by closing an eye. When we knock out STAT1 mice have abnormally enhanced open eye responses after MD. So I remind you, if you close an eye for 4 days in a critical period mouse, the closed eye drives neurons less, but then after 7 days, the open eye drives neurons more, the same neurons. In STAT1 knockout mice, the closed eye drives neurons less, all right, but the open eye within 4 days is driving neurons more. So it's an enhanced, faster feedback response. Therefore, the function of STAT1 is to dampen this neuronal feedback response. And this is what the surface receptors look like. Here's a second gene, and this is my last data slide, and then I'll close. There is a region in the human genome called the 16p11.2 region. It's arguably the most common region that has been correlated with autism spectrum disorder. It's a 500 kilobase region in which there are 29 protein coding regions. There is one 5 protein coding region that is expressed in a Belgian family that also is within the 16p11.2 region. It's been very hard to figure out what do these 29 genes do within this copy number variation. In fact, CNV deletions are very hard to analyze in human mutations. There is one gene that caught our eye. It's called major Walt protein. And we think major Walt protein is a key modifier gene within this microdeletion region. What does major Walt protein do? Major Walt protein acts in a feedback loop with STAT1. It represses STAT1, and it inhibits STAT1-mediated transcription. And Jacques showed that if you have MVP heterozygous mice, then this open eye response is blocked compared to wild-type mice. So it's the opposite of what the STAT1 deletion is, and indeed MVP acts through STAT1, because if you knock down STAT1, then you can rescue the effect of MVP knockdown. Okay, so in conclusion, I've shown you that closing an eye is a powerful model system within the critical period for looking at mechanisms by which synapses can be depressed and other synapses, adjacent synapses, can be potentiated. And there's a host of molecules that we now know are involved in just this, just the potentiation of the non-deprived eye. These are two completely separate mechanisms, though they are related through phenomena like ARC. So feedback mechanisms are specifically regulated by many molecules. And surprisingly, and it's not escaped our notice that many of these molecules are made by microglia or are expressed by microglia. All these red molecules are molecules of the immune system, and this is through work of Carla Schatz as well as Mike Stryker and our own lab. And finally, we think that many, many genes, all genes of autism spectrum disorder or syndromic autism that have been studied in this model affect this mechanism, the feedback mechanism, and these are the ones in red. And we think, therefore, there may be something to the idea that while there are hundreds of genes of autism, at least those that act at synapses, they have a very specific, very subtle, and very important role. They act in this feedback pathway to counter the effect of a plasticity-inducing stimulus. So in conclusion then, ARC is required for feed-forward and feedback plasticity induced by monocular deprivation. It bridges these forms of plasticity. And feedback plasticity, the plasticity in neurons as a response to change, is synapse-specific and cooperative, and its dysfunction may be a central component of autism spectrum disorders. I want to thank the various people in my lab who have contributed to these studies, people who used to be in my lab, and our collaborators. Thank you very much.
The first part of the talk, that the rules or findings you have that nearby to a spine, that one get the potentiated and the other one get a depotentiated or depressed. My understanding is it's based on the stimulation of the specific receptive field stimulation. I wonder, and I don't know what, I think it's still controversial whether this spine organization on the dendrite is clustered, having same receptive field, or it's more randomly distributed. So the rule...
we know the answer. It's not controversial.
Okay.
It is randomly distributed.
Okay, so it's
right.
Okay.
There is no evidence that all spines on a dendrite have the same receptive field. Zero. In fact, one of the great interesting mysteries of circuit organization is how much heterogeneity there is on dendrites. Both location of receptive fields, eye-specific drive, they're highly random. And we think one reason is, potentially, post-hoc reason, is that if you strengthen one, then others decrease in a cooperative way to make the neuron change, be it for eye-specific drive, be it for receptive field, or for orientation specificity, which I haven't talked about.
Okay.
So even orientation selectivity is randomly distributed on dendrites.
Right, so I was wondering whether you haven't seen a cases that just that because it's random, two nearby spines that have the same receptive field, and whether there are any signature that in that case, there one lose and one win, or?
No, they both increase, but if randomly it is possible that two spines next to each other have highly overlapping receptive fields. They both will increase. We have such evidence. But then within 40, 50 microns, there will be a concomitant decrease as well. Yeah.
I want to continue that discussion about heterogeneity within the dendrite. So, you know, the paper by Chen and Connerth showing within the auditory system.
That is a very similar idea for orientation tuning.
Right, right.
Yeah, where This
So
is a
my
paper
question
that shows
is-
that mouse visual cortex neurons, adjacent spines, have wide range of orientation tuning.
Right. So my question is, how much dynamics can you expect? If you have attentional effects, you might expect that you could get changes on a very rapid time scale. But you're also talking, and you and Takao have been talking about changes which are taking place over a much longer period of time. But have you thought about the time frame?
Oh, yeah. Attentional effects are changes of gain. So you don't need to do synapse-specific changes. You change the gain of drive through altering, let's say, parvalbumin neurons. If you reduce the activity of parvalbumin neurons, you will raise the iceberg, and the orientation selectivity of the neuron will not change, but the neuron will respond to all orientations more. That is the classic orientation, the attention effect on a visual cortex neuron.
So
it is well explained by
considering-
non-specific changes of gain.
So considering that you're technically wizard, can you try training an animal doing an awake behaving prep and looking for changes in the strength of individual spines?
It is possible. In fact, we have a project since Takao mentioned, and I thank the organizers for putting me after Takao so that I don't have to explain everything that he said by myself. Mike Stryker's work on locomotion is that locomotion facilitates recovery from long-term monocular deprivation. If you close an eye for weeks at a time, then you open up the eye, the animal has gone past its critical period, the eye does not return to driving the neuron. But if you put the animal on a treadmill and you show it a certain stimulus, the response to that stimulus will recover almost to baseline, which means there is a synapse-specific effect of that stimulus gated by the cholinergic and, in my opinion, also the noradrenergic system. We are very interested in this phenomenon because we think this is a particular way by which the inhibitory system that Takao described, that cholinergic systems act on, but also there are cholinergic and adrenergic receptors on neurons, on excitatory neurons, and the two work together in order to mediate spine-specific plasticity. It's a long answer, but it's a very subtle question, very interesting question.
We'll just take one last question, then we need to move to the... Yeah.
Okay, well, I had two questions, and I think they're related, but maybe that's too long, so you can choose which one you want to answer. So the first question is sort of following up on this issue about the spines. So if the spines are randomly distributed in that way, then presumably you could shift the responses no matter where you put how far you put the flashing the light outside the receptive field, that you could move the receptive field.
You have to put it within the Gaussian of inputs that a neuron receives.
Okay.
Otherwise, you are outside of its space of possibilities.
Okay. And then the second question is about, do you think in the autism-related argument that if we assume that argument is correct, that these kinds of changes are impacting sensory systems in which you're showing these kinds of plasticity phenomena, and that's cascading into the more complex symptomatology that we see? Or would you say that there may be specific deficits in these type of plasticity mechanisms in higher cognitive areas?
So the sort of broadest answer to this is that by extension from these experiments, a deficit in autism is improper learning by neuron-specific mechanisms that cannot consolidate that learning. That's the way I think about it. The consolidation of learning involves a Hebbian change and a coordinated response such that that change is implemented, strengthened, cemented. That does not occur because a crucial step in this, a somewhat unexpected step, is missing.
Oh, over here. I was wondering if the arc is expressed in interneurons or in the neurons that are also being innervating the motor neurons.
Arc is expressed only in excitatory neurons.
Thank you. Yeah, so next we'll move to data blitz section. The first speaker is Jan Fisher from University of Oxford.
All right, so I'm going to get going. So I'm Jan, and I'm going to tell you today about neuroserpin and specifically its role in neonatal hypoxic ischemic encephalopathy. Just to go back a little bit, we need to go back to the subplate, and Zoltan has touched on it previously. It's a transient structure in the fetal brain, and only a small remnant exists at birth in humans. And interestingly, the subplate has been found to have secretory functions, and one of those molecules that's secreted is neuroserpin. Now, what's so interesting about neuroserpin? Neuroserpin is an inhibitor of the tissue plasminogen activator and has been shown to have neuroprotective properties in stroke and adult mice models, specifically by reducing ischemic damage, improving functional outcomes. It has been suggested that it does this by a variety of processes, starting from reducing inflammation, reducing excitotoxicity, or even reducing oxidative stress. Here I've just shown the broad expression pattern of neuroserpin in the brain. As we can see, it's expressed in the subplate remainder here in the layer 6B, in layer 5, and in the hippocampus here, as well as some expression in the thalamus. However, neuroserpin expression varies during development, and here I've just shown some representative heat maps. Just to quantify this data here, the density of neuroserpin expression peaks at P8 in layer 6B, and it peaks in layer 5 at P10. Contrary, in layer 2 and 3, it increases throughout the observed period, which is P4 to P14. It's interesting that it peaks at P8 just because this is kind of the period that is most equivalent to human neonates or the human perinatal period. Just a brief overview of neonatal hypoxic ischemic encephalopathy. It's one of the leading causes of morbidity and mortality in neonates worldwide, up to 1 million affected every year. And it's essentially due to a lack of oxygenated blood reaching the brain, most commonly during the perinatal period. We're using the Rice-Venucci model to look at this in the mouse. And here we stained for TTC, which is a mitochondrial marker, so an absence of staining, i.e. the white, shows ischemic damage. Some areas are simply more susceptible than others, particularly the hippocampus and the striatum are particularly susceptible, whereas the thalamus and the cortex are less so and are only affected at late stages. We're using a neuroserpin knockout mice. However, there's no significant difference in this aspect between the knockout mouse and the wild type. Neuroserpin specifically in hypoxic ischemic encephalopathy, again comparing the knockout and the wild type mice, there's only very marginal differences, also attributed to the wide variation seen in the Rice-Venucci model. However, particularly interesting is that in neuroserpin expression, particularly in the subplate, there is more on the control side, i.e. the side that has no ischemic damage in comparison to the ligated side. Now, this can be attributed to subplate vulnerability to damage, as it has been suggested that the subplate and other lower lying areas are particularly susceptible to ischemia. Finally, moving on to human neonates, just a sample temporal lobe. Here we can see neuroserpin expression in all cortical layers. And if we just zoom in a little bit here, we can see that it continues to be expressed in the lower layers, particularly in layer 6 and layer 5 at this 38 post-gestational weeks embryo, or fetus, sorry, shall I say. And this very closely matches what we see in the P10 mouse, suggesting that the model we're using is a valid model for replicating what we see in the human. And with that, I'd like to thank the lab, our collaborators, and our funders.
I'll just say about, so do you think the neuroserpin expression in humans represents an intermediate developmental time point of mouse?
Well, mouse continues to develop kind of postnatally, so I'm suggesting that it kind of is equivalent to P8, although obviously in P8 mouse there's a kind of a strong remnant of the subplate. Yeah.
Because I mean, there's this idea that some functions in the adult human brain remain in a more immature state. So I'm wondering if in the human brain the expression pattern matches a more immature state that you see in mouse.
It's something I haven't considered, but it's certainly something I'll look into. Sorry. Good, thanks.
The next speaker will Gervasio Batista from Harvard University.
Good evening, everybody. Thank you for staying so late. So today I'll show you some preliminary data on how the brain may be regulating its sensitivity to thyroid hormones during cortical critical periods. So let me first tell you that around birth and across species, there is usually a peak in the synthesis of thyroid hormones. So circulating levels of T4 and T3 is particularly high during this perinatal period. And if you have too much of these hormones or too little of these hormones, the brain fails to mature correctly, and cognitive, sensory, and motor defects appear, and they are maintained during the whole life. And recently, it's been shown that the peak after birth of thyroid hormones in chickens is required for the onset of the critical period for filial imprinting, and it's necessary for its underlying behavioral plasticity. So we started to wonder whether in mammals enhanced TH signaling is also part of the network of events that underlie critical period plasticity. So instead of looking at circulating levels of thyroid hormones, we are interested in how the brain can locally regulate TH signaling, and basically the brain can either change the expression of the enzymes that locally convert T4 into T3, the transcriptionally active hormone, thyroid hormones. Also, it can regulate the availability of TH receptors, and also regulate the expression of transcription cofactors in different brain areas that would make sense for a brain that has different critical periods at different times, as Takao was mentioning before. So I will map how the genes that encode these elements map into pre-critical period stages during the peak of the critical period and post-critical period stages during the critical period for ocular dominant plasticity in B1 that has been mentioned before. The first thing that we notice is that the enzyme that locally can degrade T3 is downregulated at P28 in the visual cortex of mice, which may lead to an increase in local availability of T3 to start T3-dependent transcription. And as you can see there, after the critical period is closed at P150, there's a decrease in the expression of diode2, the enzyme that can actually convert T4 to T3. This would be consistent with a high TH signaling during the critical period. Then we looked at the receptors. In particularly, we find interesting the alpha receptor of thyroid hormones, which has two variants. One, alpha-1 variant, that when it binds to T3 is transcriptionally active and increases T3-dependent transcription. And also there is another variant, alpha-2, that does the opposite. It actively downregulates T3, suppresses T3-dependent transcription. And interestingly, we found that at P28, at the peak of the critical period, there is a downregulation of the repressing variant of THR alpha receptors. And after the critical period is closed, the alpha-1 variant is downregulated as well, also contributing, in theory, to an increased TH signaling during the critical period and decreased after the critical period. We didn't find any differences in MED1 and mCore, two cofactors that can suppress or enhance T3-dependent transcription, but we are kind of digging in other cofactors to see whether there is some changes over time. So now we wanted to see whether this would also apply to another critical period, the one that Takao also mentioned in A1, the one for reorganization of tonotopic maps. And just to make this story short, because I have only 30 seconds, we found that there is during the peak of this critical period, there is an upregulation of the expression of DIO2, the enzyme that can locally in A1 increase the availability of T3 to increase T3-dependent transcription. And we find also that the alpha-2 variant is downregulated at the peak of this A1 critical period. We also find that both co-regulators of transcription are down-regulated, and we are interested to understand why is this happening locally in A1. So we think this is important in the context of psychiatric diseases, because many of disorders or conditions such as autism, Alzheimer's, schizophrenia, and even depression has been linked to fluctuations in thyroid hormones levels. But we think that maybe during development there are environmental and also genetic insults that can disrupt the local regulation of TH signaling that could be informative about the origin of many behavioral and psychiatric disorders. With that, I would like to thank my lab and the Evangeline lab, and thank you for your attention. Any questions?
Which neurons express the receptors?
We're working on that. We are doing some in situ experiments, and we are interested in looking into that. Yeah, I could see in the list of genes that you showed there is some changes in DIO2 and also the beta variant of the receptor, of the thyroid hormone receptor, that if there is something there. We are mostly interested in PV and whether PV expresses these receptors or enzymes, but we are going to look around.
Thank you. The next speaker is Jack Ip from MIT.
Good afternoon, everyone. My name is Jack. Thank you. I'm a postdoc at Merganka's lab. To follow up on Merganka's talk, I will go into an additional paradigm to examine the mechanisms of locally coordinated plasticity, which is eye reopening after monocular deprivation. Just to recap here, we hypothesize that potentiation, Hebbian potentiation of a spine can lead to heterosynaptic depression of neighboring spines. And this mechanism is mediated by immediate early gene product arc. So the paradigm I'm going to talk about is a more physiological paradigm as compared to the STDP plasticity induction paradigm. This is our experimental design. We use postnatal day 25 animals. We use single-cell electroporation to deliver the plasmid that express calcium indicator, GCAM6S, and structural filler, mRuby2. And after the electroporation, we suture the ipsilateral eye. When I say ipsilateral eye, it means ipsilateral to the imaging hemisphere. Then we wait for 4 or 5 days, and then we image the electroporated neurons to get the baseline. and then we reopen the eye and wait for 6 hours. Then we re-image the same dendrites again. So the top left panel shows you the procedures of single-cell electroporation. Basically, we do a craniotomy, bring the pipette containing the plasmid under the 2-photon scope, go near the cells in V1, and then we electroporate and deliver the plasmids. So this is how it looks like after 2 days, expressing GCAM6S and mRuby2. And we also use intrinsic optical imaging to make sure the electroporated cells are right in the binocular zone of V1. And this is how it looks like after 6 days of electroporations. And after eye reopening, animals were subjected to eye-specific functional imaging. Functional response to drifting gratings for each eye was recorded. So for example, spine 1 here responds to both contralateral eye and ipsilateral eye. Spine 2 here responds only to contralateral eye. Spine 3 here responds only to ipsilateral eyes. That actually goes back to the questions of heterogeneity. So it shows that in these short stretches of dendrite, you can see a very heterogeneous of response to specific eye inputs, eye-specific inputs. And we can score or classify the spines by this ocular dominance index, as reflected by the formula here. So based on our models, the ipsilateral eye responding spines, after eye reopening, they will increase in size because we suture the eye and then we now reopen them. And then, and this can also lead to heterosynaptic depression of the neighboring spines that are responding to the contralateral eye. And this is mediated by ARC. And this is actually what we saw. So as shown in this analysis here, the ipsi eye responding spines, they increase in size. And there is a distant effect here. Within 10 microns of the LTP spine or potentiated spine, we see a higher density of depressed spine. We also use a molecular probe, ARC-EGFP probe, expressed under the natural ARC promoter to track the distribution of this immediate early gene product ARC. As you can see in this graph here, for the spines increase in size, they have less ARC. For the spines that reduce in size, they got more ARC. This is our model. Plasticity or activity induce production or translation of ARC locally or in the nucleus. And then, but they don't translocate to the potentiated spine. But then they will translocate to the depressed spine. So because the depressed spine got like a high concentration of inactive CAMK2 beta, and this is shown by our collaborator, Okuno and Bito. They show that ARC has a much higher affinity to inactive CAMK2 beta. When we say inactive CAMK2 beta, it is not calcium bound, and the calcium bound active CAMK2 beta, they have much lower binding affinity to ARC. And ARC would go to the depressed spine or inactive spine to remove AMPA receptor and lead to structural LTD there. Okay, lastly, I want to show you some of my ongoing work. Using this new technique, magnified analysis of proteome, or MAP, which has the capacity to linearly expand any tissue by 4-fold, we can visualize synaptic proteins like ARC or other synaptic markers like presynaptic marker bassoon or postsynaptic marker HOMO1 at a super resolution level by using a conventional confocal microscope. And you can see the beautiful localization, co-localization, but non-overlapping of presynaptic marker and postsynaptic marker. And you can even see the donut shape of the PSD as marked by HOMO1 immunoreactivity here. This is quite similar to what other scientists observed in using EM. And we can also observe ARC. And we can do a 3D reconstruction. And you can see that this red immunoreactivity or reconstructed volume is PSC95, and the green is ARC. So the idea is to correlate the expression of these proteins with in vivo two-photon imaging. And my ongoing work is to use this technique to identify novel molecular candidates of locally coordinated plasticity. And this work is done in collaboration with Kwang Hong Chung's lab at MIT. Thank you.
So the translocation of ARC seems sort of magical. And what do you think? Do you think it's an active process? It's stochastic? Does it involve cytoskeletal remodeling? Or how do you think that process occurs?
So there's some evidence that ARC can be locally translated. So one possibility is when activity comes in, it stimulates a local translation of ARC right at the dendrite shaft. And then when ARC is produced, it will try to go to find the binding partners because inactive CAMK2 beta has a much higher or stronger binding affinity. So ARC will get accumulated in the inactive spines, but because active CAMK2 beta has lower affinity, so that's why they get repelled from the active spine. So that's how we think it happens. So another line of ongoing work, we're trying to develop a probe for ARC mRNA so that we can track the translocation or the production of ARC mRNA as well, so that we can have a clear picture of how this happened molecularly.
I just had a quick question about synaptic spine shape. You're looking at volume, but are there any shape changes? Like, does the spine shaft narrow? And a related question, you show very nicely in Sudhir Ridanka the picture which is in the bottom center here.
This one?
Yeah. But there must be cases where you have two adjacent spines that are similarly tuned, let's say, to the ipsilateral eye, in which case you don't want to have suppression of the adjacent one, presumably.
Right. Then,
so have you been able to analyze the selective tagging so that
So for the analysis we did, we mainly focused on the single isolated spine. So we didn't focus on the cluster spines. I think that would be interesting. And we can look at it like in this paradigm. And the first question is the shape. Oh, you're right. Yeah, we just look at volume. We didn't look at shape. But I agree we can look at the distribution of mushroom spines or filopodia or stubby spines. So, yeah, we didn't look at it, but I think we should in future. Thank you.
Thank you. Yeah, so for the next discussion session, we would like to invite the speakers today to come forward to sit in the chairs at the front. And so we're basically using this as more like a panel discussion to encourage dialogues between the audience and the speakers. And yeah. And yeah. Su Yun Lee and I will be moderate this. Yeah. Oh, only have two microphones? Okay. Yeah, I guess maybe just a quick opening the dialogue. And the first session, we heard a lot about the connectomes and of the mammalian brain and the cross-species study. And the second session, we heard many beautiful molecular mechanisms underlying the cell type specification, migration, plasticity. So I guess one of the question we could start with is how the speakers from each session could see how the other
field could help to cross-fertilize the ideas and how we understand the brain development from both an evolutionary and a developmental perspective. Yeah.
So you want us to say something?
Yeah. Yeah. Sure.
Well, what I was pleased to see today, and I hope to see more of, is the ability of, or people doing experiments that allow translation of findings from one species to another. And the biology of one species is not simply reflective of the same biology, not identical in another species. There are similarities and differences. The challenge is that I'm always getting jealous of my mouse colleagues that, where the molecular tools are so much more developed, that we also need to learn how to translate molecular tools from one species to another to make this possible, and to be able to generate then general principles of brain function and what is unique or different about each lineage, or humans or some other lineage. And I think that's going to take some investment, and I'm glad NIMH is bringing us here together to at least have this kind of discussion. Oh, I covered it.
Unless somebody else wants to say more about that. So my question is about the learning that we heard about in the last session, and so it's mostly for Takao and for Mriganka. I'm going to read a little bit more into a couple of aspects of what you said maybe than you intended, but I just want to see if it sort of resonates. In Mriganka's case, you mentioned that for the closed eye experiments, it's not the amount of activity, but it's the patterning of activity. And if I read a little bit more into that, I'm thinking maybe it's, if you want to study learning, maybe in the high-level visual cortex, you have to take into account the temporal dynamics of the stimulus structure as well. Otherwise, you're in this rather false regime of getting the spike-dependent plasticity. This doesn't really match the way the circuit has sort of been trained for the whole life of the animal. And before you respond to that, and in Takao's case, he also mentioned that opening or taking off the brakes can be enhanced, but when the animal's running, when the animal's getting cholinergic input or maybe other things through that sort of thing. So what I'm wondering is, are we making a mistake by trying to understand learning in some paradigms by having very artificial, like flashed stimuli and contrived stimulus paradigms, when in reality the conditions in which animals learn are when one has that sort of natural rhythm of natural experience, and maybe even when one is actually actively interacting with the environment? Is that reading too far into some of the things you were saying?
Go ahead, Takao. Okay. No, I think that this idea of EI balance and these lifting of brakes is useful for adult plasticity, and that the regulation of the brakes could be very much activity dependent. So what I didn't have time to talk about, unpublished work, is why perineuronal nets around inhibitory neurons, which are so sparse, would be so strongly related to closure of these critical periods. For one thing, it turns out that synaptic input onto these cells is also plastic. And so having a net around them to physically trap those boutons might be one reason. And in fact, Mriganka talked about MD for 4 days or 7 days, these slow processes. The very first synapse we find that changes as soon as you close the eye is thalamic input onto the PV basket cell. Much, much earlier than any changes onto the excitatory cells. And this might be one reason why they're so pivotal. The nets come on, and that plasticity is blocked, essentially. What we're finding is that the activity patterns of the PV cells, which is famously linked to things like gamma oscillations, can actually regulate the net itself. And so if you can find a regime of neuronal activity that keeps these dynamic structures weaker, you might get more plasticity. And running might be one condition. Other conditions might apply.
My response to that is that I don't think there's any question that the actual stimulus or learning paradigms have a great impact on human learning. And in fact, there is no such thing as non-specific learning. This goes back to Thorndike and how school systems should be structured and so forth. The advantage, perhaps the beauty of animal models, is that under the right conditions, they allow you to abstract away into something that might be an essential component of a mechanism. And I think there is a danger in over-interpreting animal models as really representing the human condition. I don't think you meant that, but it's one of my pet peeves that people study autism models by studying social behavior in mice in this and that context, and I think that should be seen a little bit skeptically, as I think actually a recent NIMH panel under Josh Gordon has actually recommended that one should treat an animal. It even goes on to say that animals are experimental systems. They are not models of anything. So this is a great extension from your point, but just extending into an important point for this symposium on psychiatric overtones of plasticity or development. I think one should be aware that an animal in which a ASD gene is mutant is just an experimental system. It is not a model for a human disorder, and it should be seen that way. But if we are lucky, we can abstract away into some electrophysiological or perhaps molecular kind of component or substrate of the disorder that, if we are lucky, will apply to humans.
Can I say along those lines, the thing that struck me today when I heard in your talk was, about this idea that long-lived animals with more neurons in their brains might be the ones that are going to be more subject to find mental health disorders rather than these short-lived mice. And I was curious to wondering, I'm not sure, what the folks at NIMH would think about that, since the funding probably, the majority of funding is going to, in terms of non-human funding, is going to mice. If they're not going to acquire these psychiatric disorders, somewhat along the line you're just saying. Or other speakers.
Short of studying humans directly, what are you going to do? And so the GWAS studies, and we will hear about that, I think, from Dan Geschwind, are revealing more and more genes, complex, multigenic substrates of our complex psychiatric conditions. It'll be a very long time before there will be good animal models of these, if ever. Maybe if Paula is here, maybe the very early stages of development of multigenic dysfunction could be captured. But other than that, we need to be very creative and think about what we should do once we have this genetic information.
I'll add to that. I think it's one more very good argument for the use of primates for research, because one of the key differences between primates and all the other mammals is exactly that you have all these extra neurons, let's call it, for a given brain volume. And with that, you get not only the longevity and the longer time to assimilate information and get to that point where maybe things start getting critical and any differences will have an impact in behavior, in mental activity, or whatever you call it. So you certainly get an experimental system with a primate that you cannot mimic with a mouse. You can, of course, try to understand what is it that when you introduce that modification in the mouse, you actually get something that behaves like a primate or now becomes susceptible like a primate, but it's, of course, not a substitute for the real thing. Since I have the mic, Mriganka, I was wondering, when you think of ARC in the context of autism spectrum disorders as learning disabilities of sorts, how do you put that together in your mind with the well, let's call them the really good learning capabilities that savants have. Is that incompatible? Do you think that this happens in particular circuits or?
So I am not an expert on savants or most of autism spectrum disorder. I do not know whether the hyperfunctionality, domain-specific though, of certain individuals, certain autistic individuals, and I use this term because there is some debate as to how should people with autism, particularly high-functioning autism, should be referred to. I don't think this is necessarily hyper-learning.
But it must involve learning.
It is a spectacular capability in one domain. It is not generalized to all domains. This one domain, how does it get to be so prominently expressed and represented? It is a unique question, but I think it is not necessarily a question of just learning in that one domain. It is a particular set of circuits that are on the manifold of acquiring knowledge about that domain. That is a very vague answer.
I guess one other way to put that question would be, when you introduce these ARC-related modifications in a systematic way in the animal, would you find, I understand you're looking at visual system manipulations, but is there any indication that you do find this kind of alteration in LTG, LTP in any part of the cortex?
Oh, yes. I mean, Kwan has done experiments in which in ARC knockdown mice or in heterozygous mice, there are significant deficits in motor learning or in many other things. There are deficits in many other kinds of learning. Our experiments are extremely singular. In one neuron, we are knocking down ARC, but you have mice in which ARC is removed generally, and they in fact have schizophrenia-like symptoms or autism-like symptoms. And this was on one of my slides, but I went over it quickly. This has been studied by many other people, in particular Kwan himself.
I just wanted to make a comment. I would like to play devil's advocate in the comments about the usefulness or not of, for example, mouse models, rodent models. First, I'm not sure. First thing is that these are spectrum disorders. There's several traits. I would agree with you that you cannot discuss an animal model necessarily as equivalent to the disease, the disorder. But I think you can talk about the traits and you can talk about the underlying mechanisms and circuits that may underlie those traits, and I think it's useful in that sense. And it is something that we can do now. It's not theoretical. So that would be my first comment. My second comment would be that the question would be, what would you expect the mouse to express so that you would be convinced that that trait is equivalent to that you see in the disorder? So I think one of the strongest arguments that I've seen, for example, is that you can find particular substances that produce behaviors in humans that are very similar to the traits that are observed in disorders. And if you now use those drugs in animals, very often you can replicate aspects that affect the same traits in the animals. And to me, at least, that seems to be valuable information that could open some insight into this disorders. And third, very often the genes that have been associated with disorders and are associated with the traits are also associated with the traits in the mouse. Again, that's a pretty strong correlation. So I would like to support that. This reminds me a little bit, this was the arguments that we used to have when Seymour Benzer was suggesting that we could study learning in flies, and there was a big resistance to that. And I think that we have been there before in this argument.
I can
probably. Can you hear me? Yeah. Mrganka was bringing up a really interesting point. You guys were talking about the savants, and I was kind of curious. It kind of reminded me that there's probably a 3 to 4, maybe even substantial increase in the prevalence of synesthesia in people with autism, and especially relevant to Mrganka's early experiments with Mrganka and Doug Frost and others who were misrouting, thalamocortical inputs, various ways of doing that during development. Kind of harkens back to that, and then of course that's come much farther over the last 15 or 20 years. I'm just wondering, Mrganka, if you have any thoughts on that or could think about mechanistic ways of exploring that within some of these mouse models. It would seem that that would be actually something that you could look at with regards to the impact of autism risk mutations on the prevalence of synesthesia measured electrophysiologically or with fMRI and in model systems. And synesthesia is strongly related to autism, just in case, in general, outside of autism.
So I think these are interesting intersections with various of the talks that we have heard. I think that we heard from Larry how much intercortical, interareal connectivity there is, interregional connectivity. My own interpretation of synesthesia is that there are existing connections that might normally die away or in other people might get severely reduced, but that now are carrying cross-modal information. And we heard from Larry, even the primary visual cortex gets input from 30 different regions, including there is reasonable projections to the auditory cortex, in fact, in mice at least. So there may be various degrees of pruning or lack of pruning in multiple genetic conditions, and hence mouse models that we will discover with the right tools and with these large-scale methods. And those models will be appropriate structural. We'll have the information to then probe these models for the functional bases.
I think Takao had one comment on this as well. Yeah, go ahead. Why not?
Yeah, so we did that study in mice, Dan, that you were asking about, I think. So testing whether there was incomplete pruning in mouse models of autism, for lack of a better word. Well, we took the BTBR model, we took SCN mouse models, and as was already mentioned by Larry and others, the cross-modal input to visual cortex remains much stronger in those mouse models. So it's consistent with an incomplete pruning. We've also done a study with synesthetes, humans, to see whether they have a retention of non-native phoneme discrimination capabilities, and they do. So it's consistent, at least on the surface, with an incomplete pruning situation. Non-native phoneme discrimination, something that infants are losing by 4 to 6 months of age by not having exposure. All kinds. We can talk offline, but yeah, not autistic necessarily.
Okay, we're kind of getting over maybe one final question, if that's okay.
Well, this is going to be a comment.
Comments are even better.
Sorry. So linking some of these, I'm a program officer at NIMH, so at the risk of speaking for all NIMH extramural funding priorities, linking what Mrganka has said with these comments about traits and even with what Dan's talking about in terms of synesthesia, for example. I just wanted to say one way to think about this is, what do you mean by a trait? So what we're interested in is how do we link between the very fine-grained molecular mechanisms you're talking about to the microcircuit dissection, to the circuit-level projections that Larry's talking about, and then beyond to what somebody said as the most difficult thing, right, which is behavior. And maybe there's an electrophysiological thing in between there. So then the question is, when you're talking about behavior, when you're talking about the behavioral phenotype, what are you talking about? Are you talking about a DSM diagnosis that you're trying to model and the phenotype is that diagnosis? Or are you talking about a trait or a phenotype or a behavioral action, such as where is your visual receptive field, which is much more fine-grained? Or are you talking about anxiety-like behavior, like elevated plus maze? Or are you talking about, performance in a very specific way, in a very specific context that you can measure at the same temporal resolution that you can measure the electrophysiology and the molecular changes. And so I feel like from a systems circuit neuroscience level NIMH funding priority perspective, that's really the nitty-gritty where we want to get into. And because the DSM diagnoses are so non-fine-grained as phenotypes, we're hoping to push people a little bit away from those.
I'll pass it down right. I just want to say when I heard that, I wanted to speak up to say that I think maybe the statement that you can't model something about humans using non-human animals was too strong of a statement. And I think you can model things to a certain degree, and there are things I mentioned in my talk about being along a continuum, closer to humans, less closer to humans. But I think people might interpret that incorrectly. I mean, I don't think you thought that nothing happens in non-humans. Okay.
I think the major point I was trying to make is that when a disorder is a very human disorder that involves subtleties of human social interaction, that one should not be modeling it in a simple-minded way. But of course, the RDoC criteria, going beyond DSM, speaks to circuits, and the more homologous the circuit in a mouse and a human, the more relevant it is to study it in a mouse. If there is a visual retinogeniculocortical pathway that is demonstrably homologous in a mouse, and that's the pathway I know, I study it in a mouse because I hope that it will apply to human beings. The more subtle, the more sophisticated the behavior, the more I believe the bar is high to show that you are studying homologous circuits. When a mouse recognizes or does not recognize a conspecific in a 3-chamber assay, I'm going to really go out on a limb and not make too many friends here. I don't know whether that is the same as a person with autism avoiding eye contact in order to avoid interaction with another human being. And I think that the NIMH panel recognizes this. I'm not the first person saying this. So the more homologous the circuit, the more relevant is the mapping. And my plea to NIMH is to be eclectic and be sort of fund a bunch of different things, of course.
Okay, that's great. So let's stop it there. And thank you so much, all of you, for these wonderful talks today, and to the Data Blitz speakers as well, and to the audience members staying till 6 o'clock. That's amazing. Thanks so much, and see you bright and early.